So many of the different fields in science, most people aren't exposed to in their day-to-day life. The more you can get out there and communicate to the young people coming up that are the future of science, the better. You never know what's going to grab one individual, and so that's vital to inspire the next generation.
The Nobel Conference brings together students, faculty, and members of the general public to explore revolutionary, transformative, and pressing scientific questions and the ethical issues that arise alongside of them. We bring leading researchers to Gustavus, including nearly 100 Nobel laureates. We're trying to explore the ideas of science using all of the liberal arts, so the arts, the humanities, the social sciences, all of them working together.
There are a lot of people who are not scientists who like to share in the adventure of discovery and to be able to share that joy with people who also get it, that there's something really exciting and cool going on. It just makes it just an enormous amount of fun.
There's sort of a festive atmosphere, which makes it part of a broader experience.
There aren't that many forums for us to have the discourse at that level with that sort of range of expertise, yet still focused on one sort of overarching topic.
One of the wonderful things about the Nobel Conference is it does give people the opportunity to ask people questions in an informal way. And you never know what statement somebody makes or what interaction that happens will make a change in somebody's life.
It's always very, very encouraging to see other colleagues of yours who are at the top of their game, and it makes you want to lift your game.
As you can feel the audience, and you can really feel when you've helped people to know something that they never knew before. It's really a feeling of discovery that you share.
Hi everybody. My name is Tane Danger. You are tuning in this morning to day 2 of the Nobel Conference at Gustavus Adolphus College. This year's conference is all about insects, little body, big impact. I am so excited to be back for this second day as one of your co-hosts for the live stream. And to help get us started, I've got 2 amazing guests, 2 of the people who are most directly involved with having put this year's conference together, who are going to help us kind of review a little bit of what happened yesterday and get set for the rest of today. But do you mind each just sort of, because you all both have like several ways I could introduce you, so I'll just let you go ahead.
I'm Lisa Helke, and I'm a very good bread baker. Oh, you didn't mean that. Okay, sorry.
I mean, honestly, did it involve some sort of like mealworm flour?
I have yet to try cricket flour in bread, but that's an experiment that is yet to be tried, and I will certainly do so. More appropriately for this context, I'm the director of the Nobel Conference. This is, I believe, my 8th year in that role, and I'm also a professor of philosophy at Gustavus, and secret fun fact is that, not very secret, I'm a Gusty grad, and this conference was really influential on me when I was a student. In fact, I tell people my dissertation topic came out of a Nobel Conference when I was about a junior or senior and took notes and never knew a single thing I was writing down.
Huh, wow.
Yeah.
Fascinating. Margaret.
I'm Margaret Block-Kazi, and I am chair of this year's Nobel Conference. And I'm also a professor of biology at Gustavus.
So yesterday, big day, we got 3 amazing presentations, 2 panel discussions. We started out with a big talk on sort of why do we care about insects? What is the sort of like...
What's the buzz?
What's the buzz? Ding,
I'm
ding,
here all
ding.
week, folks.
Yeah, take a swig of your coffee with that one. So we started with that. We moved into the sort of life and world of flies. And then we ended up, we had a presentation that talked about sort of some of the ethics around social aspects of bees in particular was a lot of the last piece that we talked about. So again, just as sort of a recap so people remember these pieces, but let me ask each of you, you were there in the room, you were moderating the panel discussions with each of those. day after, how are you feeling? What are the things that are really still sticking out to you, you're thinking about from yesterday?
You want to start, Margaret?
Sure. We, I think, really explored this idea of, we know that insects have brains, but do insects have minds? And what is the meaning that insects make of their environment? And why does that matter to us? Both in very practical ways, but also in ways that influence how should we treat insects? Can we love insects, and how might we love insects? So everything from very pragmatic to really kind of big ideas.
I will say, this is entirely true. There was a fly in my hotel room last night, and it was buzzing around all night. And I did not try and do anything to it, in no small part because that was one of the challenges that came out of yesterday, was like, think differently about these creatures that are around. And so, you know, the fly and I just hung out last night. So again, small first step maybe towards what this conference is driving us towards. Lisa, what did you take away yesterday?
I would echo what Margaret was saying, and also observe that the speakers really gelled really quickly. I always remind people that these are not people who normally go to conferences together and talk to each other, and yet they were quickly really modeling what we talked about. I talked about with the audience a little bit about the importance of disagreeing well. And we saw, moments of disagreement and moments of coming together. For me, that's always really one of the exciting things. In a time where good disagreement is perhaps at a premium in our society, for me, the idea that we are modeling for, not just for high schoolers, but for ourselves and each other, what it looks like to disagree well.
Can you just say another word about that? Because I'm a big fan of the Nobel conference, and I do think that this is one of the things that's very interesting. But a lot of folks who are maybe not connected to academic conferences or whatnot might not realize, like, what do you mean by, like, these people wouldn't normally be together? Aren't there a lot of things like this?
That's a great question. Yeah, and we know that there's only one thing like this that's called Nobel, but it's also true, and I will let Margaret speak from her own experience at conferences, but it is not the case that there are very many academic conferences or even sort of para-academic conferences where an anthropologist and a plant geneticist and a philosopher will be engaged in scholarly discussion with each other. It's really extremely rare. It's a thing that people comment on year after they've been here. And I'll tell you, it's a little bit of a high wire act. Like last night I said to Jessica Ware, oh my God, when you said collection of ganglia, I thought, why didn't I know that word? And it's because I'm a philosopher, for heaven's sakes. But we who are used to being professionals, used to being articulate about our disciplines, it is very scary to me to sit in front of people and say, you know that one part in the insect's body that you said is in between the head and the abdomen? Like, what's it called again? Right? It's hard for we who have gotten a PhD in something to be ignorant. Yeah. And that's a really exciting thing, to watch my colleagues be intelligently ignorant with each other and listen. Yeah, that was long. I'm sorry, Tane.
No, that was perfect. Anything you would add about the...
Yes. So
that's good. The improviser in me is like, correct answer.
Yes, and.
Yes,
well
and.
Yes, and. I think to really understand an insect, one cannot just listen to a biologist, or just listen to an anthropologist, or just listen to a philosopher. I think that when we hear these different voices, different perspectives, people coming from different sets of experience, that's when we really get the depth. And so, yesterday's processional,
right,
with jazz and dance and a light show, like that's a way of experiencing an insect, and each of them have truth to them.
Right.
And so that coming together is so powerful. And I think that that's part of what the liberal arts is about.
Right.
When we look at these big pressing challenges of our time.
And this is actually something that I'll brag for our live stream audience that not everybody necessarily gets, is that we got to have a couple of conversations over the lunch hour yesterday with a professor of religion from Gustavus who's leading a session today about sort of what the Hebrew Bible talks about in terms of insects. And we had the director of the Schaefer Art Gallery come and talk about the exhibition that's happening there, which is inspired by this year's Nobel Conference. And again, these different things all coming together. If you are here on campus, you're seeing there's art all over the spaces and the buildings.
Yeah.
And I will say, one of the pieces when we were debriefing towards the end of yesterday was this conference already, we knew that we were going to learn a lot of science about insects and how they operate, and we got a lot of that, and it has seemingly very much been challenging us to think about our relationship to these creatures, to the world more generally, to nature, sort of capital N nature. And I'm wondering, so that was a lot of what we heard yesterday. What are we looking forward to today? Can you give us just maybe some high-level sort of things that we might be listening for today?
Today, we're going to focus a little more on insect-human interactions. So part of what yesterday was about is trying to understand an insect a little more on insect terms. So today, it's going to be about entomophagy, so eating insects as a source of nutrition, about our attitudes towards insects, and it may be surprising to learn that human attitudes towards insects really differ in different parts of the world. And then we're going to learn more about the value of collecting. So it's not just a sixth grade science project. Right. It really has value and importance today. And finally, we're going to learn about lonely flies and what we learn about how flies in isolation teaches us something about ourselves and how our bodies function in isolation.
Yeah.
I'd add a couple of slices to that. One is that those of us who have never really understood what a model organism is, with that last speaker, we'll be learning about that from a guy who knows a lot about it because he won a Nobel Prize for his work on fruit flies and their circadian rhythms. So I don't think he'll probably talk so much about the circadian rhythm research today. But he will talk about what do we mean by model organism, or what do we mean by basic science. I'd also say one of the earlier talks will explore some really exciting, extremely preliminary research about malaria involving mosquitoes and involving something that has been identified as occurring naturally in a certain population of mosquitoes. And given the devastation of malaria, this is really an exciting, hopeful solution.
I am super excited about these presentations. And then I will say, not just because you're sitting here, I love the panel discussions, getting these folks up on stage, which you two moderate. And you noted yesterday we started to see some healthy but intellectually honest disagreements between some of these very smart people. I think we're probably going to see more of that today, which is, again, if you're somebody who sort of revels in seeing people actually kind of bring their full selves and not just sort of recoil into politeness, like I think that we're going to get some very interesting conversations today. So I would highly encourage folks, stick around for the whole thing. Thank you both. We are actually, I'm going to cut this because we are going to go back down to Londorino because we are getting started this morning with the Gustavus Wind Symphony, which is going to be kicking us off right here at about 9:15 and doing a prelude, before we go into our first talk, which will start at 9:30. So without further ado, thank you both so much. You all are going to go down back to the arena and get some of this started. We are going to go back to Londorino, but stick with us throughout the day. Join us on the social media. Send in your questions and enjoy day 2 of the Nobel Conference here at Gustavus College.
Good morning. First of all, thanks to the Gustavus Wind Symphony for that wonderful performance under the baton of Heidi Johanna Miller. And some of you probably got to learn a little bit about that last piece. Thanks so much, Heidi, for making that happen here, the monarch migrations piece and the discussion yesterday that enabled us to hear from the composer. So in addition to them, I'd like to this morning thank the indefatigable Marketing and Communications Department at Gustavus, including Neely Prenslow, Ann Volk, Sydney Noel, Steph Ash, Luke Hattlestead, Tim Kennedy, 25 student workers in the marketing department who have helped with tickets, managed the learning lab and workshops, and created content for the live stream and promotional emails. And with respect to the live stream, thanks upstairs to Matt Dobozinski of our marketing office and Gustavus alum Tane Danger, who's serving as our online MC in conversation with science journalist Maggie Kurth. Don't forget to submit your questions to ushers in the room or via the email at Nobel Conference, one word, at gustavus.edu. And as always, have a conversation with somebody sitting next to you at lunch or across the room from you. Just keep the conversation buzzing. I'm sorry, I had to do that once during this conference. And speaking of things that had to happen once at this conference, I am happy to report that we have filled the international requirement that botflies be discussed at least once per insect conference. Last night at the amazing Moth Story Slam, the winning story involved a St. Thomas student, I believe a St. Thomas graduate, who studied abroad and, yes, came home with a botfly on her hip. That she told a hilariously terrifying story about having this creature incubating, I guess would be the right word, right, on her hip. And she won the slam. I'm also happy to report that I was not able to tell a story because there were so many entries. We almost broke the Minneapolis-St. Paul Story Slam record for number of submissions. We had 22. Their all-time record is 25. It was a rollicking evening, and people came out of Bjorling Recital Hall to see the outside walls of Bjorling turned into a giant movie screen, courtesy of Eric Leonardson, a 1992 grad, who showed amazing videos of insects, everything from beautiful ones to more challenging ones. So it was a really wonderful thing, and thanks so much to Eric for doing that. All the time I pray to Buddha, I keep on killing mosquitoes. That's from Isa. At this point, I'd like to introduce my colleague in the Modern Languages, Literatures, and Cultures Department and the Department of, and in the Program of African Studies, Pascal Chor. He will introduce our next speaker.
Good morning. I am greatly honored to introduce Dr. Segenet Kelemu. Dr. Kelemu is an Ethiopian scientist noted for her research as a molecular plant pathologist. She has served in key positions in high-profile science organizations and has received numerous international awards. Just to name a few, she received the 2014 L'Oréal UNESCO for Women in Science Award. The People's Republic of China gave her the Friendship Award for her contribution to China's economic and social development. The Academy of Sciences for the Developing World gave her an award in 2011. Dr. Kelemu has published widely in refereed journals and other reputable academic locations. Her scholarship includes publications on edible insects, prediction of breeding regions for the desert locust, and genetic diversity. Dr. Kelemu is the director of the International Center of Insect Physiology and Ecology in Nairobi, Kenya. One of their research activities focuses on the well-being of bees. They want to find out what makes African bees more resilient, more tolerant of diseases. This research would have an impact beyond the African continent. Dr. Kelemu's research helps us understand how plants endure so many challenges: drought, herbivores, pests, climate change. She wants to keep farmers from devastating losses by better understanding the symbiotic relationship between plants and insects. She believes that insects play a great role in our lives, that they are a great contributor to food security, for instance, by being big pollinators, but that they also have damaging impacts. They destroy crops in pre- and post-harvest periods and transmit diseases to humans, animals, and crops. Please let us welcome Dr. Kelemu to the podium.
Yeah. Don't have for the slide. No? Okay. Okay, until I get it for the slide. Okay, good morning, everybody. Good morning. Okay, thanks. Thank you. Okay. Thank you very much. So English is one of the most tortured languages spoken around the globe in so many different accents. So please prepare to adjust your understanding of Ethiopian accent, influenced by other languages, weird and non-weird languages I speak. So I thank the organizers for this opportunity, for inviting me to this flawlessly organized conference. It's really a great pleasure to be here. So before I go into my main talk, I want to talk about why insects. I say this because people often ask me, what do you do for a living? I say, I'm a scientist. Oh my God, then you are too smart to talk to. Then what exactly do you do in science? I said I run an organization, research for development organization, focused on insects. You mean the entire organization focused on these tiny insects? I say yes. So they don't ask what are insects, because everybody knows insects, right? Then the next question is why insects? So with a tone, with a connotation that why do you waste millions of dollars and effort on these tiny useless insects? So I think the organizers, they coined this insects, little body, big impact. It's a beautiful title. Thanks, Margaret. So I'm going to tell you today that really big impact is really true. And I'm going to give you some examples. But why insects? Because many of them are beneficial. There are estimated 10 quintillion insects, the most abundant animals on Earth. But the vast majority are useful. Only a small fraction are harmful. They are great indicators for environmental change. They are beautiful in the landscape. They decompose waste that we generate. And without insects and microbes decomposing this waste, actually, our planet would be a really stinky mess. And they are also extremely hardworking companions, hardworking on the farm, pollinating lots of our food. And they themselves are also good in the food web. Like I think the speaker yesterday told you how many insects up to 800 million tons are consumed by spiders alone. And birds also alone consume up to 500 million tons of insects annually. Just imagine that. They also give us fabric through by silkworms. But there are also other roles insects play. The insects, nobody said yesterday about the role of insects in forensic science. When detectives try to determine a homicide, when a person died, often that insects are used in a decomposing body to determine the time of death, based on their life cycle of the decomposing maggot. Also, insects have been also inspirational in designing new equipment, medical equipment, based on wasps, for example. Wasps have a special way, structured like a needle that goes into their predator, their prey, to, in a sophisticated way, to go in and deposit eggs. So a medical equipment has been designed based on that structure. They have been also inspirational in designing helicopters. For example, dragonflies, they maneuver while flying at high speed. I think you have seen many jewelries this week, designed, inspired by different insect designs, fabrics and so on. Robots have been also designed based on ants and many, many ways. And I think we can also learn social behavior from social insects, also teamwork, discipline, organizational skill. So there is a lot that we can learn from insects. So I think I have to take this opportunity also to introduce you the International Center of Insect Physiology and Ecology, the only research for development organization, not only just in Africa, but in the world, focused on insects. So this was founded in more than 50 years ago, about 53, a little over 53 years ago, by a Kenyan entomologist, Thomas Eisner. He was the first CEO and served the center for 23 years, followed by a Swiss entomologist, Hans Herren, then a German entomologist, Christian Burgmeister, and me currently running the center, a fascinating center. We are about a little over 630 regular staff from 40 nationalities and some other contracted workers. I think the major workforce, the movers and shakers of the discovery we do in insects and insect-related science are our graduate students. We don't give them a degree, but they are registered in 85 plus partner universities around the globe, but they do their research with us. So a lot of the discoveries are actually made by them. Our mission is to alleviate poverty, ensure food security, and improve the overall health status of peoples of the tropics by developing and extending management tools for harmful and also useful arthropods. This is our mission. We do high caliber, high quality basic science, but we always translate it to impact to make a difference in people's lives. So we use insects, but in around four thematic areas: in human health, in plant and animal health, and environmental health, often in a way to combine this in one health approach. And we have, as I said, a huge capacity building that cut across. We don't do this in isolation. We have more than 300 partners around the world. We are the regional center for Stockholm Convention, regional center for reduction of pesticides. We are also FAO reference center for vectors and vector-borne diseases in animals. and also for the center, collaborating center in bee health in Africa for a World Organization for Animal Health and so on. So you see all this also in our top of the buildings of solar panel. We have made the whole campus 100% solar. So that was one of the infrastructure upgrade I have done, thanks to the government of Switzerland for financing that. So let me just give you context. Context is always important. So let me give you some context that our way of life globally is very unsustainable. It's very destructive. We generate millions of tons of food waste, most of it 60% coming from households. We waste a lot of food. We produce also animals that we farm, produce billions of tons of manure. The brewery industry produces a lot of millions of tons of waste. Thousands of potato chips that we enjoy generate thousands of tons of potato peel, and so on. So to manage that also, waste is a multi-billion-dollar also business, and so on. So we are very destructive. We are wasteful. We are messy in our way of life. So and to also feed a lot of our desire to eat a lot of meat, we use a lot of soya for the feed, for animal feed. And so to meet that demand, a lot of trees are being cut in Brazil to get way to produce more soya as the demand grows. So the Amazon forest, for example, in the last 10 years, about the size of a small island nation, 1,000 square kilometer area of forest has been cleared. About, to make even matters worse, about 36% of the fish that is being fished also is used not for human food, but macerated and produced for animal feed as protein source instead of going to human food. So why am I telling you this? All this I told you and more, we are using the mighty, tiny black soldier fly to clean up the waste, produce high-quality protein, insect protein, and replace soya and fish meal and other protein source for animal feed. That is a beautiful, beautiful way of creating sustainability. So this is a black soldier fly, how it looks like. What I'm showing you in the center is a larvae. The larvae produced on waste, on manure, on human waste, on brewery waste, on potato peel, on avocado waste, on any biological waste. So basically, you use the waste to produce, to rear these insects, black soldier flies, and multiply in mass in a very short period of time. We harvest the larvae, we process them, we feed the animals, and the remaining, the insect frass is high-quality fertilizer. It's the most sustainable way to live our life. And this fertilizer also that we produce, fertilizer price is very, very high in Africa. So it is used in any production system. It is high quality. It's better than the commercial fertilizer. It also improves the soil health as well. And the insects also, during all this process, also, they are a good source of oil. We will never think of insects as a source of oil. The source of, sorry. So I think, yeah, the oil, again, the oil, the cooking oil, the cosmetic industry uses a lot of oil. Lots of the oil also comes from plant-based, is plant-based. So it needs a lot of water, a lot of land, and so on. But the insect source, the insect oil, is also extremely high quality. It has omega-3, it has antioxidants, and you can produce a lot of products. So these are some of the products generated at ICIPE and passed to the private sector. Soap, the omega source oil, the cooking oil, some of this oil also, particularly from the black soldier fly, can be used also as diesel oil as well. And it's absolutely fantastic. You can use also the oil to cook or as lotion. I use some of the oil for my hair. So it's absolutely beautiful. So, but also the other thing that we discovered is when you feed insect larvae to chicken, you can actually change the microbiome content in their guts, and making them more resilient. And also, there is no need to add antibiotics in the feed. So, resilient, disease-resistant, and so on. So, we would like also to see whether we can change the microbiome of people who are on a regular insect diet. We would like to see that, actually. So, this is extremely sustainable. So based on our work, this is not a theory. Now, in East Africa alone, more than 1,000 private sector has been sprouting in producing black soldier fly for animal feed. So all these dots you see in the different countries are where the companies are concentrated. So this gentleman with the jacket is actually the former president of Kenya. He skipped one Sunday from church to visit one of the companies. So immediately he said, oh my God, I'm going to actually do this in his farm, he said. So currently, about 10% of the total requirement of Kenya's animal protein for feed is insect-based, and it is growing. So this is an unbelievable innovation that has been extremely popular. We train a lot of people in this, but we can't keep up with the request, the demand. So what happens then to the plastic waste we generate? So we can clean all the mess, biological waste. So I have been pushing my team to look for also to diversify the type of insects we have for feed. So during that search, they have identified a worm, African lesser mealworm, that can degrade plastic completely to its compound level. So you can see in this graph about 70% survival on just plastic alone, or after 30 days living on plastic. So it's not then they found out that it's not the insect itself chewing it, but they have microbes inside that degrade these plastics. So it is amazing to be an insect scientist. So for food, look at this variety of beautiful delicacies across Africa. So I always say that the next scramble for Africa is not going to be just for its minerals, it is cobalt, it is uranium, it is diamond. It's going to be for the variety of insects that Africa generates, edible insects. So I think any five-star chef would like these colorful things on their plate. It's just really wonderful, the variety. So there are more than 2,000 species of edible insects globally. And about estimated 2 billion people have been consuming insects traditionally in Africa, Asia, Latin America. And more than 550 of them species are in Africa. And this dark spot here is a hot spot of insect diversity in Africa. So what we had done was to do survey in Africa, all the edible insects, and we did chemical analysis to see the the protein content, the mineral content, and so on. So most of these edible insects actually have much higher protein content than the animal and plant source proteins. So many countries also, they are short of nutrition, fulfilling nutrition requirements for their population. So, and we found out that insects are extremely rich in minerals and in zinc and iron. In many remote corners of poor countries, actually women, pregnant women, who need extra iron, they actually eat dirt to get the extra iron they need. So that is absolutely not needed, because a lot of these edible insects have more than the recommended daily allowance of a lot of these minerals. So we have also generated a lot of insect-based products. You can see for food, the iron, the protein bars, the feed products for fish, for chicken, for pigs, fertilizer products, plant also protection products from chitins of insects, and oil products, a multitude of things. So this actually is, we believe, is the future. It generates a lot of income. It generates a lot of products. It generates also jobs. Many jobs are now being created in this industry. So I'm absolutely, really happy that how we are changing the continent and influencing also the world. And this is a program that I established when I first started at the company 10 years ago. And we do this successfully because we work together with governments, policymakers, influencing policy, educating policymakers, politicians, and developing standards and policies that accept this. So currently, Kenya, Rwanda, Tanzania, Uganda, Ethiopia have officially accepted this, and companies are flourishing. And we are working now with African Union to develop that. So, but some countries are not waiting for their governments to approve. So as you can see in this map of Africa, so now 14 species are farmed as edible insects, and there are over 2,300 black soldier fly farmers, producers in Africa, producing the larvae for for animal feed. So this is the fastest growing technologies that we have generated, and the most impactful. And I think so Australia recently funded us, also the government, to take this technology to the Pacific countries, to the islands, with our support to expand that. And so FDA had approved also a few years ago. European Union has approved. So this is the future. We are not pushing it just for Africa, but I think for the whole world, because the whole world needs this. So this whole circular economy contributes to many sustainable development goals. And this is in the environment, in plant health, in animal, human health. And we won the Global Food Planet Prize, which came with $1 million for this innovative research in 2020, December. And we went to Sweden to celebrate it last year because of COVID, we couldn't go, but we spent the money already. And in the last 5 years, our work, not only this, but overall other parts of work also, we won 155 individual and institutional awards. And together, the insects won the award as well with us. So it's an uplifting area to be in insect science. So the second example I will give you is a technology that was developed at ISUPE some 20 years ago called the push-pull technology. This was a technology developed to control a very major insect pest of the staple crop maize called stem borer. It is a very simple approach, but scientifically really complex one. So this uses two companion plants, intercropped, one as a border of your target crop, and the other one intercropped with your target plant crop. So there's a lot of chemistry involved here. So I don't understand it myself, and I'm not going to expect you to understand it. But what was really amazing is that the companion plants, one of them called Desmodium, produces naturally compounds into volatiles into the air. We don't smell it, but the insects smell it, and they don't like it. So they run away from the crop, from that plant, which is push. The border plant, the grass, produces other set of compounds that the insects smell and like it, they are attracted to it. But then when they lay their eggs, they find out that actually it's not, it's a wrong host. And so the egg doesn't hatch, so the life cycle finishes. So this is how in real life looks like. The trap, the push plant along with the maize in the center, and then the grass around is and the border to attract the insect. So both these companion plants are high-quality animal fodder. It's a beautiful system for African crop livestock mixed production system. But the science is fascinating. But over the years, we discovered this system doesn't just only control insects, it controls one of the most food security threats of Africa, striga weed. This is a parasitic weed. These beautiful purple flowers, so looks are deceiving. It doesn't have roots, so they have minute seeds that can stay in the soil for 20, 30 years until they find the exudate from the roots of the host. And they detect it, they germinate, they go, they attach themselves to the root, and they suck the life out of the crop. So all this you see dead. You see all these women bending, trying to hand weed all day long to remove that, but the crop is already gone. So what makes me so happy is that this technology works for this, like in the same way that, in a different way that it works for insects. So you can see this now. This is real. So here the lady is the same lady. Before adapting the push-pull technology, completely her maize is gone. This is starvation, basically. And then in one season, with the technology, you have this healthy maize. So this technology was highlighted by the UN Secretary General as one of the most innovative production systems created ever in science. And I'm so proud it was generated in Africa by Africans. So, yeah, thank you. Thank you. So this technology is now over 15 countries adopted and produced. And one of the most uplifting things is when I go to the field, talk to all these women, and they tell me how this changed their lives. One time I was talking to women farmers in Ethiopia who adopted this, and one of them told me, "Oh, thank you, madam, that this changed my life. They did this and so on." And then she says, "But it's not you who gave us, it is God who gave us this gift." I said, "No problem, I work in partnership with God." So she didn't like that joke, so don't joke about religion. So because this has been successful, and we have developed it now with a $6-7 million grant, we are developing it for vegetables as well, because this is women crop. It has a multitude of pests and so on. We are developing it successfully for vegetables. So this is one of the technologies that can handle multiple constraints, major constraints in production system, insect stem borers, fall armyworm, weeds. It's a fodder plant. It does climate change mitigation and adaptation, soil fertility improvement, because one of the companion plants naturally fixes nitrogen, so you don't have to apply fertilizer. So it really makes me jump, you know. So it also controls aflatoxin, mycotoxins. Mycotoxins are common globally and more predominantly in Africa. So the mycotoxins are carcinogen. They cause stunting in children, but also if it is in high concentration, also it can kill people. So this can solve that also, and so on. So what tells you is that nature works in sync. So you develop one effective technology for one thing, and then it solves a lot of other things. And then I think this is once in a lifetime discovery. And this is a technology that keeps on giving. We still don't know everything about what this does, actually. The last example I'm going to give you is, again, exciting discovery made in our institution. Malaria. Malaria is an old disease. The rest of the world has managed to eradicate it, but I think recently it popped in Florida, I believe. This is an old disease. It has changed history. If you read about the history of Panama Canal, so Panama Canal, the French started building it, but malaria completely devastated their workers, engineers, killing more than 20,000 people. So they had to abandon that project, and the Americans took over, tried to disinfect and stabilize the situation, because they did not know, at that time it was not known that it was mosquitoes that transmitted. But eventually in 1904 or so, Americans discovered that it is the mosquitoes that transmit the disease, both malaria and yellow fever, which are deadly, both of them. And so, but malaria still is a killer in Africa. More than 90% of the incidents are in Africa. So a child even today dies every 2 minutes from malaria, and this is not acceptable. So all the existing technologies, control measures now have plateaued. The mosquitoes are getting smarter. They are biting earlier and outdoor and transmitting and killing people. So we have a major malaria program. We started working. We have many different research areas. But this was basically an accidental discovery by one of the students. So in the area where there is endemic presence of malaria, the mosquitoes that naturally harbor a microbe, a fungus called Microsporidia, cannot transmit malaria. 100% transmission blockage. This was like, are you kidding me? This was a wow moment. So you can see these red ones in the microscope, under the microscope. These are the Microsporidia associated in the insect, in the mosquito. But what was exciting is also that the mosquito, the female mosquito that harbors it, she transmits the next generation through the egg, which is fantastic for large-scale application. And then we later discovered actually it is also sexually transmitted. So the male mosquito that has it can transmit it also to the female during sexual mating. And You can see this red one where the microsporidia is penetrating in the embryo of the female mosquito in the ovaries. And then in the male, it is associated with the male gonad in the ejaculation duct of the male gonad. So it was evolutionarily designed for transmission and long-term also maintenance. So this was great. So once we published this discovery in Nature, so a lot of funding started flying towards our door so to control this. So the beauty of this also is that, for example, for dengue control, Wolbachia, a bacterium, has been used for control of dengue at a large scale in Australia. So the results show, the preliminary results now, that Wolbachia containing mosquitoes actually, you can reduce dengue by 77%, and the reduction of also hospitalization of dengue patients by 86%. So this is great. But the malaria, it is unique to us. So, but this is encouraging for us. Now, what we are doing is that a large-scale trial, semi-field trial, together with the regulators, to actually to determine what environmental factors influence this, how many male infected mosquitoes do we need to release, and what are the conditions we need. So a multidisciplinary team composed of modelers, and ecologists, and entomologists, and molecular biologists are working now, more than 25 people on this. So we are really excited. But I think also the beauty of this also, for some reason, the female mosquitoes, they prefer to mate with male mosquitoes that have this microsporidia, the fungal symbiont. So we are very excited with this. I think we are towards eradicating this toxic disease, killer disease. So thank you. So these are the figures. The figures, female mosquitoes, they prefer to mate with microsporidia containing male. We don't know the reason why. Why do they prefer that? So, but then it will be really beautiful for us for the large-scale eradication or application. So we also believe that for every problem, vector-borne diseases, there may be a symbiosis-based solution. So I think we are towards that major move to control other vector-borne diseases like Zika. You remember Zika? And many other, yellow fever and dengue and all others. So we are really excited. So I'm grateful to all the funders, direct funders to us who make all this possible. So I just hope also that the students who are here, I think it is really fantastic to be a scientist. There is nothing more rewarding than using your skill to change lives, to solve global problems when we have tons of global problems. A few years ago, when my daughter was little, like 4 or 5 years old, she declared one day that, "Mommy, Daddy, you guys are so boring. All you talk about is science, and you work so hard, and you are not even rich. So I will never be a scientist." So we told her, "No, you don't have to be a scientist, but you can choose whatever." But she ended up being a scientist. So she's working now, her PhD on dung beetles. Dung beetles, on the role they do on what is it called, nutrient cycling in African savannas. And I think she's going to be a spectacular scientist. She's really good. So, so she will, yeah. So I encourage all of you to be scientists. That's what I'm saying, or at least work with scientists to contribute to society. Thank you so much.
Yeah, you can do that.
Okay, hi everybody. I'm Tane Danger.
I'm Maggie Kurth.
We are your co-hosts for the live stream here of the 59th Annual Nobel Conference. We just pulled out of our first presentation for day 2 of the conference, which was great. And we were just going to kind of recap or give a quick reaction to that.
Yeah.
Maggie, tell me, like, what should I be taking away from this morning?
Well, I think like one of the really interesting things in that presentation was that she was sort of talking about the ways that insects can be used for food that are not necessarily like us eating the insects. Because I think a lot of people kind of have this like, oh no, they're gonna make you eat insects to save the environment kind of thing. But like she had this really cool ecosystem-based idea of where the insects get used as livestock feed.
Yeah.
And the insects that are being used for livestock feed are then eating plastics.
Yeah.
And like what a cool way to kind of integrate all of these different steps of environmental protection.
I love it. Okay, that was a very quick recap. We are going to throw back to Lund, where we are already getting started with our second presentation for the morning.
Insects. Her recent and delightful book, Edible Insects and Human Evolution, examines the role insect consumption played in the diets of our earliest human ancestors, as well as the role it continues to play in contemporary human societies around the world today. Dr. Lesnick is also known for her work on edible insects as a potential solution to the global food and nutrition crisis that many countries currently face. As our planet's human population continues to increase exponentially, and global food production and distribution are increasingly disrupted by international conflict and extreme weather related to climate change, among other things, meeting this challenge will require us to overcome our entomophobia, or our fear of insects. And this is where Dr. Lesnick has been playing an important role by engaging the public through radio talk shows and edible insect events, and providing expert testimony for government policymaking relating to insects as a safe food source. The title of her talk today is Latitude and Attitude: Environmental and Cultural Impacts on the Perception of Insects as Food. Won't you please join me in welcoming Dr. Julie Lesnick.
Thank you so much. It's just such an honor to be here and an honor to follow Dr. Kellemu. So to start, I actually want to acknowledge land acknowledgements to get us started. At the beginning of the conference, we heard a wonderful land acknowledgement. President Bergman acknowledged the Dakota people as the stewards, as the traditional stewards of the land we are gathered on today. And we even got to hear the words and the song of the Dakota people thanks to Everett Black Thunder. And so the point of land acknowledgements is to try to reverse invisibility. We have this sort of notion that with colonization here, that with removing indigenous peoples off their land and by forcing assimilation through boarding schools, that their cultures have disappeared. And that is not true. There is rich, diverse indigenous cultures. And so the least we can do is acknowledge it. And so simply, anywhere you are here in the United States, you are on native land. And acknowledging it is the bare minimum. It's the start of a conversation. In order to reverse invisibility, we need to provide more opportunity, and we need to do so much more. And so just as a simple way to start, this image here is actually a sticker from a clothing brand, a native-owned brand. And so one of their lines is, "You are on native land." And so I encourage you to paste these stickers everywhere and wear their t-shirts and start forcing more people to have these conversations. But they're just a streetwear business. They have amazing other lines that are not as in your face. So to my talk. To start, I will go over a little bit about edible insects and human evolution, really just have time to touch on two kind of key time points. Then we'll get into the latitude and attitude, which is my work on trying to understand our attitude towards edible insects here in the United States. And then lastly, try to understand the role of insects in the future of food. So starting with edible insects and human evolution. So in my book that has this title, I go through the stages of human evolution, and I try to reconstruct what it looked like, what the insect diet looked like at each of these stages across the course of our evolution. Now, this diagram here, though, that you're familiar with is really not an excellent representation of evolution. It is much better to think of it like this, and even this is a way oversimplification. It isn't just branching, it's more weaving, and a braided river is another metaphor we're using. Continual population, mixture, and integration over millions of years. We don't bucket ourselves into beautifully, easily defined species, and definitely not ones that we can name millions of years later. So one of the issues with this diagram is that the chimpanzees on the left and the humans on the right are both alive today, and that these other stages that it represents are extinctions that happened over the course of human evolution, somewhere, over the course of the 6 million years that both humans and chimpanzees have been independently evolving. And one thing that we have in common, amongst other things, with chimpanzees, is that we both use tools to forage for termites. And so it goes to reason that if chimpanzees use tools and we use tools, that this last common ancestor and across these stages of human evolution, they may have been using tools to forage for termites or at least eat other insects. And so to give you an idea of what that intelligence looks like, I'm going to give you a quick video of a chimpanzee in the Republic of Congo. And so this is Maya, and you'll see she approaches with a stick, and she's going to use this stick, and she's going to perforate open a hole in the nest that's going to lead to a tunnel. And then what you're going to catch here that you might not have noticed is she has a second tool in her mouth, and she's grabbing that, and she's threading it in. And then the termites will bite the end of the tool, and she'll use that as an opportunity to pull them out and eat those termites. So this is a very complex task. It involved two tools. It involved pre-planning. She showed up with both tools. She knew exactly what she was doing. And this gets passed on generation to generation in these populations of chimpanzees. So the one time point I want to start with is about 1.8 million years ago. That we are, at this time, this is the genus Australopithecus, but we also have the origins of the genus Homo around this time. And at sites, it's actually kind of difficult to tell who is who and who's doing what with what artifacts. And so at this time, we are definitely bipedal. We are very human-like in a lot of ways, but we still have a lot of retained, more ape-like characteristics. And so one site is Swartkrans in South Africa, where we have these bone tool artifacts. They're clearly some sort of digging implement based on sort of the polish and wear pattern that is isolated on one end. And they preserve scratches that show that they were digging into something. So researchers had done a bunch of experiments with their own bone tools on different things, like stripping bark off trees, digging in the ground for tubers, digging into termite mounds, and they found the best match of the wear pattern of the artifacts looked most similar to the tools that were used to dig into termite mounds. And so this is really the research that launched me into the direction that I went. I followed up these experiments in my own dissertation research, and I really started looking more at the termites as a food source. If we know these tools were being used, or suppose they were, what can we actually get nutritionally from those termites? So that's really what I focused on. But more recently, at another fossil hominin site, so Olduvai Gorge in Tanzania, right around the same time period, now we're at 1.7 million years ago, We have part of the reason why Olduvai is famous is that the fossils are so well preserved. And that's because of a volcanic ash layer that fell over. So one, the catastrophic death of whatever that volcanic ash killed, but anything that was already there then got extra preserved with this ash. And so that same tuft of ash just beautifully draped over this ancient termite mound. And so we are currently analyzing it. It's amazing, the organic materials and what we can analyze in it. We can name the termites to the genus level 1.7 billion years ago. And we're just still teasing through the data and trying to figure out what other beautiful things we can learn about the ecosystem back then. But we know that hominids used tools that look like they were used to termite mounds. We know they were walking around where the termite mounds were. We don't have evidence they put them in their face, but this is about as best we can do in paleoanthropology to reconstruct what was going on in the past. And so it seems that termites were a valuable resource. So that time period here, now we're zooming out. So this is humans within all of primates. And so we were just up there in the top right looking at kind of the last common ancestor between us and chimpanzees. But I want to shift down here. This is about 50 million years ago. And at this point in our evolution as primates, we were all insect specialists. And so this primate here, the tarsiers, if you notice the branch goes all the way straight down, like there's not branching from it. So tarsiers today very much resemble this primate that lived 50 million years ago. They're different, time changes, but there are a lot of fundamental similarities. So this adorable bug eater here, something just like it, is part of our collective evolutionary legacy. We come from insect eaters. So we know that insects are primate food. We know they're an ape food. We know they're a hominin food. And so today we know that they are food for billions of people around the world. And so looking at this, this is data, this map is from 2015. It is tough to get all of the data in one place. I'm sure it's even been updated since then. But with what we have, you can see that in this data that is number of insect species consumed in the country, that the darkest filled countries, the ones with the most insect consumption, tend to be more equatorial, more tropical. And so when I first saw this image, it reminded me of this. This is a phenomenon we have in ecology called the latitudinal diversity gradient. And it basically is that there is more biodiversity at the equator, and then as you get further away from the equator, it is going to gradually reduce. This is due to a lot of different things, just the quantity of sunlight, different weather patterns, a lot of different factors. It's not super cut and dry, but we definitely see this biodiversity gradient. So I wanted to study this. I wanted to kind of test it statistically to see if I could see this gradient in insect consumption patterns around the world today. So in doing this test, I also added some other variables in there to kind of ask other questions about insect eating patterns. Starting with the basic, to test this gradient, I need to know, the number of insects consumed, but also a centroid latitude or some mark of latitude for that country. And then I looked also at area and population as possibly more space, more people, more chance someone's eating insects as a possible hypothesis. I then used percentage of farmed land because there is this sort of idea that once we became agricultural, insects became pests and we stopped seeing them as food. And so that was sort of an explanation that I'd been hearing about why we weren't eating insects here. And it didn't really hold up to observations I've had around the world, but I wanted to statistically test this. And then lastly, gross domestic product as a measure of wealth. Are wealthier countries avoiding eating insects because they have better choices available to them? And sort of this idea that insects would only be consumed as a fallback food. So going and doing these analyses, what I found is that the significant correlations between insect consumption and these variables existed for latitude and also for gross domestic product. It was very strong for latitude. The logistic regression could predict insect consumption 80% of the time, which is a really strong result. And so we're going to look through that. We're going to look at the gradient a little bit, and then we'll get back to gross domestic product. So in order to test a gradient, the predictions that you make is sort of, if you're eating insects between 0 and 10 degrees latitude, and then looking at insects between 10 and 20 degrees latitude, like those two areas right next to each other might not look very different. You might not see significant differences. You also are arbitrarily making those boundaries. So I tried to make sure in my statistics that I changed that boundary so that I didn't have an artifact of how I measured and cataloged my data. But then you should expect that these groups that are further away, so between 0 and 10 and maybe 30 and 40 degrees latitude, you should expect significant differences between those groups. So adjacent groups shouldn't be different from each other in a gradient, and more distant groups should be different from each other in a gradient. So I'm going to show you my data slide, and to prep you for that, what we're going to look for is there should be asterisks suggesting the significant differences in gray shaded boxes, and the bolded outline boxes should not have asterisks in them. So the bold outlined are adjacent, and the gray are more distant. And so in this ugly data slide, what you can see is that the pattern really holds, but there are just these three. So really you don't see many asterisks in those bold boxes, and the gray have a lot of the asterisks, and that holds up to prediction. But there are these just three little asterisks that are kind of out of place. And so of those 30 paired tests, 3 of them resulted in this. The interesting thing about them, though, was all 3 of those that were kind of outside of prediction fell between 40 and 46 degrees latitude. Now that is exactly where we are today, right? This is the 45th parallel is the transition outside of the mid-latitudes into more northern latitudes. A transitional environment. We know north of here it just gets harsher. And so maybe that's what we're seeing. Maybe we're seeing that there is an actual boundary up here. Maybe insect eating can't happen past, very easily past the 45th parallel. But another thing going on is that most Western countries are in this area. And so Western is a difficult thing to define. It's basically Europe and then countries that have had continual emigration from Europe and long histories of European colonial history. And so in order to test what's going on here, I took Western countries out of my analyses. So again, trying to work with the best geopolitical definitions in order to remove the correct countries. But what I see there then is that correlation with GDP goes away. The latitude correlation sticks, and that there is no relationship anymore between gross domestic product and insect consumption when the Western countries are taken out. So there's just this artifact by all of us being in this area and having such aversion to insects, we're breaking the gradient, right? We've created this sort of artificial boundary in that data. But that doesn't go to explain why we have such strong negative reactions here in the US and across Europe. And so really this comes down to our colonial history. If you think about it, if you think about Europeans, Columbus, 1492, in an environment that doesn't foster a lot of insect eating, traveling across latitudes in a way that had never been done before, entering into the Caribbean and encountering people eating insects, this was something that they had to understand and try to figure out and had strong opinions about. The only real associations they probably had with insects as food was sort of either what animals ate or with spoiled foods. And so to encounter indigenous people consuming insects, they had some strong opinions. And so here is a quote from Diego Alvarez Chanca, who is a companion of Columbus on his second voyage. "They eat all the snakes and lizards and spiders and worms that they find upon the ground, so that to my fancy their bestiality is greater than that of any beast upon the face of the earth." Over 350 years later, US Senator John C. Fremont an actual participant in the California genocide, a government-sanctioned murder of thousands of indigenous people in California. And he is quoted as saying, "Roots, seeds, and grass, every vegetable that affords any nourishment, and every living thing, insect or worm, they eat. Nearly approaching to the lower animal creation, their sole employment is to find food, and they are constantly occupied in a struggle to support existence." And so again, we have this dehumanization, beast-like, lower animal creation. It's taking people that are as physiological, the same as us, we're all the same, we all have the same capacities, and reducing them to something lesser. And very specifically, we know, partially because here we have it directly stated from John C. Fremont, that dehumanization is one of the stages of genocide. We have enough data to know what precedes genocide. We know that multiple things have to produce the right climate, and dehumanization is a critical key component of that. And so to remind you again that you are on native land. We need to recognize that our aversion to eating insects is not about the insects. It is about a fear of looking uncivilized. And that is a definition that was given to us by colonists who wanted to steal land. So moving on to the future of food, which unfortunately isn't going to go much more upbeat. If you didn't know, we're in a global food crisis. So the global food crisis is there are 350 million people suffering extreme hunger. And most of that isn't here in the United States. Most of it we can stay, blind to. But we have degraded our soil. We have drained our aquifers. Our land is not going to continually support the food production we're asking of it. And so eventually this problem is going to hit home, and it's going to hit hard. And we've been fighting it. We've been fighting it hard for a long time, right? We've been buying those green products and trying to make a change. And we're really not seeing those changes. And what we now are learning is that we have this myth that we can vote with our dollar. And it's this idea that if we buy products that reflect our values, then there will be more products that reflect those values. And there'll be more of those positive changes in our society if we just express our voice through how we spend our dollars. The problem is it doesn't work because we're constantly being lied to. Eating organic Doritos doesn't help anyone. It only helps the manufacturer of Doritos. And so we're not giving any data on how we want to see the world look different. We're giving data to marketers so that they know what to market to us because they want to know what we will buy. And so to make change, we have to consume less. And so So one way we can consume less is to consume things that consume less things. And so in 2013, the United Nations Food and Agriculture Organization was the first to put this data together for us, to look at the resource use of traditionally raised livestock, to compare cows and pigs and chickens to crickets. And I'm not giving you any actual numbers here because they're constantly changing and you have to account for different methods, but the trends are the same. There is a physiological difference in these animals. As large mammals, a lot of the food you put into those animals just turns into heat that we release into the environment. But when you scale down, we are more efficient with our heat, and so we get more efficient as you scale down to pigs or to chickens. But then when you get to crickets and insects, they're not losing heat the same way mammals are, and they're so small and they're so efficient. The energy conversion ratio of what we put into insects and what we can get out of them as food is incredibly efficient compared to our traditionally raised livestock. So all of the arguments, though, for the best sustainable ways forward don't change our behaviors, right? We can make the most rational arguments, but we have a lot to overcome. Like, we have this deep history of insects being gross to us, and we don't. And that the colonial quotes, the 1493, the 1800s quotes, still exist today. 2022, as a reaction to this idea of eating insects, Alex Jones says that they're alien to our normal life. This is anti-human and anti-life on this planet to eat insects. So again, dehumanization still going on. We still use this rhetoric. Not only are we making insects less potential of a resource that can make difference when we speak this way, but we're dehumanizing people and contributing to a genocidal environment. And so we need to be careful about how we talk. We just do. And so one way, a great exercise is just to, and we're all here doing it, we're learning to love insects and learning to talk about them differently. And just acknowledging that they are food is a great place to start. The end of this quote I find the funniest, though, the idea that it's anti-life on the planet, because we have all of the evidence to say that that's not true. Almost like every sort of life form eats insects. Across animals, right, we have reptiles, amphibians, birds, insects, mammals, humans. But then even outside of animals, we have carnivorous plants can eat insects. We have fungus that can kill ants. So everything eats insects. Insects are the scaffolding that life is built on. And so when we're thinking about ways to be eating, we need to go back to the basics. We should be looking at that scaffolding. And so one of the most efficient diets is actually what you could call an entovegan. And this is what gorillas do. They have a largely plant-based diet, and the only animal products that they consume are insects. And so if you are looking for the most efficient diet, it would be an entovegan diet. I do not have an entovegan diet. I'm not asking you all to go be entovegan. I'm not even a vegetarian, right? But the truth is, it doesn't fall on us as individuals. If everybody here left today and became an entovegan, we would not see a change in the environmental output in how we're using our resources. This is not an individual thing that we can change. We need new food systems. We need revolutionary ideas. We have this idea of sustainability, sustainable food systems, sustainability, and I use the word all the time. But I've caught myself, though, that if I'm not saying environmental sustainability, the word sustainability can also mean a way to preserve your business to make sure there'll be profits in the future. And so many corporations have sustainability statements, but it's really about how they're going to make sure they still have enough resources so that they can produce their product and make money. And so we have to be careful about sustainability, because what sustainability does is just fixes the little immediate problems in front of us. It's the system that's the problem. And so by correcting the system, we need to dig it out at the root. And so we need to start thinking less about sustainability and more about stewardship. And so stewardship is a relationship with the land. This is responsibly using and protecting the resources of the natural environment. And so on a superficial level, they might sound the same, but they're different here. They're different in how you actually interact with the world. The first one thinks about us. The second one thinks about all living things. When I was a student in anthropology and was taught about animal domestication, I was taught it was symbiotic, that we couldn't live, the humans couldn't live without those animal resources, and that those animals needed us to live. Without us, they wouldn't have the food and the safety, and they surely would die. And this was very mutualistic, right? Beneficial to both of us, really. And it's this idyllic farm, the idyllic farm you see on the green, grocery containers. And so this can exist. You can find this on homesteads. There are people who can call their cows in. They'll come in when they want to be fed, when it's time to be fed. Chickens wait outside the coop to roost for the night. You can see mutualism if you look for it. But it doesn't scale. And I would argue this, if we're talking about symbioses, this is beyond just the predator-prey relationship. Factory farming is parasitism. We had a lot of empathy for that cockroach and the parasitic wasp. And we need to remember to have empathy for the animals that we're going to have on our plate in a few minutes. And so this is a hard system to change, right? We all know, we've known this for a long time, and we just kind of put it outside of our mind because we can't change it. As individuals, there's so little we can do. It needs to be a systemic change. So we need to start thinking differently. We need to start revolutionizing how we think about the future of food. And we can learn a lot from insects. Bees are semi-domesticated. They are literally free range. They are the only thing that are actually free range. They go and they live their lives and they come back to the hive that is safe, and they know that that is there. And so we are providing them something useful. And they are semi-domesticated, though, instead of what we would call semi-cultivated, because we have selectively bred them, right? We have selectively bred the bees that are going to produce more honey, but really they then get to live their lives, and we do have this sort of mutualism relationship with them. The most common consumed insect around the world is the palm weevil larva, and these are semi-cultivated. The palm weevil will lay its eggs on downed palm branches. And so people around the world, anywhere in the tropics where you can find palm trees, there are going to be some palm weevils too. And so if you're going out and you're going to fell the trees, maybe you want fronds or palm hearts or something else as a resource, you can leave some branches on the ground, and if you show up a few months later, you'll have an excellent harvest of these palm weevil larvae. And so the domestication, and especially the factory farming, is not stewardship. But looking at semi-domestication and semi-cultivation, that looks a lot more like stewardship. And so I don't know what it's going to look like, but I think about ant farms, and it gives me a place to start to think about this and what role insects can play going forward. And so one, there are at-home farms where you can farm your own crickets, or you can farm your own mealworms. Now, mealworms are a little more popular because they're quieter. But crickets, I think, are actually a little easier to rear, but they're loud, they sing. But you can farm your own mealworms, and it's not very hard. And so in an ideal sort of, in thinking about what the ideal ento-vegan diet would look like, everybody would have a mealworm farm. Or maybe even beyond, maybe it doesn't have to be ento-vegan. You could have chickens. You can have backyard chickens that you share your mealworms with, and then you have their eggs too. Right? And so this can be done at a household level. It can be done at a community level. It can be done in the neighborhood or the town. And so rethinking food systems to not be global, but instead be local, is a really good place to start. And then I think about what semi-cultivation of ants would be like, and I just imagine that maybe we'll have giant anthills, like we create this perfect environment that ants want to live in. They already want to live in our house and eat our food, so I think this could work. But we could imagine a way that you could have an area of just giant anthills that we go and we sustainably harvest from. There are new ways of thinking, and I think one of the beautiful things about insects is it challenges us to think differently. And so in our food system is one way that I think they could really help us. So we are on native land, and we need to be stewards. But we have so much to learn. Good place to start is to read and listen to more Indigenous voices, to elect officials who will protect the environment before they protect corporations. And so I'm going to leave you with one last quote, one that does not have a dehumanizing statement in it. And it's from Robin Wall Kimmerer, a professor of ecology and a citizen of the Potawatomi Nation. If you have not read Braiding Sweetgrass or any of her other work, consider that your homework. What she gives us here in this quote is what she refers to as the honorable harvest. And the honorable harvest is just a part of her culture. It isn't anything that's written down. It's just the relationship you have with Earth. It's the relationship you have with all living things. We don't have that. So she kindly wrote it out for us so that maybe we can learn and start feeling that for ourselves. Take only what you need. Take only which is given. Never take more than half. Leave some for others. Harvest in a way that minimizes harm. Use it respectfully. Thank you.
Hi, everybody. Welcome back to the live stream coverage of the 59th Annual Nobel Conference here at Gustavus Adolphus College. We just got done with our second talk for the morning. I'm Tane Danger. I'll just go down the line.
I'm Maggie Kurth.
And I'm Amy Cusick.
Amy, maybe just introduce yourself again, because this is the first time you're with us this morning.
Yeah, absolutely. So I am a science professor. I'm in the Department of Biology, and I'm also in the Department of Environment, Geography, and Earth Science.
So that was a very powerful talk. Immediate reactions. What are you thinking?
I mean, the system level thinking is something that I have been thinking about for a very long time when it comes to energy, because it's the same sort of issues. I mean, people expect you to individually as a consumer solve climate change, and that's a complete impossibility, because all of the systems that make it possible for you to live your life, to travel, to have basic levels of functionality in society are built up in a way that you can't just change by yourself one at a time from the bottom up.
Right.
And that is simultaneously realizing that is simultaneously exhausting and relieving.
Yes.
In some ways, it's not your fault, but also now what?
Yeah.
Yeah, absolutely. And I think in both of the talks, I saw some similar concepts. And one of those things is sort of the diminishment of externalities when we think about food production. I think that as Midwesterners, we sort of understand that our landscape is really sort of dominated by two crops, maize and soybean, and that is not going into human stomachs, right? That's going into animal stomachs, and then human stomachs sort of secondarily. And so, part of what I heard people say is there should be more local solutions. And I think we saw local solutions in Dr. Kelemu's talk. And I think we need to understand that for a lot of the world, synthetic fertilizers and pesticides are very, very expensive and out of range for many people. And so when we think about food production, particularly in Africa, we're thinking about smaller farms that are farmed by women, that are feeding their families. And so the solutions on that level are going to look very different than sort of what we're talking about here in North America.
Yeah. I mean, I feel like, again, these sort of big systems questions and then what the interaction, I guess, is for, like, what do we take away from that? Like, I totally hear that, and I feel the same way you do, Maggie, about, it can be both liberating and frustrating to think about these challenges and think about, it's like a system, like it's not, and yet I also just sometimes wonder, like, okay, if that, how do you walk away from that, or what, not walk away, but what do you take away from that? What do we do with that?
I mean, I think like one thing you can take away from it is the fact that you can make, we meaning collectively, like as a society, can actually like make massive changes if you're doing it in a systemic way.
Yeah.
That like systems are things you can harness, not just things that can oppress you, right? And you can see this just as simply as like when you have a city that really takes public transportation seriously, that systemically changes what is available to people and like what choices are available to people.
Right.
And I think you can do the same kinds of things with food systems. It's just a little harder, I think, in some ways, because a lot of this is stuff that has so much of financial incentive behind it, right? But when you see things like what we were talking about earlier, where you have the whole like ecosystem train of a mealworm that eats plastic, that the chickens eat the mealworm, that we eat the chickens, like that's the kind of system that could really, really work for people in some really cool ways, and like open up a whole lot of new opportunities.
I think that was just like a gorgeous example. I would say, so we're getting very close to going back down to the panel discussion, which I am very excited about.
Yeah.
I feel like after yesterday, in particular, there's going to be some really interesting sort of back and forth between what different folks are thinking.
I think there is. I think I wanted to also kind of like talk a little bit about that, like what she was saying about like the cultural dismissal of eating insects too. Because like that's such an interesting, and obviously like troubling, but like interesting problem as well, right? Because we have this such ingrained idea that like this stuff is gross, this stuff is like what happens when you're bad.
Yeah. But I also like the first talk saying, we're not even talking about eating them directly necessarily.
Right. And
like there's all these other things
like that it's a way around that.
There's a way around it.
Like if you can't if you can't like get people on board with that, there's a way around it. But like also like we all ate bugs last night.
Yeah.
I got
and it was super. It was actually really cool.
Yeah, it was.
We had like these really amazing crickets that were from a recipe from Owamni, from the restaurant in the Native Owned restaurant in Minneapolis, and they were made here at Gustavus, and they were delicious. They were so tasty, and the literal ants on a log were also amazing.
Celery, almond butter, ants. And the least exciting part of it was the almond butter.
Right, yes, exactly, yeah. And I think one of the things that I learned is that I am happy to eat bugs as long as they're the crunchy ones.
Yeah. Absolutely. I think that's the least scary.
Although I will say
crunchy
that
bugs are delicious.
They did have a variety of like flatbreads or different pieces that were made with mealworm flour.
Right.
And
great.
the mealworm flour things were delicious as well.
Yeah.
Yeah.
So I agree. There's this old line, culture eats politics for breakfast. And I do think that that's like a really important point to keep in here is that, we can talk about the science and the ethics of this. And there is this like cultural component of like challenging sort of like what our norms, assumptions are.
Right.
That has to be part of it.
But just sort of like where you fit it into what already exists. And like, one of the things that already exists is that we like salty, crunchy things.
Yeah.
Yep.
Salty, crunchy. That's going to get us out of this.
Bar snacks.
Bar snacks.
Bar snacks for the future.
All right. With that, we are going to go back down to Lundarita where the panel discussion is going to get started. We'll be back after that. But again, I'm really looking forward to seeing what our entire panel has to say following these two really amazing talks. So Nobel Conference, panel discussion 1 for our second day here, getting started in just a moment. And we're going to go to Lundarita right now.
a lot outside in hopes that I won't cough in the microphone.
I think if you take also candy constantly,
Wow.
Coat my
So
throat.
different. Yeah, that's amazing.
I think we can reconvene. If you would take your seat,
Now I have to.
we'll begin our next discussion. And as you do take your seat, I want to mention, and I'll try to remember to mention it when we break, there are going to be several emergency announcements, several emergency tests, well, several, I guess 2. St. Peter regularly at the beginning of each month holds an emergency test of its sirens, and that will happen today, I believe, at 1, I think that happens.
Yes.
1?
Yes.
It's 1 p.m. today. And then also there is a national test of the emergency system, and everyone will receive a notification on your cell phone, no matter what you do to prevent it. So just do not be alarmed. We have emergency protocols in place. If an actual emergency should emerge, please pay attention to Miguel, the manager, the stage manager here, and he will tell you what to do, but we're not going to have any of those. We're just going to practice having them, so please do note whether your equipment works or not. I also have been asked to announce that sessions, at the noon time, there's a session listed. Go to the ant, thou sluggard. It's the session about the Hebrew Bible and references to insects in it. It had been going to be held in the sukkah, which has been constructed on the south campus. There has been what is described as a catastrophic event there. I didn't ask for details, but at any rate, it has been moved to the Banyer Interfaith Center, Multi-Faith Center in Anderson Hall. So please go there during the noon hour if you would like to go to that session. So thanks for that. With that, I turn it over to Margaret, who will launch our questioning.
Great. Well, the first question I'll ask is, do our fellow speakers have any questions for Julie and Seganet? Jonathan.
Yes, I mean, thanks very much to both Julie and Seganet for a fascinating pair of talks. I think there was a lot of overlap in the content of the talks and some of the recommendations coming out of them. And it seems as though one big point of agreement between the two of you is about recommending people to eat more insects. And so for some of the reasons I was discussing yesterday, I'm a little bit hesitant about this because it does, in a way, involve recommending to people to consume far more individual animals than they have been consuming in the past. For example, someone might eat 100 crickets in a single sitting. And so it seems very important to be confident that we have the welfare standards right and that we're giving these animals good lives when we recommend that people eat them in such large numbers. And then I'm really unsure because it seems like very little research has been done into the welfare of these animals and how to give them good lives in farming environments. So I'd just like you to reflect on that, really. How does welfare feature in your own thinking about these issues? And what do you see as the most useful things we can do to try and make sure these animals are having good lives?
I have lots of thoughts. So to start, I always find it fun when somebody's like, I can't eat insects, I'm a vegetarian. And so, so I say, okay, that's fine, but it's like, why are you a vegetarian? What is your philosophy behind being a vegetarian? Because a lot of vegetarians are health-based or environmentally based, and then some are literally vegans who believe that harm can be quantified by numbers of lives lost in your consumption. So then eating thousands of insects is more harmful in their eyes than eating a single cow or chicken. And so it really comes down to definition, and those are very philosophical debates and thoughts, and everybody has very personal views on it. But so from the environmental side, if you are an environmental vegan or vegetarian, the insects can provide this really interesting way that makes it easier to get all of your nutrients. Being a vegan is hard. It's not easy to get all of the essential amino acids by pairing. Like, you have to be intelligent in how you pair your foods. And also, it requires a lot of highly processed foods a lot of time. And so in trying to eat better for ourselves and the environment, less processing is good. So there is this little ento-vegan group that I find really has kind of connected a lot of the dots. So yeah, insects isn't gonna, if you are a vegan, then insects might not be for you. The second thing is about the welfare and the killing of the food insects. And this is something that I find interesting and important, is that right now, we mostly freeze the insects. So the insects go into just a natural state of torpor or hibernation. They just kind of fall asleep, and then they stay in the freezer long enough, and they eventually die. So that is mostly how people process it. However, refrigeration is a really expensive cost, energetically and dollar-wise. And so I have fears that when this scales up, that when I came into these ideas 10 years ago, and I had sort of the rose-colored glasses and the idyllic perceptions, to start realizing that when we scale, that we're going to likely start losing that insect welfare piece that is critical to how this movement is starting, but I think that can so easily be lost. And so I do think it's really important in sort of regulations of insect foods going forward to even if it's a labeling issue, so that you can make your decision on how the insects were harvested. I think that's going to be important.
Yeah. Good. I mean, I've got to be honest and say I'm a bit concerned even about freezing insects to death in ordinary household freezers, because that's quite a slow process. Something that is more recommendable, I think, is liquid nitrogen, which does kill them essentially instantly. But as you say, that's even harder to scale up on a large commercial scale. Yeah, so I'd love to hear Seganet's thoughts on this as well.
Yeah. Well, I think, life is always a balance, a choice. So what do we do? Do we care most about eating the insects, or letting people die, or letting people malnourished when there are more than 1 billion people who are malnourished, when they could solve this? I don't want to sound cruel, but I think, my scientists tell me that we think about these issues. So the least painful way to kill the insects is freeze them slowly. But this is also not practical when we are telling people you can raise a chicken in a very low-tech, in your garden, in your backyard. But the vast majority of African households don't have electricity. So are we going to say, just freeze them and eat them? So I think people are not going to care how the insects feel. They are going to eat them, whatever. The way the chicken running around the backyard are digging and swallowing the worms, what they find, So it's a balancing act, really. But I think from the environmental point of view also, when you look at the figures, the efficiency of insects, some of the figures which is commonly cited is to produce 1 kilo of beef, you need 25 kilos of feed and 22,000 liters of water to hydrate the animals. You need 95 times more space. So it just makes sense to have these things. So, but having said that, I'm not saying that eating insects is a silver bullet to solve food security or nutritional security. No, we are not saying that. There is alternative to do things better. So I think what Julie said also in the thing is also, this, we have tried many things. Eat organic. Well, organic, as she said, is not solving our problem. So we have really to think out of the box And it's not this one, eating insects is not even out of the box. It is we are just doing the traditional one, what people have been eating for centuries, animals have been eating for centuries, how we bring the science and do it better. And mainstream it. And I think the only country that I know that have really mastered mainstreaming this is Thailand. Thailand is amazing. So I think, we have to think about those, but we have also to balance what is really most important. So, yeah.
I think that's a really good point. I think pragmatism is what maybe is needed here. I mean, we're in a position of privilege. Our bellies are full. We don't face food security. We were able to eat a variety of animal and products this morning that may not have been killed in a humane fashion. And so we can sit here nourished and have this kind of academic conversation. But I think the real world scenarios that you're describing, where people, I have friends and colleagues who are faced with food security issues in other parts of the world. If they're choosing between humane euthanasia for the soldier flies that they might be eating or feeding their children, they will, of course, choose feeding their children. And so I think we should be thinking about these issues because they're vital. Animal welfare includes insects. Insects are animals. But I think we also have to really understand that we come from a perspective where we are maybe making suggestions, but not necessarily following them. I mean, in the types of food that we eat and the way that other animal products are being killed. I think human suffering is something that we forget about, or it's easy to forget about because we can turn on the television and be distracted, but it is a real problem, food security globally.
I saw Michael Young and then Anna.
Yeah, well, I was going to head in a little bit different direction, but I was impressed that you were able to show the efficacy of trap plants, for example, and particularly Microsporidia in the control of malaria, which is an enormous scourge. But it brought to mind a question that is connected to a great deal of resistance with regard to genetically modified organisms. Because one of the principal worries is that you're creating an organism, it can be free living, it can escape, et cetera, et cetera. But if you're putting Microsporidia together with mosquito, there's going to be somebody that comes along and saying, well, what's the host range for the Microsporidia? Are we going to, how many different types of mosquitoes are we going to affect? But the resistance, you can't go into a grocery store without picking up a box that says non-GMO. And there were 150 plus Nobel laureates in 2021 that wrote a note to the UN, among others, begging for some balance in the attitude toward genetically modified organisms. We've got a long history. I mean, corn, maize is something that we created, and it's genetically manipulated, and in those cases, you're manipulating things that you have no control over in order to get an outcome, which in most cases with a genetically modified crop, you're doing something very specific. Another example that might be even comparable to malaria is golden rice. Yeah. You've got this huge problem of young people dying from vitamin A deficiencies in parts of the world where they don't have access to sufficient vitamin A, and a simple production of a crop that varies by a single gene, that allowing production of vitamin A, can eliminate the deficiency. So I just like to get a sense from this group about how you look at, this is another part of this feature of trying to come up with justifiable ways to respond to health disparities and disease disparities, et cetera.
So it's a good point. So the history, the story of genetically modified crops, I think that debate was lost due to bad communication, bad PR, non-scientific based communication. The environmental group activists won that battle. So first it was sold as a silver bullet that is going to solve Africa's problem, which is false. I have worked on genetically modified crops. I have advocated on that. So a lot of what the advocates really sold was false advocacy. And then the Greenpeace and all the environmental activists came hammering it. A lot of misinformation went into Africa, into others. Yeah, the GM crops are coming. It's going to make you sterile, and you know, the manhood is a big thing, right? So all those things. So it was lost in that debate. And then to make matters worse also, Monsanto was linked to that. So they are going to control your materials, planting materials, and so on. So there is a significant role for GM crops to play in sustainable production system, but that's not the silver bullet. So in Africa and many countries, you know, apart from South Africa and a few countries, and now Kenya also allowed GM crops, that was lost cause. In terms of microsporidia, this is not a genetically modified. This is naturally existing association that has evolved with mosquitoes for many years. We just happened to stumble on it by luck. And to avoid that type of thing, we are working with communities and with regulators so that they work with us through the process together, so that there is no magic to this, there is no modification. We are just amplifying the naturally existing thing. So we have zero doubts that this will be allowed to be released in a couple of years, at least in Kenya and Rwanda also. I had a talk with the president of Rwanda. He's excited by that. He said he's very happy to be the first to get it released, and so on. So I think how we communicate with the public, how we also work with the policymakers, it makes a difference in the uptake of the technologies that we develop here.
Yes, first of all, thanks for two amazing talks. And I guess there were several things I wanted to say. First of all, I think it's important that we, although this is a conference about insects, that we sort of lift our gaze and, like you were saying beautifully, think about sort of the environment, and the stewardship is for the entire environment. So we need to find this balance between feeding the people. I mean, we have a right to be here, of course. We need food, and that should be a fair. I mean, that goes for everyone, of course. While finding a way to coexist with the entire environment, not just the insects, but everything. And what I thought was so nice in your talk was that you both talked about these examples that were sort of very simple with the push and pull technology, and those more fancy tech solutions. And I think regarding the tech solutions and the GMOs and things, like you say, the PR has been a problem, but I think that is partly because there's a risk when big corporations have control of this technology and use it for profit, not necessarily for the good of everyone, like you were both talking about. So I think this is part of it too. And so trying to work from sort of taking 3 steps back and trying to widen our perspective more often. I mean, circularity, like with the soldier flies, that is such a beautiful principle, and we need to try to find more of those solutions that are good in the broad perspective, and not sort of be tempted to dive into this small techno fancy stuff that is super interesting for some business that deals with it, but might not really be good for the greater picture.
Can I ask my question?
Did either of you want to say anything in response to that?
Yeah, I was just gonna point out that some of these large corporations have been controlling seed availability far longer than GMOs have been around.
Yes.
Maybe a solution is governmental. That is, someone like the UN or the World Health Organization has to be a repository, and that those deposits are made available to anyone.
Yes.
And as well as, on the other side, the protection of having a seed bank where the original naturally occurring seeds are, I think there's a place in Norway or Finland.
Yes, Svalbard.
Yeah. And, I think those are essential to have stashed away, a guaranteed way back if something does go wrong. But so far.
Which has already gone wrong at Svalbard, of course. Climate change has meant that many of its vaults have been destroyed by melting.
I just, because it really leads really nicely into the question that I wanted to ask both of you, actually, because you broadly have been talking about food systems. Julie, you mentioned it explicitly, and you talked about stewardship, and right before this I mentioned that's something that I use with my network. We talk a lot about stewardship. And what you just said too, Michael, food systems, I just have to intro this question a little bit so I have context. If you try to look at even a specific commodity, a food system, right? Let's take for India, like we'll take rice, and you start to map the stakeholders involved in this food system. You start really generally with the producers. You can do then the processing and the marketing, and then the distribution, and then the consumers at the end. Within each of those levels, there's so many different stakeholders. Like you mentioned, there's the seed companies, the fertilizer companies, the pesticide companies, the farmers, then the transportation services, then the processing stations, then the packaging stations, then the distribution, and you go all the way up to grocery stores and restaurants and then consumers. So when you're thinking about changing that system, you quickly find yourself, as our network has done, in a quagmire of diverse interests, which even government, if I can say, can't even penetrate, because it goes far beyond even the regulatory capacity of a government, because it's dealing with so many different aspects. So my question to you, and also my hope that you can solve it so I can go back to India and then we're set, is I really admired what you said. I agree with you, Anna. I really like this more broader approach of thinking about this. And one of the things that we keep railing against is when you start talking about how to fix these systems, the onus often gets put back on the least empowered stakeholder in that process. So it's we have women farmers in India too, right? 87% of the farmers in India are smallholder. 70% of those are women, just like in Kenya, where I know ASIPEs work well. Those women are often asked to adopt these technologies or these techniques, while the other stakeholders that are much more powerful interests are not able to, and for whatever reason, do that. And so my question for you is, because stewardship also is a little bit of a loaded term. I'm with you on sustainability. I hate that word, but I have to use it all the time. Stewardship also suggests, at least classically, this very much tied to the land sort of concept, and that's usually the least empowered person in this process. How do we, get to my question, how do we foster a stewardship concept that works at this food systems level, that puts the onus on these much larger, much more financially salient stakeholders, and not on the people that I also care most about, who are the people that aren't here at the table and have no voices. And I struggle with this. So I'm hoping that you have some insights, also for the people out there, to recognize that we have a lot of work to do and that we often put the blame on the people that have no power in this equation.
Yeah, I think in going forward, it's all in steps. We need to make sort of dismantling steps in our current systems and start building blocks for a new system. Currently, yeah, everybody's livelihoods is based on our system. Everybody's, like, those are the jobs that people have to feed their families. And so if we're like, oh, we get rid of these food systems, then we don't have jobs. So whatever the bricks we need to lay, it's a value shift. And so when I was talking about stewardship, I was talking about that we, who are not traditionally the stewards, need to become stewards. And our government needs to figure out how to be stewards. And so with that, whatever that looks like, there should be new jobs, right? There should be new job creation that is literally going out and taking care of the land, renewing habitats or whatever that might be, or taking care of these animals or whatever's in the food system. We don't know what it looks like yet. It's an imagined future. But I think I don't like to think of it as like a naive, blind, that, oh, it could be so perfect in the future. But if we don't start making the steps now, then it never can be. And so we just need to start thinking about what that could look like so that we can start finding ways to create new jobs that are more of stewardship on our shoulders instead of the people who have traditionally had to do it.
Yeah, so I think that's a good question, but a hard question also. So the food system is a very complex system. It's different from one to the other. So when people talk about food system transformation, they always talk, oh yeah, the global south, those guys, they don't produce enough, they are hungry, so that is a system which needs to be fixed. But in reality, it is a system in the north that needs to be fixed more. So during COVID, I don't know how many people remember, there was a shortage of meat, beef, pork in the United States. So shortage, your shelves were empty, we were watching on TV. So what happened? It's not because there was no production. There was a lot of production, actually. The farmers that were producing pork had to cut off. The reason there was shortages is, I didn't know that the whole meat distribution, pork distribution system in the US was controlled by 4 companies. 3 of them were foreign companies. So, so when the COVID came, then the workers got sick and the plants were closed, there was no movement, distribution. So I always say food system, food distribution, food security is everybody's issue, not just a developing country, developing thing. So I think there has to be a political will also to really transform. And all these players, as you mentioned, along the way. And one of the things which really bothers me big time is how little we value farmers. During COVID, there was, once the vaccines were developed, there was priority list. Priority list of elderly people, this, that, health workers, this, that. Farmers were not in the list. So I'm like, are you kidding me? Why don't you put them on the list of the priority? Because we need the food. Eh? So in developing countries also, the system has deliberately, the smallholder farmers puts them at poverty level, poor. So how do we transform actually the food system when we don't value the producers who feed us? Mm-hmm. It is, yeah.
Very good point.
Yeah, it's difficult, yeah.
Thank you, everyone. We have a question coming from the audience. While food is necessary for nutrition, and we've talked about challenges with malnourishment, for many of us, here at least, what we eat has many other benefits. Eating can fulfill kind of sensory satisfaction. There also is cultural connections to the foods we eat. And so we were wondering what your ideas were for how the United States, a dominant culture in the United States, might go about embracing entomophagy.
I'm trying to think of where to start. I had like one thought, and then the question kept going, and I went somewhere else.
Sorry.
So one of the things I love when I'm giving examples of insect eating around the world, and focusing always on termites mostly, because that's what I've studied, is, I have these examples of just like women that go out foraging together, right? And then they find a termite mound, and they sit down, and they enjoy a bunch of termites, and then they take some, and they take them home. And so it's a social thing. Food is so much more than nutrition. We don't make our food choices based on nutrition. We make them based on so many more things. And so I think, in some ways I love the insect-eating parties, right? Like we had the bug bites yesterday, and I know a lot of students have baked cookies and brought people together. And so it's hard. It's difficult because it's a little bit of it's novel, right? It's a funny thing to gather together and, ooh, we're going to eat bugs. But I think the more we do it, but the act of doing it is making it more known, more accepted, and it's making it social. And so I do think that social component to it is really key to going forward. I really think another major part of sort of entomophagy in our culture, and things need to happen, is that we need to find ways to make them delicious. We get these freeze-dried crickets, and they're dry and stuck in your throat, and it's not always the best sensory experience. And so challenging ourselves to play with the flavor, to treat the insects as an ingredient that contributes to a dish, and what can you put with it to complement it, not just hide it. I think that's how a lot of times we treat food insects here. And so the more brilliant chefs like Sean Sherman that are doing this, the more we'll start seeing it in more places in delicious ways that we're going to want to eat.
Yeah. And in Germany, in Berlin, every year the government sponsors a big function. One of them, the major popular function, was inviting the public, the people from Berlin, all week long, to taste various insects. So they bring in this big television chefs, and because I have participated many years there with it, at least 3 years. And it was fascinating that all these elderly people coming with a stick and with their grandchildren and telling the grandchildren, taste it, it's delicious, yeah. So I said to one of the elderly couple, this is great, yeah, wonderful that you are encouraging. They said during the war, they ate literally dirt, tulips, everything, because there was no food. So this one is a delicacy, it's no problem. So I think it is changing mentalities, changing perceptions. So in Kenya, based on our work now, and a large 3,000 or so school feeding program, now Kenya has approved fortifying porrages, the maize meal, porridge, ugali, bread, flour, 25% cricket powder to fortify. It's labeled baby winning porridge with termite, 10% termite. labeled. People bite, no problem, because you are not seeing the tentacles, everything of that. You don't see the difference, actually, you don't taste it, but these are packed with nutrients, which are really important. I think more of this is going to happen as we know more and as people benefit from the nutrition, from the health aspect of it. I mean, cereals for breakfast that most people eat in North America and even in my own home, cereals, cornflake cereals. Imagine if instead of sugar you add the cornflake is made also fortified with insect things. This will be great breakfast for the kids and adults. So I think this is coming. So it's no problem. I think you will see. It will come. Yeah.
Yeah, I also wonder if, you had some striking statements that have been made about eating insects in the Americas. But I wonder if part of the problem or part of the reaction has to do with the fact that for 10,000 years, we've had cultivation. Almost everything you eat. I mean, sure, there are people that will go out and hunt elk or deer. But almost everything you eat is something that's supplied as something that's cultivated. And so there's a sort of a distrust that builds up generation after generation about something that doesn't come to you now, packaged, in a grocery store. And perhaps this is part of how you get around that. If you've got things that are showing up in a grocery store and they say, yeah, I'll try it, then these attitudes could change as well. But again, 10,000 years of agriculture and animal husbandry, it seems to me, could have a big influence on just what you think you should be eating.
Well, I would argue it's not 10,000 years of agriculture. It's however many hundreds of years since industrial agriculture.
Yeah.
Because the #1 food system around the world is small-scale agriculture. And in many of those places, pest control is harvesting the insects out of your field, and those also being a food resource. So out of your plot of land, you have the crops that are going to provide you nutrition, but now you have the insects that were drawn to it, that if you can harvest them, you've just doubled the output of your plot of land. And so that is a traditional land stewardship of cultivation. It's when we industrialize it and it becomes the industrialization of the chemical pesticides, and it really starts demonizing the insects in this relationship. But small-scale agriculture has kind of had to learn that insects are part of it. You're going to lose some crops to insects, and you can benefit from eating the insects.
Right, but we were talking about, why is, North America?
Yeah, so here in North America, it's industrial, right? It's the industrial, but I think it's, a lot of that is based, I think, we have the industrialization, and then from that, things start looking more uniform, and then we expect things to be uniform. And so part of the success of fast food chains is that you get the exact same hamburger no matter which McDonald's you go to. And so we start expecting that. And so we have that packaged foods and everything. So I think it's all bundled together where, insects were not a food that got put into our packaged food system.
Right. I think Jonathan had a comment, and then Shannon had another question.
The exact same terrible hamburger wherever you go. But I mean, I'm thinking about, inevitably, I think people will end up eating more insects. I suppose what I fear is that they will be eating them indirectly through other animals, because the insects will be used to make animal feed for chickens and pigs being raised in exactly the conditions you presented on your slide. And that's a really serious fear. And I don't know what the solution is, but I suppose part of it is just trying to inform people how inefficient it is to get your protein in that indirect way. To be eating these animals that have themselves been fed on other sources of protein. And you have to understand the pathway through which you're getting your protein and think about how you might do it more efficiently. And when you think about it in that way, you see there are different options. One is to eat insects, and one is to just eat more plants. in the first place. And I tend to myself recommend the eating more plants option. But the main thing is to, thanks. But the main thing is that all of us are thinking about the pathway through which our protein reaches us and how we can make it more efficient.
I think it's also, we also have to take into consideration the differences, right? Because it's funny you say that you know, livestock we're talking about, or animals. In India, that's insurance. That's not just protein. It's actually an insurance way for farmers to have money by selling the livestock or eating the livestock in cases where they lose their crop from floods or droughts or the effects of climate change and all the things that are happening. So at the same time as I agree with you to have a plant-based diet, I think this isn't something that we can, I think this goes back to my previous comment, we can't just shift. We can't just say, even though India is a very predominantly vegetarian culture, they do have cows, and they do have a lot of pigs and other things as well, goats and things, because that is an insurance policy that allows them to sustain their livelihoods. So I think that it's important also to be respectful that what we do here in Minnesota and what we should be doing here in Minnesota, and I really appreciated, Julie, your talk referencing really, I think, us here in North America, it can't be equal. And I think this is something that I often worry about when we talk about like the sustainable development goals, which is like kind of this thing that we should all do across the world, and how it works where segmentis, you know, in different African countries has to be very different because, you know, the way we live and we make money and we survive is different in different parts of the world. So I do agree with you, but at the same time, we have to be cognizant that we don't all live the same way.
I'd like to ask, did you?
So,
yeah, of course.
I think we have also to accept that no matter what, a certain proportion of the population globally will never eat insects under any circumstance. So we have to accept that as well. So, you know, as you know, I'm from Ethiopia. In the 1980s, there was a huge publicized famine in parts of Ethiopia. And so the Western countries sent, I remember, a lot of food donation, and one of them was like a shipload of ham. So ham in Ethiopia, we are 60% Coptic Christians, 40% Muslims, neither eat pork. There is no way you are going to eat pork. So people said, are you kidding me? I would rather die to go to heaven without eating pork than prolong my life by a few days and go to hell. So all that pork just got rotten. So there are portions that under any circumstance are going to eat insects. That's fine also. That's all. Yeah.
Okay.
Boy, there are so many really great questions here. It is killing us up here. Unfortunately, we want to stay on time, so I'm going to cut it short. If I had been let's just say we should stay on track. So at this point, note there will be a test of the National Emergency System at 1:20. There will be a test of the St. Peter's System, we're pretty sure at 1 p.m. Information about food choices on page 23. Some of my colleagues have graciously agreed to convene discussions in the forum today where the buffet is, and also you can bring your bag lunch there. If you'd like to just sit at a table with some other people and talk, my colleague said, don't expect me to be an expert on what just happened. I said, no, you're just a convener. People will talk. Please do join one of my colleagues. The Learning Lab is fantastic. Also, please make your way down to the south end of campus to see diminutive messengers in the Schaefer Gallery. And again, we're in the middle of Sukkot, the pilgrimage festival, which marks the end of the agricultural season in Judaism. Please head down to go to the Ant Thou Sluggard, but remember that it's now in the Banyer Multi-Faith Center, not in the Sukkah. And Gustavus students, don't forget 12:30. You have exclusive access to our speakers, so please do head to Beck Hall for those. Other activities you can find in the program, don't forget to recycle, compost, and store waste in the right place. Please do not smoke anywhere, except maybe mentally. And we will reconvene at 1:45 when we will hear the Gustavus Wind Orchestra. Thank you for this wonderful session. Thanks again to our amazing speakers.
Crickets over there.
Hi, everybody. I'm Tane Danger. I'm one of your co-hosts for the Gustavus Nobel Conference live stream. This is the 59th annual Nobel Conference. This year, I don't know, it'd be weird if at this point you didn't know that this whole conference is about insects. You just wandered into this.
Surprise!
Surprise! What am I watching? It's an entire conference about insects, subtitled Little Bodies, Big Impact. So we've had two great presentations today, and then we had this like phenomenal panel discussion unpacking a lot of that. I'm here alongside my co-host Meg Kurth, who has been with us. I'll just we are joined also. I'll just keep going down the line. Actually, do you mind introducing yourself, Heidi, just to say a little bit about yourself? Because, again, you're one of these folks that we're happy to have here, but you wear a lot of different hats here at Gustavus. Today, you are here to talk to us about your work with Nobel, but can you just say a little bit about who you are here at Gustavus today and 364 other days?
Sure. Hello, my name's Heidi Miller, and I work in the music department here at Gustavus. I conduct the Gustavus Wind Symphony, and you may have seen us perform this morning before the 9:30 session. I also teach a lot of classes in the music department. I teach courses in music education and conducting and sometimes theory. And I'm here today to talk a little bit about one of the pieces that we performed, which has to do with the theme of the Nobel Conference. But we'll get to that soon, so I'll turn it back over to you.
Yeah, no, thank you so much. So yeah, I'm very excited about this. So this was a piece that came together that you found for this year's Nobel. I mean, we should just talk even maybe, before we get to the specific piece, we've had a couple of folks in the Schaefer Art Gallery from other places talking about, this is really the spirit of the liberal arts. Can you help us just sort of talk about that in terms of music and using music to bring to life elements of the Nobel Conference? This spirit of like music is helping us think about some of the topics at Nobel every year.
Well, I mean, I think we feel so privileged to be able to perform at the Nobel Conference every year. I think that, for me, I can't imagine living without music.
Yeah.
But my second love really is science. And the reason I love science is a lot of ways the same reason I love music and art. It's kind of this wonder and awe that I feel when I look at something bigger than myself. And I feel like knowledge is so vast, and we can't really fully possess all the knowledge that we have, and that we can just start to like grasp at the edges.
Yeah.
And so it's like being able to look at a little bit of eternity.
Wow.
So,
Whoa.
yeah.
Wow. Very cool.
And so it's a privilege to be in the presence of science, and I feel like it's a privilege to be in the presence of art as well. And so, I don't know, I feel like being able to present some of that art alongside of all of this science just really fits well together.
Well, you were telling us earlier about how this particular piece of music came to be, and that this is a consortium of different universities, I guess, that came together to commission it from a composer in Mexico.
Mm-hmm. Well, it was different colleges, different educational institutions, also like high schools and some middle schools, and some community organizations as well. And there's an author, a St. Paul author, who wrote a picture book that was the inspiration for the piece of music by a composer in Mexico. And the connection there is that we've got these butterflies that migrate from Minnesota and other places in the Midwest, and actually in California as well, and they migrate down into Mexico. And so their connection from an author in Minnesota, a composer in Mexico, and it makes a really cool connection.
The very like creation of this is indicative of the science. That's really, really cool.
Yeah.
So let's talk. We've kind of said a little bit about this, but let's talk about this. So this piece. The piece is called Monarch Migrations, correct?
Yes.
And can you tell who is the composer who put it together?
So the composer's name is Nubia Jaime Danwan, and the piece is called Monarch Migration, and then the book is called Monarch Butterflies.
Got it.
Yes.
Got it. Okay, and this was part of the prelude for the Nobel. And again, for folks who maybe missed it or whatnot, can you just give us tell us a little bit about this piece? Like, how did it feel? Like, what was it like to have it come to life here today?
Well, the piece is really, it's an amazing piece of music, and how Nubia structured the piece was a lot of times in music we talk about form by using letters to talk about different sections. And so she structured the piece A, B, C, B, A, to kind of give the form of how the migration pattern works.
Oh, wow.
And so the A is the beginning, the butterflies, the eggs, the hatching and everything, and the start out, in the northern area. And then they start to progress down, and the B is kind of that migration period where they're heading down southern, through down to the south. And if you look at the migration pattern, that starts with them kind of sparsely, there's lots of different locations, but the arrows start to coalesce as they go south and they come together. And you can hear in the music during that B section, it starts to come together, and there's more people playing together in a more unified way, and they come together. And then the C section, the music changes quite a bit, and they're down in Mexico, and it's more dance-like, and they're all together. But as Nubia stresses, the theme, the butterfly theme, stays the same. It's just that it's surrounded by different kind of musical ideas. And then once that section happens, that C section ends, they come back out, and they go back through the same pattern out through that B section and back to the A. So the whole piece follows the migration pattern. And it's you can hear the butterflies, you can hear them flying. It's beautiful.
I think one thing I'm really curious about is, like, as a director who's working with a piece that somebody else composed, how do you go about, like, interpreting that? Like, is there a creativity that you bring to that table, and, like, how does that work?
Well, I like to think so. Composers are some composers are more specific than others with what the instructions they put in the score. Nubia is pretty specific, but no performance is ever exactly the same. So there's a lot of, taking what's on the score and making sure that your players are doing what the score tells you, but ultimately there are musical decisions that happen that have to do with the space that you're playing in, like playing in our rehearsal space, playing in Lund, which doesn't have a whole lot of reverberation. And then next weekend, we'll be playing the piece in Christ Chapel, which is like playing in a bathroom. It's got a lot of reverberation.
Yeah.
So there will be a lot of adjustments that we have to make.
Interesting.
So there's a lot of different things that go into that, and into interpretation too, like how much are we going to slow down here? How much with this set of people do we need to slow down to make this effect work? So there's a lot of little intricate details that go into making sure that that the effect that the composer's asking for is coming across.
And this, just to set the expectation, this is the first thing that students are performing back here on campus for the fall semester, and so it's very cool and exciting. But I've got to imagine, there's a mixture of excitement and also like, oh my goodness, like this is kind of a big show that we're getting started with right here in the first week in October.
Yeah, it's their first performance, and the group that played this morning is a lot of first-year students, and so this is their first performance.
Yeah,
it's very exciting.
Yeah. Can you just talk about, so we talked a little bit about how you shaped this piece, because this is a piece of Nobel every year. How do you decide every year, like what kinds of music you're going to try and pull in, whether you're going to commission something, use something that's been written before? How do you go through this process, which I imagine starts long ahead of when the actual conference is going to happen?
Well, for Nobel is kind of a special situation in which we know there's a theme.
Right.
So it's nice to try to find some music that supports that theme, but you can be heavy-handed by just saying, I'm going to do this piece that has this in the title, this piece that has this in the title, and that doesn't always work out. But this one was so charming. What a charming idea to have these insects, and there's pieces that have like insect names in the title. So this one I was deliberately trying to find things that were inspiring. But in terms of monarch migration, this was a complete, happy accident in that I was up at St. Paul Central High School, has a friend of ours, is the band and orchestra conductor, and he was the one who initiated this consortium. And in I guess it was a year ago, I was up observing a student teacher there, and we had a conversation, and he told me about this commission that was just in the process of getting off the ground, and he said it was going to be called Monarch Migration, and I was like, wait, the Nobel conference next year is about insects. And he's like, well, you have to get in this commission. And so I immediately thought we have to do this because what a perfect and unique setting to be able to show this piece off. So that was an obvious, we need to do this. But it doesn't always work out exactly that way. But, that because of having the themes, that generally pulls in why I program the way I do for the Nobel conference. But for other concerts, it's different. It's a little bit more, I don't know if I answered your question.
No, that's beautiful. That's perfect.
I mean, I think I'm kind of curious about, like, I think for a lot of people, if their only experience of classical music is old classical music, that it's kind of almost surprising to know that there is new stuff being made.
Mm-hmm.
And I'm sort of curious, like, how you've approached that kind of education with people that aren't familiar with this. Like, what do you tell people about the creation of new classical music?
Well, it's funny that people
Or
have
do we
that
call it
impression.
classical music, even if it's new?
I think when we say classical music, I think what we really kind of mean is music written in like concert style that's performed by a large classically oriented ensemble. And I think it's very important to not cut off the fact that we have this huge repertoire of music that's incredible. It'd be like deciding that we were going to get rid of all of the art galleries that we go see. Like, let's not go look at the stuff in the Louvre anymore because there's new art being made. And so we have a trajectory, we have a history. And so although not here because there's all this new stuff that I'm doing, there's wonderful new music being written, but we need to connect it to what's happened in the past too. So when I program normally, I try to make sure that I include music that is from the past as well as music from the present so that thread gets connected. And so the new music actually gets supported by the fact that we can see that it comes out of something. But there is so much new music being written that's really exciting. And so I like that people are able to see that, okay, this isn't a dead form of art. This is something that's new and thriving.
You talked very eloquently a moment ago about this connection between music and science. I'm just curious, like, how students feel about that, or if you sort of hear from students, like, I get this, or if it's still maybe it's a little bit something that is illusory. How do the students, like, sort of feel about this? Like, we're doing music as a way to bring science to people in a different way.
I don't know. I guess I haven't talked to them in that specific way.
Yeah.
That's a really good question.
Yeah.
It's maybe something I should ask them.
Yeah. I mean, you said a little bit about this before, but I mean, I don't know, if you were just sort of like making that pitch, like, music is quintessentially like connected to how we think about the world. Like, what would you say?
Well, I mean, I answered with how I feel.
Yes, that's fair. That you're you, and so that's what we want. Like, I love what this is like. One of my favorite parts about Nobel is like using the arts in order to bring some of these pieces, this like work to life. Because there is, we were talking yesterday with some folks from religion department, and as I mentioned, Schaefer Art Gallery. But like, you can learn the science, but I don't know. It feels
making
sort of
emotional,
incomplete.
like the emotional connection around data is like such an important part of, I think, getting like learning, right? Like sometimes you can like learn a fact and not really understand that until there is a connection through your life to that thing.
And I feel like we heard that
art does
from our speakers, like the panelists on some level too, right? Where they weren't necessarily speaking as much about just like bringing the arts into it, but they really were saying like, it is not enough sort of just to like do the science and like see how a fly sees the world or whatever. Like, we need to like put ourselves like into an emotional space where we like understand this relationship. And that to me feels like that's what the arts offer us, is like a way to like experience like with something more than just sort of like the, I don't know, critical thinking intellect part of our brain, but something that feels bigger.
Awe.
It's
like
awe.
the eternity piece you were almost talking about.
Yeah, my daughter and I talk about a lot, like how in order to really do something well, you have to care, and you have to not be afraid to really care about it. And a lot of times, those are the most successful people. And I feel like science and art have a lot of those people, the people that aren't afraid to, be a little nerdy. And a lot of times, the nerdy people, those are the ones that aren't afraid to show how much they deeply care about something.
Expressing your love for something without reservation is in some ways just really nerdy.
And think about scientists, and think about how deeply you have to care about what you're studying and what you're learning, and think about artists and musicians and how deeply you have to care about that in order to be successful. And I think there's so many parallels. And, just for me, like how long I wanted to be a scientist, and then how, but I've always been a musician, and then I went the other way, but like, I still love the other. So, they connect a lot for me.
Oh, that is so wonderful.
Heidi Miller, thank you so much for caring so deeply and not being afraid to care very deeply.
And I'm such a nerd, too.
Yeah,
so.
I love it. That's beautiful. Well, this is beautiful. Oh, we should just say quickly, so the piece was played. You mentioned it. You are going to do it again in Christ Chapel.
Yes.
When is that?
At our Family Weekend concert, which there are two this weekend.
Okay.
There's this one on Saturday at 1:30, and there's one on Sunday at 1:30, and we will be playing Monarch Migration on Sunday at 1:30.
Okay, this Sunday, 1:30, for folks who are watching and maybe here on campus or in the community and want to come to Christ Chapel and hear it live, but then there'll also be a recording of it on the Nobel Conference website, so you can always get it that way. But of course, it's more fun to be here. So yeah, so thank you so much, Heidi. Really appreciate this. It was absolutely lovely to get to talk with you about this amazing piece and the Nobel Conference. So
well, thank you for having me.
Yeah, thank you for joining us.
Enjoy the rest of the conference.
Yeah. Okay, we're gonna do a quick switcher changer with a guest. I will say, you're just while we're doing that, this is the Nobel Conference. It is our 59th annual. This year is all about insects. We are kind of in a lunch break period here at Nobel, and so we, on the live stream, I was literally telling one of the producers of the Nobel Conference this year, I am feeling like this live stream is getting to be like a really amazing, wonderful thing that, I don't know, I don't want to say that it's better than being here in person, because I want you to come here in person and it's extra special, but we get to do these little conversations with some folks that nobody else is really getting to see if you're not watching the live stream. So you should come, our next guest should come on stage.
Even if you come in person, you should just skip lunch and watch us instead.
Yeah, exactly. If you come in person, you could have us on like your phone or your tablet and see some of these things over your lunch hour. And this is like another great example, is like we get to talk here and most people aren't going to get to hear our conversation with Tom. Tom, do you mind introducing yourself?
Sure. I'm Tom Ansley. I'm a 1975 alumni of Gustavus, so many years ago in the back last century. And I have a long experience with the Nobel Conference here. I went to a number of them when I was a student. I was on the board of trustees, as you are now, for a number of years, and came and still come. And one of the benefits of being on the board and being a scientist and an academician was that I had the fortunate assignment of picking up speakers in the cities.
Yeah,
and
that's fun.
Picking up Nobel laureates, people who got the Nobel Prize after they talked at the Nobel Conference, and spent personal time with them. And that's why I got so much interested in the backstory of the Nobel Prizes.
Is it like the nerdiest version of Taxi Cab Confessions?
That's it. That's it.
Yeah.
Yeah.
I love it.
Yeah.
That's good. So that's and so you're here to talk to us because you've been doing a lot of work on looking at and studying and writing about folks who have gotten Nobel Prizes, which partially came out of exactly this thing. You literally picking them up and bringing them down to Gustavus and having an hour and 15-minute car ride with them.
That's exactly right. Instead of talking science, we talked a little bit about the person themselves. And I started to realize, as you and others know, that, sometimes the backstory is just as interesting. And you find out that not only is individuals brilliant, but sometimes they're quirky a little bit.
Just sometimes?
Yeah, just sometimes. Yeah. And really that, that sometimes the backstory or the interesting facts about these individuals are just as interesting as the formal part of their biography.
That's wonderful.
And I thought that's a story that needs to be told.
We have in our notes a question of how did ants in a bathtub contribute to quantum electrodynamics, and having seen that, I need an answer.
You need that.
I need
I
to
feel like
know
we should
what the ants
restate
did.
because there was a lot in that I feel like people. So
How did ants
Yes.
in a bathtub
Yes.
contribute to quantum electrodynamics?
All right, a good question.
Yes.
A lot of my talk yesterday actually discoveries that were made in bathrooms of all things. So we'll talk
That's
about
great.
the bathtub. Yeah. Just briefly, here's a 30-second update on quantum electrodynamics. QED describes basically everything in the universe, and it's basically, in its simplest diagrams, are electrons that get close to one another, and then something interacts in them, which is why our chairs don't fall apart, they stay together, and then they interact, and then they disperse in a new direction. And somehow in all of that, there's this like nebulous, what was it, essentially? And physicists, for years, back 40s, 50s, and 60s, were all trying to describe this. And one of the things they thought about was photons, and in fact, the Nobel Prize just the other day was for photons, which were like light sources that interacted. And like a lot of scientists, and especially physicists, they want to make everything complicated. So one person would come up with a simple equation that for us would be hideous in and of itself. This explained it. And another person would come up with an equation that took 3 or 4 lines. And another physicist came along and described an equation that was 6 or 7 lines long. This accounted for it. And an individual by the name of Richard Feynman, who was probably as much of a comic as he was a scientist. One of his hobbies was cracking safes. So to give you an idea, he thought you got to make it simpler for this. And he took all these equations and he turned them into little diagrams. And he said, but that doesn't do it too. It still doesn't, explain it all. He was in his bathroom in California in the 1940s, and he was sitting there, and he noticed he had ants in his bathroom. And the ants were crawling around. Now, most of us, given our choice, would run out, grab a can of Raid, and we'd go in, and Richard Feynman went to his office, and he brought out a packet of colored pencils. And he said, I'm going to follow these ants. So he put a sugar cube down, and when one ant found the sugar cube and made the beeline back to the its nest, he followed the path, and it was a little circuitous. It wasn't straight, but he took a colored pencil, and then when another ant came, he followed that one. And after he got through about 10 or 12 ants, he noticed that the pattern that the ants were creating, as they were creating a straighter and straighter path going back to their hive, followed exactly the diagrams that he was trying to prove for how electrons work.
Oh, wow.
So he said, how can you be more simple in nature and explain things than what ants are doing? Now, of course, that's a simplified story, and physicists will say, well, you can't say it like that. But the idea, he believed in not making things more complicated than they need to be.
Right.
Right. And he said, this is it. It proves what I'm thinking.
What I love about that and some of your other stories with this is that I don't know, I feel like a lot of us probably have a notion of science that it's like somebody going into a windowless room with a piece of paper and a pencil and coming up, as you said, almost with those other folks, coming up with a bunch of equations and just sort of typing into a calculator or whatnot. But what you're describing is that a huge part of science and discovery is allowing yourself to be open to seeing things differently, to imagining what could be and visualizing sort of what does this tell me about a question that I'm pondering, which requires sort of a different headspace than I think sometimes we imagine.
Go ahead.
And it opens up all these other questions too, because my next question is do we know why ants and photons behave the same?
No.
Oh my God!
No.
That's
now
so
having
cool.
said that, we don't know how ants and photons and how they interact together like electrons. One of the things that has come about in the last 50 years is they will actually have computer programs now that will make predictive models of, for example, how would you cross the United States and reach all 50 states?
Sure.
And that's based on models that were developed where they would take ants and they would put them all in a bunch, and then they'd watch how they migrated.
Yeah.
And how the ants migrated in their patterns was very predictable. You could almost predict it with a computer.
Wow.
So it's kind of like, what does it have to do with it? I don't just, ants inherently know what to do, and we've learned from it, basically. That's kind of the answer.
That's interesting. Well, I mean, it's interesting also just to have, like, I think Tane was kind of getting at this, where is to use another like idiom that is silly for this context. Science doesn't happen in a vacuum.
Exactly.
Right? It's like science is part of. You have one science, but it's connected to other sciences, which is connected to other sciences, which is connected to art, which is connected to how people think, which is connected to people being weird enough
Oh, yeah.
that they are willing to think in weird ways.
Yes, they're willing to be curious.
And that is a hard.
It's hard. And
Yeah.
what is exciting to me is that and we have almost hit on the two sides of it, there's the importance of asking the right question.
Right.
And there are scientists that have made breakthroughs, Einstein said, I need to ask the right questions and then I find the answers. There are other people that just sit around, but they observe, basically. They don't know what the question is exactly, but all of a sudden they see something by observing it and actually see the answer first. Oh, that's why that happens.
Yeah.
Right? Rather than why does it happen and I'm going to prove the answer. The answer's there and you have to observe it, basically. And people have that mindset, going both ways.
I mean, a lot of this raises some really interesting questions, which hopefully we're getting at in some of Nobel conference generally, of just how do we structure education? Like, how do we encourage young people, but lifelong learners to sort of be in that space? Because again, I frankly, I think that I worry sometimes that a lot of our education is not set up to encourage sort of creative, wide-ranging thought this way. It's kind of set up to train people to fix something or do a task or whatever. And a lot of what you're talking about requires a certain amount of encouragement to wander, to dream.
Exactly.
Yep.
Yeah. And we certainly need what I would call structured science in schools, because that's the fundamental principles. But sometimes kids are turned off by that.
Yeah.
It's very strict. It's very structured. Oh my God, I'm not a great mathematician.
When you train the creativity out of people,
You train the creativity.
you're not careful.
Let's go outside and look around today.
Yeah.
Yeah.
They mentioned that in one of the lectures today. Go outside and every kid come back with an insect they found.
Yeah.
It turns out to come back with dozens of insects.
Yeah.
Now, why do you think there's so many around? And then the kids start talking about it, and the kids become creative, and then they realize, I didn't have to come up with any great idea. I didn't have to come up with a great question, but I went outside and observed, and then we started talking about it. And, that gets my juices going then as a student in science. And I think that's something we need to do. Curiosity, observation.
Have you learned anything from working on these stories about Nobel Prize winners? What kept them curious long into their lives?
That's a good question, and it's something that I haven't specifically explored. It might be something innate in these individuals, because the few stories I read about the childhood and the younger stages of these people, they somehow were curious at a younger age. They had outside activities. Okay, I mean, some of them read books, but some had instruments. Some of them loved baseball. They had some outlet that just kind of gave them a curiosity. It's something to think about outside of their regular schooling. So, and I think, is there something about these individuals? I think it's that they had outside activities, they had outlets that they took advantage of, and those created different opportunities, different ways for them to thinking.
Well, because we're running out of time, but I'm kind of hearing two lessons, I feel like, out of some of what you're sharing with us. One is this systemic piece, encouraging folks to allow themselves to sort of wander and imagine, daydream, and especially with young learners, try and build some of that space. But then from you personally, I'm hearing a thing which is take advantage of opportunities you have to just talk to people and talk to them about their who they are as people.
That's it.
Because you learned a lot doing that, and are learning a lot doing that, which is you aren't just sort of sitting down and say like, oh, explain your formula to me, but you're letting yourself sort of wander and daydream with these people in a way.
Daydream a little bit,
Yeah.
It's not just what was the answer, but how did you get there?
Yeah.
You know, what did it do? It's nothing's a straight path. Everybody has a circuitous path. Tell me some of the interesting things that you ran into.
Nothing is a straight path except the ants.
Except ants.
Yeah.
That's right.
This is great. So you gave a talk on this. You were
Yesterday, I did a breakout session.
And I don't want to like scoop anything, but you're working on a way that people will be able to hopefully learn more about this in the.
Yeah, about a year ago.
Yeah.
I don't want to call it research because that's a compliment. It was more of a rabbit hole that I went down in research. But I started to synthesize and collate all these things, and I thought, I've got to pull all this together.
Yeah.
You know, and I've got to tell people, not just scientists, but people that would be interested in reading. Here's interesting stories about how Nobel medals were lost.
Yeah.
Here's how somebody found their long-lost sister. Here's how somebody did a self-experiment on themselves, which you wouldn't do today, but it turned out to win a Nobel Prize. And these just the little stories like that. You know, what were their hobbies that they had? You know, there was a young child in China who was interested in baseball, and he remembered how foul balls glanced off of bats, and he ended up doing trajectory physics because he always thought about, you know, it's like, that's where it all comes from. We all see the finished product, so to speak, but we don't see the story behind it. Yeah. And that's what I kind of just want to do, is open up that side of it. How successful it will be, we'll see, but it's been a blast doing it.
Well, we really appreciate you coming and sharing some of this with us today, and like I'm very excited to hear more about this. And again, I hope that you plan to come back to next year's Nobel conference and
I've
maybe
come
share
for
a little
years.
bit
This is
more.
about number 30 for me.
30?
Oh yeah.
Yeah, I feel like you should get like a star or something.
Oh
like a
no,
button, like-
no!
Special badge.
I
A
sat
badge.
next to people today that started with number 1. This is their 59th
Really?
year.
And said they need a special badge.
They
need a
totally.
hat.
Yeah.
They need a seat on the dais.
Yeah,
like-
so I'm a newbie, so to speak. I'm you know, beginning to be scientifically mature after 30 of these conferences.
I love it.
But
This has been great. Thank you so much for coming and
thank you.
spending a little bit of
Nice
this
to see
time
you.
with us. This has been absolutely great. We're gonna pause here because we're gonna say goodbye to you, and then what we're gonna do is we're gonna go, I think, back to a video that we have, and we're going to do that, and then we are going to come back a little bit later. So in case you missed it, basically what we're going to do, yesterday morning, we opened Nobel with a really spectacular...
It was extremely cool.
A spectacular opening ceremonies, which included a processional, which it always does, but this year in particular, it was the Gustavus Jazz Ensemble, and then a light show that was put together inspired by fireflies and bioluminescence. It included dance and music and a whole wide range of the arts, really starting to set the tone for this entire conference. And it is spectacular to see and to watch, and lucky you, we recorded the whole thing, and so we are actually going to replay that now so that you can see that, and then we will be back in just a little bit ahead of our next set of panel presentations this afternoon. So take a look one more time at the opening of the 59th Annual Nobel Conference, and we'll see you back here in just a little bit.
Good morning. So, after 59 long years, we're doing this opening processional in a slightly different manner this fall. Trigger warning, there will be no Elgart, no tried and true pomp and circumstance, no Brahms academic festival overture, or grand march from Act 2 of Giuseppe Verdi's grand opera Aida. So sorry, folks, no Egyptian chariots, no real live elephants. What we do have for the first time ever in the history of the Nobel Conference is a full-bodied and vibrant celebration of America's greatest contribution to world culture thus far, jazz. So when these, thank you. So, when these particularly homegrown saints that you see in the back there that are here among us today in the town of St. Peter come marching in, they will be led by modern dancers performing original choreography by my very able colleague from the theater and dance department, Melissa Rolnick, and our great jazz ensemble playing an original composition entitled Flash Patterns by their director and leader in all things jazz, Professor Dave Stamps. This processional will employ two primary and essential elements of this African-based Black American musical form we call jazz: rhythm and dance, intertwined to weave history and story and spirit into one. In this case, the story of our most important fellow beings here on this planet, the tiny little insects. But also in that intertwining of rhythm and dance, weaving an essential story of our own true, interdependent, interconnected human selves on this earth. The great jazz arranger, composer, and tenor saxophonist Jimmy Heath called jazz music the true democracy of our creativity. Everyone has an equal voice in jazz. Everyone has an important story to tell to the world in this music. From the highest to the lowest, from the most powerful and manner-born to the least ones here among us. No matter if you're born black, white, or brown, jazz is to be felt deeply, spiritually, as well as to be heard and danced to. It embodies not only a distinctly American art form, style, and tradition, but a whole way of life, a way of being, seeing, and hearing. To paraphrase Walt Whitman, jazz is simply a cosmos, embodying not just one person or one group, but multitudes. And in jazz, every improvised note and chord and song that belongs to me as well belongs to you, and as well belongs to all the other saintly living creatures of our shared and precious and very fragile world, both large and, as we will soon learn, very, very small too. So, after 59 years, let's all join in right now for something slightly different for this year's Nobel Conference. In this time of trial and trouble, let's all just try and walk together, children, to be a part of this number. And don't you ever get weary, because by and by we're going to lay down this heavy load when all the saints, every single one of them, both large and small, come marching in.
People ask me all the time if I have a favorite bug. Yes, the parsnip webworm is a little nondescript moth, the caterpillar stage of which feeds on a noxious roadside weed. It produces a group of chemicals that are called furanocoumarins, and these are amazingly toxic compounds. Parsnip webworm can eat its own body weight in the flowers of parsnip, which is, from the human perspective, almost unimaginable. It's hard to even imagine eating 10% of your body weight in anything, even chocolate. I've tried, and it's really hard to do. People might wonder why I would spend so much time on an interaction of a roadside weed and a little brown moth, and how society could possibly benefit from such an obscure pursuit. And it turns out it's actually a natural example of biological control. The nice thing about basic curiosity-driven research is that new knowledge that you find has the potential to be useful. For example, there are viruses that insects, including caterpillars, are very vulnerable to. They're called baculoviruses. They basically turn a healthy caterpillar into a little bag of goo. The viruses have an unusual property of being able to produce proteins sort of directly into the cell that they're infecting. And as it turns out, baculoviruses can be manipulated not only to produce proteins useful to them, but proteins useful to us. There's an entire industry now based on baculovirus-mediated expression of proteins, including, among other things, flu vaccine. Baculoviruses enable people to produce vats of vaccine without any risk of allergy. Came from the guts of sick caterpillars that were being investigated for reasons completely unrelated to flu vaccines. What I really like about, studying plant-insect interactions applies not just to entomology, but research in general, that you have an opportunity to find out something no one else knows. You can create new knowledge. And how cool is that?
Dr. May Berenbaum joined the faculty of the Department of Entomology at the University of Illinois here in Urbana in 1980. She has served as head of the department since 1992 and has held the Swanlund Chair of Entomology since 1996. Dr. Berenbaum's research is focused on interactions between flowering plants and insects, ranging from pollinators to crop pests, and on the application of ecological principles towards sustainable management practices. A member of the National Academy of Sciences since '94, she chaired the 2007 Committee on the Status of Pollinators in North America. She testified before Congress on issues relating to honeybee health and pollinator decline. She is devoted to public engagement in science and has authored 6 books about insects for the general public, including a honeybee cookbook. And she founded several outreach and citizen science activities, including Bee Spotter and the UI Pollinatarium. The community may know her best for the annual Insect Fear Film Festival, now in its 38th year. In 2011, she received the Tyler Prize for Environmental Achievement, and in 2014, she was presented with the National Medal of Science from President Barack Obama. As next week is Pollinator Week, we hope you will all leave here today with a message for the general public about all doing our piece to protect our pollinators. Dr. Berenbaum, we give you a very warm welcome.
Thank you, Dr. Jeffrey, and thank you all for coming today. I mean, it's a beautiful day, and I know you could be out enjoying real-life flowers and real-life pollinators, so thank you for coming inside and sharing this time with me and letting me share with you my enthusiasm for flowers and pollinators. So with that, let me
I have it muted.
Stop my video. I did not stop my video. There we go. And talk about the pollinator apocalypse, if there is such a thing. So there's been a lot of talk about the insect apocalypse or the pollinator apocalypse, and it actually it's worth looking into the origin of the word. It's actually from the Greek, meaning revelation. It's from the book of Daniel, which is an account of the life of the prophet Daniel, who was saved from his enemies by God and who learned in one of his many visions that Israel would similarly be saved from its enemies in the end of days. From Daniel 12, there shall be a time of trouble, such as there never was since there was a nation, even to that same time. Kind of sounds like 2020, doesn't it? Well, non-biblically, an apocalypse is considered to be a disaster on a cataclysmic scale. And in fact, there's a whole genre of apocalyptic fiction that imagines a sudden catastrophic event that causes the collapse of civilization. Now, causes can be natural and out of human control, or human and the result of evil or greed. And just a few examples of the genre. Death of Grass way back in 1956 was the story of a mutation of a virus, a fictional virus, Chung-Li virus, that wipes out all members of the grass family, including grain crops, with predictable cataclysmic consequences. Hot House from 1962 imagines Earth getting locked in its orbit facing the sun, where plants overdominate all other life forms by basically photosynthesizing nonstop. The Blight is when a powerful international organization aimed at world domination does so by not, killing all the trees. And as far as I know, there's really only one novel, apocalyptic novel about the loss of any arthropod, and it's kind of a cheat. It's not all insects. Well, all insects disappear. This is Charles Pellegrino's Dust. All insects disappear because of an intermittent cyclical extinction event. Humans had nothing to do with it. This intermittent cyclical extinction event, which happens every 30 million years or so, is not documented in the actual fossil record. But it was, the reason I think it's a cheat is that yes, all the insects disappear, but all the ecological niches are subsequently filled by mites, which are also arthropods. So that, I did not find this very scary, but many readers did. But in November of 2018, the New York Times ran a story, a cover story, on the magazine section about what they dubbed the insect apocalypse. And this generated a conversation on a global scale about what exactly was going on, and recurring questions of whether there is or is not an actual apocalypse. Now, what triggered the stories was a series of studies that had appeared in a relatively short period of time, suggesting that in fact there were massive declines of arthropods, particularly insects, happening all many places in the world. One of the notable ones that was mentioned in that story was published in 2017. It was a study done in Germany over a period of 27 years in 63 nature preserves. That involved sort of citizen scientists doing a series of regular surveys of insect biomass. So just the weight of flying insects, not the number of species, but the weight. And over this 27-year period, this study documented a seasonal decline of 76% in the weight of flying insects, and midsummer decline of 82%. This was a staggering amount, and this study received a lot of attention, and some of the attention was critical in that there were some weaknesses of the study. First of all, biomass is not the same as biodiversity. Biomass, you could have large quantities of a single species, and if that single species disappears, then you have a decline in biomass, but not much impact on biodiversity, strictly speaking, the number of species. Sampling was not done in the same plots over the 27-year study span, so data didn't really depict a long-term trend for any given site. And there were only flying insects sampled. A trap called a Malaise trap was used to acquire the insect samples, which leaves many other parts of the plant communities where insects could have been increasing in diversity. Then early in 2018, well, in October of 2018, there was a study that came out that was equally shocking. It came out in the Proceedings of the National Academy of Sciences, and it was a study done in rainforests in Puerto Rico, comparing a 2-year period of sampling in 1976 to 77 to a 2-year period of sampling at the same place, 2012 to 2013, documenting, as you see in the figure here, massive declines between the early and later samples in both ground arthropods and canopy arthropods. The news coverage was quite breathless. One of the most disturbing articles I've ever read, scientist says of study detailing climate-driven bug apocalypse. I'm pretty sure the scientist is not the one who called it a bug apocalypse, but people were alarmed at these reports. And again, the scientific community offered some critiques of the methods, mostly that in this two sets of 2-year samples, they failed to take into account population cycles of insects, which routinely hit highs and lows. There are sudden losses in biodiversity that, especially in the tropics, according to hurricanes and successional changes, and these were not taken into account. It was just drawing a line between 2 points, basically. The authors responded, and there was a little cryptically, basically questioned some of the statistical analysis of their critics, and then offered yet another correction because they've misidentified one of the arthropods in their survey. So that's another problem, is identifying species in order to calculate diversity. So early, this is the one, early in 2019, another shocking story came out. This was based on a meta-analysis, a review of the literature. This review demonstrated or documented a 40% that of the insect species evaluated, 40% were threatened with extinction. And some, in fact, aquatic taxa, those living in the water, were at risk of extinction. They identified among the main causes conversion to intensive agriculture, but also pollutants, invasive species, and climate change. And this also generated headlines in several different languages and dire forecasts for the survival of humankind, and also elicited criticisms by other scientists. Two of the main critiques of this this literature review, first of all, when these authors reviewed the literature, they used the search term decline, which immediately biased the results that they would find. If they had done a comparable search looking at increases rather than declines in abundance, it's not clear they would have found the pattern they found. The other problem was there was a bias in the geographic areas where these studies had been carried out. Their search was in English, so the studies that they cited were published primarily in North America and Europe. North America and Europe do not possess most of Earth's biodiversity, especially arthropod diversity. So it's unclear whether this non-random sample of the world's biomes was necessarily predictive of global biodiversity loss. At least 6 studies in 2019 were focused just on bee decline based on museum collection specimens, bumblebees in particular. So there's 6 different studies where authors went into museums, pulled out drawers of specimens, and then documented based on the species and how they were represented in past collections that in fact bumblebees were in decline. And now the thing to remember invariably when there are stories about bees that are covered by newspapers or any other media, bad puns seem to be irresistible, and you will see many more of these. So the buzz about bumblebees isn't good, right? Very original. Now, again, these all could be criticized by the limitations of doing museum-based studies. First of all, if you're looking at specimens in a museum, you can get information about the presence of species, but you cannot get for anything about their absence. You can't collect what isn't there, and you don't know why it wasn't there. Sampling is typically biased toward places that are accessible or that have populations that are interested in biodiversity. So the sampling is biased toward, in the Galapagos Islands, the island with the airport typically has more biodiversity than the islands that are harder to get to. The same thing across the US, the county that hosts the land-grant university with the entomology department always has exceptionally high levels of insect diversity because entomologists are way more likely to characterize what's easy to get to. There's also a bias towards species that are easily identified. Often collectors won't collect specimen if they can't recognize it. And you don't know how space limited any specific museum is. And often museums have to discard duplicate specimens because they just don't have space. Now, the fact that some high-profile publications have been criticized for methodological irregularities or limitations emphasizes the difficulty of documenting insect biodiversity to relatively poorly developed state of counting methods. It's also very hard to project population trends due to the deficiencies of sampling. We don't have baseline studies on which to compare contemporary sampling. So there's a lot of guesswork involved, and as a result, some of the population changes that have been documented are not particularly robust, especially because many insects have these boom and bust cycles. You probably noticed them, that there's really good year for mosquitoes this year, actually, and last year was we weren't so bothered by mosquitoes, but it's very typical for insects to have boom and bust. And you can kind of see the pattern here with one of these very famous surveys of the size of the overwintering populations of monarch butterflies who undergo a heroic migration every year. They overwinter as adults, and the eastern U.S. population overwinters in a tiny area near Michoacan, Mexico, in the Oyamel fir forests, where the abundance of the overwintering population is estimated by the area covered by monarch butterflies. All right, so interpreting these patterns without taking into account species-specific ecological attributes such as boom and bust cycles and methodologies is challenging. So there's two populations of monarchs in North America. There's the eastern North American monarchs that overwinter in the fir forests of Mexico, and then there's the western population, which overwinters along the Pacific coast. So the western population also has a pattern of decline and also typical boom and bust cycles. And this is a study that reported a 97% population drop of average historical abundances between the 80s and the mid-2010s, and a spectacular drop in 2018, 2019, with the conclusion that the available studies reinforce the hypotheses that overwintering habitat loss and loss of Central California breeding habitat, as well as pesticide use, are likely to be important contributors to decline. But it's really difficult to estimate the decline because of methodological changes, so that the authors had to use a statistical trick, as it were, to estimate how the pre-1995 samples compared to post-95 samples. And you can see this gray area is basically the error range. So the peak could have been here, it could have been here, could have been here. And how steep the decline, this 97% decline, is based on the very highest point, which overall may well have been an anomalous year. Now, none of these niceties is discussed at great length during media coverage. This is an example of how sometimes the predictions get ahead of the biology. California's most famous butterflies and a death spiral and extinction spiral, an alarming precipitous drop in the western monarch butterfly population in California could spell doom for the species, a scenario that biologists say could also plunge bug-eating birds and other species into similar death spirals. Don't think the biologists were saying that the decline of the monarch would cause a plunge in bug-eating bird populations, because very few birds can handle monarch butterflies, because they famously store or sequester poisonous substances from their asclepidaceous or milkweed host plants. They're called cardiac glycosides or cardinalides. They're heart poisons that induce vomiting. And here you see a blue jay attempting to eat a monarch butterfly, and you see the consequence here. And subsequently, the blue jays that have experienced monarchs will not only avoid them, but anything that even vaguely resembles a monarch. So that's at least one thing you don't have to worry about. People are worried all around the world. And just like politics, the insect apocalypses might be local. There may be local losses of high-profile charismatic insects like monarch butterflies, but what does that mean relative to all insects? How broadly are trends in geographically restricted studies applicable elsewhere? And here you see the concern. Hong Kong is worried about moths. The Insectageddon in New Zealand has us all buggered, as it were. I told you about the bad puns. Here you have a concern in California. Insects are disappearing in India. So everyone has a slightly different take. So the insect apocalypse has really gripped people's imagination, but it's worth mentioning it's not even our first insect apocalypse. If you do a Google Trends analysis, Google Trends is a site or an app, I guess, on Google that lets you survey searches on Google over time to see when people are searching for particular terms. And here you see, this is basically right here is when the New York Times Magazine article appeared, and a lot of interest in insect apocalypses. But then there's this bizarre peak all the way back in 2004. And I'm thinking, I'm an entomologist, I don't remember an apocalypse in 2004. And this was over the top language in a news story about, the emergence of the 17-year cicadas in the Northeast. It's one of the very large broods, brood 9, I think, of the, brood 10 of the periodical 17-year cicada. And that's one headline that got a lot of traction here. The apocalypse was a term that attracted people's attention too, but it referred mostly to 2007 concern about colony collapse disorder. And that concern arose in February of 2007 because, in fact, managed bee populations had declined. And in February, half of all of America's bees are transported to California almond orchards in order to provide pollination services. And again, the New York Times triggered worry about the bee apocalypse with a story that appeared in February in the business section about whether or not the almond crop would be endangered. But there's still bee apocalypse interest going into the present. So we've actually been discussing a crisis of biodiversity since 1992 when Edward O. Wilson, legendary biologist, entomologist, coined the term, or at least first used the term in his 1992 book, The Diversity of Life. That was 27 years ago. And pollinator biodiversity loss became really attracted attention around 1996 with the publication of a book that hasn't really gotten the credit it deserves for raising awareness. There's a book called The Forgotten Pollinators, written by Steve Buckman and Gary Nabhan. And in this book, they basically looked across years of study to demonstrate a very consistent pattern across many different groups of pollinators. So not just birds, but also bees, butterflies, bats, and the like, all of them seeming to decline in abundance and diversity. Well, this started a conversation, and in 1996, the subsidiary body on scientific, technical, and technological advice of the Convention on Biodiversity, which is a global meeting every year, was held in Montreal, led to the establishment of an International Pollinator Conservation Initiative in 96, the third conference of the parties on this Convention on Biodiversity, which was in Buenos Aires, led to the decision 3.2, pollinators became a priority group. October 1998, the International Workshop on Conservation and Sustainable Use of Pollinators in Agriculture led to the Sao Paulo Declaration on Pollinators. In 1999, the USDA and the U.S. Geological Survey joined forces in Logan, Utah to discuss the importance of pollinator loss on managed and wild populations in the U.S. In 2000, in the fifth meeting of the Conference of the Parties of the Convention on Biodiversity, led to an international initiative for conservation and sustainable use of pollinators. April 2002, the International Pollinator Initiative was approved. In 2002, an NGO called the North American Pollinator Protection Campaign approached the National Research Council of the National Academy of Sciences. That's the research arm of the National Academy, the ones that carry out what are called consensus studies. This is an objective group of scientists who examine all the literature on a particular issue and then make recommendations on policy based on evidence. And they suggested a study. The study was approved, received funding, got started, and basically got underway in 2004, which happened to be a year when many different papers came out documenting long-term population declines in all kinds of pollinators. This is a study on British moths published in 2002 that was based on 35 years of light traps. Moths, of course, come to lights. So Rothamsted is actually was founded in the mid-19th century as an agricultural research station in Britain. They showed of 338 large moths, macro moths, over half of them displayed significant declines. Here you see them decreasing. And in contrast, only about a fifth of them showed any increases. And so Britain, basically, Great Britain is very good about its natural resources, and many naturalists there that are amateurs that monitor things, and the whole country is gridded off into 10-kilometer squares. So it's there's a grid system in which biodiversity measures can be mapped and compared. And in Science, there appeared an article comparing survey data on butterflies from 1968 to 1972 and then 95 to 99 across Britain, and found that butterflies had disappeared from 13% of the previously occupied squares. The projection was made, if insects elsewhere in the world are similarly sensitive, the known global extinction rates of vertebrate and plant species have an unrecorded parallel among the invertebrates, strengthening the hypothesis that the natural world is experiencing a sixth major extinction event in history. Again, British bees, Britons are very good at natural history observations, particularly British hoverflies and British bees. In 2006, Biesmeijer et al. Reported declines both in the Netherlands and in Britain, documented the geographic extent of these declines. So here you see the species changes, decreases are the reddish and increases are the blues. And definitely in Britain, bees are in decline and the syrphids are in decline. Dutch bees are also in decline. Syrphids seem to be holding their own in the Netherlands. But this was another awareness-raising paper. In 2006, that National Research Council study was released. I was privileged to chair that committee. There are experts on every group of pollinator on that committee. And what we were able to show after about 18 months and 3,000 references, that yes, there was a very clear downward population trend for honeybees in the United States since data began to be collected around 1945. The number of managed colonies had dropped more than half, so about 5.5 down to 2.5 million colonies. The problem with wild populations, we did not have data. It was a staggering realization that we had failed to keep watch over the status of pollinators on which we depend, other than the managed pollinators. Now, this is these findings relate against the backdrop of a disturbing pattern of accelerated extinction rates across all kinds of taxa other than insects. People tend to overlook insects, but there are consistent patterns across vertebrates of an accelerated pace of extinction. That led people to believe that this was, since this rate of extinction was unparalleled in geologic history, that in fact humans had led the planet into an extinction period. So the history of Earth has been a series of disasters, and these are called mass extinction events, and some go back to the Ordovician. There are basically 5, over which three quarters to 90%, 95% of the known organisms on species on the planet went extinct. And the causes tended to be geologic events. So volcanic activity, increase in methane, asteroid impacts, rapid cooling. And the idea began to gain popularity that maybe humans, for the first time in the history of the planet, were causing extinctions the same way that asteroids, for example, had caused extinctions. So this is a press release from 1998 announcing that this mass extinction event is the fastest in Earth's 4.5 billion year history. That phrase, sixth extinction, sort of gained common parlance, E.O. Wilson was sort of a gifted writer, as well as a remarkable astute biologist, projected these contemporary extinction rates and estimated that by 2100, half of Earth's biodiversity would be extinct. He proposed, instead of the sixth extinction, the age of loneliness, which in its Greco-Latinate roots is the Eremozoic, the age of loneliness. It never really caught on. But in 2000, Paul Crutzen and Eugene Sturmer introduced the term the Anthropocene to introduce this age of human-caused extinctions. So it really didn't take us that long to go from defining biodiversity in 1992 to realizing today that we're in imminent danger of losing all of it. Now, the thing about extinction, particularly since 1700, it's the one form of environmental change in which human involvement is impossible to deny. We have it on, we have photographic evidence, we have documentary evidence of all sorts that we are causing extinctions by human disturbance, recreation, war and work, transportation, energy production, over-harvesting, agriculture, land clearing, urban development, invasive species and disease. And we can't, unlike apocalyptic fiction, blame it on alien viruses, planetary aberrations, or cyclical geologic disruptions. So the more people delved into the literature, the more alarming the picture gets for insects. This is a paper in Science Magazine, very high profile journal, of defaunation in the Anthropocene, which really for the first time brought insects into the discussion. And in fact, of all the insects with International Union of Conservation of Nature documented population trends, a third are declining. 30 to 60% in the UK have declining ranges. Globally, a compiled index of all invertebrates over the past 40 years show a overall 45% decline. And a meta-analysis of the effects of anthropogenic or human-originated disturbances on Lepidoptera shows overall Lepidoptera are displaying lower diversity rather than increasing in diversity, particularly in human disturbed sites. Now, insects may be at particular risk of decline because of two aspects of that characterize the group, the class Insecta. Most insects that go extinct are narrow habitat specialists and are associated with hosts, either plant or vertebrate hosts that themselves have gone extinct. So these are often overlooked causes of extinction. When, for example, the Hoya, which is a New Zealand bird that went extinct, going extinct with it was the bird louse that lived on it, only on its feathers, the only place that lived on Earth. So insects are more at risk because as a group, they're more specialized in their habitats and their habits. And that's how they became so diverse in the first place. They divide the world up more finely than most other animal groups. So moths, which don't get the publicity that butterflies have, more than 90% of North American lepidopterans are moths. As caterpillars, 90% of them feed on 3 or fewer plant families, very specialized. So here's one genus, which you may know, Enothera, which is evening primrose. There are 4 species of this tiny little moth genus. Well, the moths are tiny, the genus Mompha. There are 4 species of Mompha that feed on one genus of plant. And they do this by specializing on different parts of the plants. Here's Mompha rufocristatella, which forms galls on flower stems of gaura, biennial gaura, which grows in Illinois prairies. And here's Mompha stellata, which bores into the flowers on Enothera biennis. In fact, right here in central Illinois, actually, in part in my front yard, my graduate student, Terry Harrison, back in 2005, identified a previously unknown species. So this is, again, a little brown moth, just like the Momphids. It's Agonopteryx, and very hard to tell them apart. And he described in the literature were 3 species of Agonopteryx that feed on 2 plants in the family Rutaceae, the citrus family. There's Xanthoxylum, prickly ash, or Tilia trifoliata, the wafer ash. And Terry noticed that in fact there was a 4th species that was time displaced. It fed on the same hosts, but later in the season. So here's the key, and you can see that if it's feeding on Telia trifoliata, wafer ash, if it's abundant in late June and in late July, it's a new species, previously undescribed. He described it, named it after his wife. And here are the ones that were known previously. So right here in central Illinois, there's insect diversity we did not even know about, because these species are capable of dividing up the habitat and the resources so finely to allow this diversification. Okay, why does it matter if we lose species? Because in terrestrial communities in particular, insects provide ecosystem services. So ecosystem services are the benefits that people obtain from the natural world. And insects are so diverse, they contribute to all kinds of ecosystem services. They provide products, food, water, biochemicals, genetic resources. They form regulatory services, including disease regulation, water purification, and especially pollination, as well as supporting services. Termites are very important in soil formation. Insects are the major recyclers on the planet. Dung beetles, carrion feeders. They provide population regulation for other insects. Predaceous insects keep other insects, plant-feeding insects, under control. They feed vertebrates and they pollinate flowers. Because 90% of insects have a complex life cycle where it goes from egg to larva to pupa to adults, called complete metamorphosis, they can contribute different ecological ecosystem services over the course of their life cycles. They're important prey for vertebrates, including bats, rodents, and birds. Also invertebrates, including moths, spiders, and bugs. They provide ecosystem services such as detritus processing. You don't really think about it, but these caterpillars, these are clothes moths. You find them in the wool in your closet, but they actually evolved to feed on the fur and skin and feathers of dead vertebrates. There's parasites of other, there's some moth caterpillars that are parasites of other insects. There are moth caterpillars that are predators. This one is the sloth moth, which lives on algae that grows on the fur of sloths in Central and South America. It was a mystery from decades as to where the caterpillars of this moth occurred, because they were not on the body of the sloth moth. But apparently sloths defecate once a week. They come down from the trees to the forest floor, defecate, the moths flutter out and lay eggs in sloth dung, and that's where the caterpillars grow. But again, amazing diversity, but the pollinators are what we're talking about. So remember some of the criticisms of the studies with biomass matters. Yes, but it's not species diversity. Species of insects in particular may not be interchangeable with respect to the ecosystem services they contribute. So around the world, most species of figs are pollinated by only 1 or 2 species of tiny fig wasp in the family Agianidae, the fig wasp family. They're tiny. There's nothing else that pollinates them. Figs have a very strange reproductive biology where the flowers are actually in this, it's called a syconium, and the flowers are inside. The wasp can get in and pollinate them. They use some of these structures to raise their larvae, and others some feed the larvae, others are entered, and the wasp will pick up pollen and distribute it. If there are no wasps, there's no pollination. And we discovered that the hard way here in the US. We produce, California produces 98% of America's figs, 6,400 acres, 31,000 tons of fruit, $15 million industry. Commercial fig cultivation began in 1880 with the importation from the Mediterranean, where figs are native, of Smyrna figs. 14,000 cuttings were distributed in 1881. Nothing produced mature fruit. And the reason they did not is that no one knew that the pollinators didn't come with the fruits. It took 8 years for investigators to identify the proper fig wasp species in Europe, bring them back, figure out this life cycle, and allow it to acclimate to California before we actually could have a fig industry. Not just any fig wasp would do. And in natural communities, figs are even more important in that they are keystone species. So the fig tree, they're cosmopolitan in tropical communities. And the thing about the figs is they continuously produce fruit. And everything in their forest communities can eat these fruits. So we got all kinds of vertebrates. We got bats, and we've got primates, and we've got birds, and we've got rodents. Figs fall in the water and fish eat them. There are all kinds of, here are the fig wasps, pollinators, and all kinds of insects eat the foliage. They are what are called keystone species. So just keystone holds two halves of an arch together. The keystone species holds all the pieces of a community together. This is a study that was done in Eastern Australia of fig trees. 84 bird species visited these trees, and surprisingly, 55 of those 84 species were not frugivorous. They didn't feed on the fig fruits. They were insectivores. So insectivorous birds in fig trees increase in abundance when the figs are ripening, and as the figs ripen, here you see the insectivores, figs ripen, the fig wasps come out, and the fig wasps are an important source of protein for these birds. Once the figs ripen, then the fruit-feeding birds come in. So these stingless fig wasps are an important source of food, particularly in drier habitats. And the fact that the fig trees produce ripening fruits continuously all year long means that fig wasps are also available all year long, including times when other insects are not available. So they're not interchangeable. If you go into biology textbooks, invertebrates are depicted as anonymous and interchangeable in food webs. And when we get specific names of these vertebrates, and then when we look at the insects, there's just a couple that are singled out, or they're sort of anonymously compiled in a little corner here. Insects, here we go. How many thousands of species are represented in this terrestrial food web by these two little sad figures? And again, biomass is not everything. Great tits are insectivorous. They feed their young like so many birds do with insects. But this was a study that showed that size matters, at least if you're feeding your offspring and you're a great tit. And what happens is they feed initially as the season progresses on spiders. But after a point, the caterpillars that are present in the community get big enough that it's worth the great tit's time to collect them. So what happens, if you look at the triangles, caterpillars are not popular until they reach a particular mass, then they are collected and fed to birds. So a family of great tits could fail to produce offspring if there are no large caterpillars in the community, because it's energetically not sustainable for them to bring in, to make multiple foraging tests bringing in small species or small individuals of a particular species. So it's not just biodiversity, it's not just biomass, it's also more than biomass in that not all species are equally edible, and some are outright toxic. We talked about the monarch. Monarch and its relative, the queen, both the Danaus species, sequester these poisons, cardiac glycosides, from milkweeds. And they're about the same size, and they share a color pattern with the black swallowtail. They're actually some morphs. There's a lot of variation in color. Often freshmen undergraduates will mistake black swallowtails for monarchs. Fortunately, they don't eat caterpillars, because if you eat monarchs, you get very sick. If you eat black swallowtails, you probably, well, I don't know how you would feel, but if you were a bird, you'd be better off. So species matter. And we know that insects have gone extinct. We know that pollinators in particular have gone extinct. And this was a compilation we did for our study, an NRC study on the status of pollinators in North America. And here is a list of the many documented extinctions, particularly on islands. So pollinators on islands have trouble. And this is the Hawaiian Islands, and very sad stories of extinctions on these islands, particularly in the Leeward Islands. Many extinctions of moth species, more so than butterflies. Again, because of human disturbance. This is the sad story of Laysan Island, where
rabbits were introduced. Not a great idea. The rabbits basically completely destroyed the plant community, which in turn had devastating effects on the bird community. Here you see what this is Laysan prior to the introduction of rabbits, and this is what the same location looked like after rabbits were introduced. Native plants went extinct, and the native insect specialists on those plants went extinct with them. 7 out of 8 weevil species, 6 out of 8 noctuid moths. And in fact, the Laysan noctuid moth, which is now called Hagpina laysanensis, this was endemic, found only on Laysan Island, and it was likely the main species. I'm sorry, my cat just stepped on the. Okay, sorry. It was the main species eaten by a bird known as the Laysan miller bird. It was known by that name because it fed primarily on the noctuid moths known as millers, and probably the loss of the Laysan miller moth contributed to the decline of the miller bird. Host specificity leaves many species vulnerable in their larval stages. You probably know about chestnut blight, a fungal pathogen of American chestnut, first detected in 1905. Within 50 years, 80% of all American chestnuts were dead. More than 200 million acres of chestnuts were eliminated. At least 60 species of insects were known to feed on chestnut, the genus, and at least 7 were restricted to the American chestnut, Castanea dentata. So about half of the 60 species of Lepidoptera, butterflies and moths, known to occur on species in the genus Castanea had hosts in other families, but more than 20% fed only on Castanea species. About 30% were recorded on a few other genera in the family. 7 of the species that were specific for American chestnut were presumed to have gone extinct by 1978. So if you go to Bug Guide, this is what you see, no default photo. These are the 7 species. 2 have since been recovered, but when their host goes, so go the insects. And the problem is we don't even know what's out there. Less than 0.1% of all known species have been evaluated for their conservation status. So there's not a large number of 583 that are known to be, these are fairly old data, known to be at risk of extinction. But when you look out of the total, almost 1 million species. But if you look at the proportion that are threatened compared to the proportion evaluated, 3/4 of every species of the species of insects evaluated for their conservation status are in trouble. So the Endangered Species Act was enacted in 1973 with the purpose to conserve imperiled species in the ecosystems on which they depend, directs the US government to list species as endangered or threatened, designate critical habitat, and to develop recovery plans. Insects were not listed initially. The announcement that butterflies would be added to the list that was not exactly front page news in 1975. The first insect to be listed was the Schaus swallowtail in 1976. Today there are 25 butterflies, which represent, the 25 out of 575, 4.3% of US butterflies are on the list. There's only 1 moth out of 11,000 species of American moths. That's 0.009%. There's also 1 continental bee species and a continental fly among the pollinators that are designated. So this is the moth that's endangered. It was listed in 2000. It's Manduca blackburni, Blackburn sphinx. It was once found throughout the Hawaiian archipelago. It was thought to be extinct until 1984 when it was rediscovered in Maui and Hawaii. And it's thought that it was a victim of a biocontrol program for the tobacco hornworm, Manduca sexta, which looks an awful lot like the Blackburn sphinx. Similarly feeds on plants in the Solanaceae, the tomato family. So a parasitic wasp was brought in to control tobacco hornworms and attacked its close relative, Manduca blackburni, and led to its decline. Why is this consequential? Well, there is a plant in the Campanulaceae called Brighamia insignis, called the alula, is critically endangered. It's endemic to Hawaii. It's found only on sea cliffs in Kauai, where brave botanists have to climb the cliffs and hand pollinate it. Has no known pollinators. Given its floral structure, it was probably pollinated by the Blackburn sphinx and maybe another critically endangered sphinx called the fabulous sphinx moth, right here, which was also thought to be extinct until it was rediscovered. So today, you probably all know about the emerald ash borer, which is a threat to trees in the ash genus Fraxinus. There are more than 20 lepidopterans, including 5 sphingids, these hawk moths, which are superb pollinators that may go extinct if their hosts go extinct. Now, we've known about the consequence of host plants going extinct, but the consequence of some insects going extinct, not just that the plants they pollinate are in trouble, but the vertebrates that depend on them may be in trouble. And we've known this for 20 years now, more than 20 years. This goes back to 12 years ago. Birdwatch Canada, not a scientific journal, pointed out that aerial insectivorous birds seem to be in decline. Here in 2 languages, as befits Canadian publications, comparing the percentage variation trends from past dates till the present. You can see an increase increasing numbers of declining birds. So population declines were more conspicuous in insectivorous birds. And in the intervening years, people have noticed that the decline in abundance of insects is paralleled by decline in birds. Here's a study that was done in Denmark, tropical forest fragments, where birds decline, where insects decline, birds decline. Big study published just last year in estimating, you may have seen it, and since 1970, the past 50 years, North America has probably lost about 3 billion birds. And many of these, in fact, most of them are from taxa groups that depend on insects. So there's sparrows, wood warblers, blackbirds, old world sparrows, finches, swallows, swifts, nightjars, flycatchers, thrushes, and the like. Okay, so in the few remaining minutes, how do we get people to care about this? More than 10 years ago, it was considered challenging to get the public engaged. Because it was considered too complex, too detached from human interests. If you look at Google Trends, insect conservation has, or insect apocalypse has not really maintained its much in the way of a focus on Google Trends, although Save the Bees, rather than the beepocalypse, has continued to trend, which is promising. So according to one study, nearly 90% of Americans are willing to help save animals from extinction, but not sure how to help. So maybe the problem is not that people don't want to help or don't care, they just don't know how to help. And another barrier, I guess, is that there is a small percentage of the population that care and are active, and they're the ones most likely to respond to efforts to preserve or protect biodiversity. So we're preaching to the choir, essentially. We're not reaching the groups that are not yet engaged in conservation. How do we message them? How do we reach them? Well, I think part of the problem is doing something just to save pollinators might seem like a lot of work. And I say this because, well, look on the web. Here you have 3 ways to boost pollination, 4 ways you can help, 5 key ways, 6 simple ways, 7 easy ways, 8 ways you can save bees. At the very least, it might be time consuming. Here's 10 ways to protect the pollinators, or even overwhelming 20 simple ways to save pollinators. That may be too much for most Americans. Some of these tips are misguided. Here from a wiki on how to help save honeybees, this advice: leave beehives alone when you find them. Don't go disturbing the bees or taking their honey or honeycomb, which is good advice, particularly because this is a hornet nest. It is not a beehive. Honeybees nest in cavities. You find them in tree holes. This is the kind of nest a hornet builds. So if you go try to remove honey from this, you will be sorely disappointed. otherwise misguided too. This is you probably all heard about the murder hornet, Vespa mandarinia, which is an invasive species. It has shown up only in a few places in Washington State and in British Columbia. People are in such a panic that I'm flooded with queries from all over the country from people who are convinced that the murder hornet is here. They're called murder hornets, among other things, because they do attack bees, honeybees, and slaughter them unmercifully. And they're large. They can be up to 2 inches long, and their stings are highly venomous. But as far as we know, they're only in the Pacific Northwest in a few limited places. One of the pictures I got, this is actually a moth. It's a Sicilian moth that is a very high fidelity mimic of a Polistes wasp. And here is this is actually a sawfly. It is a hymenopteron, but it is not a hornet. It does not sting. So even creating an action plan.
5 minutes left, May. 5 minutes left.
What's that?
5 minutes left.
Yeah, I'm wrapping up. Creating an action plan sounds complicated. So here's my advice, 7 steps. What should we do? How do we get people involved? Because so many insects are threatened by human activities, could declines be slowed by an inaction plan? Can we convince people to stop doing things they don't need to do that nonetheless involve time, energy, and economic investment that not doing can help save insects? So look at the causes. Benign ignorance may be the greatest obstacle to surviving the insect apocalypse. Could be at least one way to help pollinators to stop doing things that don't really need to be done. This is a disaster. Insect bug zappers, 2 studies I know of. Here's one that shows that over 2 nights in 1999 and 2002, the target mosquitoes, just 27 and 84 on the 2 nights. Chironomids, 9-biting midges, almost 8,000, 10,000 killed in this sampling period. And they don't bother anybody. In fact, they're beneficial. They're important in maintaining aquatic insect ecosystems. Fly fishermen love them because they're food for fish. And yes, some of them are even pollinators, tiny chironomids. Moths, of course, are attracted to light and to bug zappers. And in this study, 405 of them were killed in the sampling periods in 1999, including 62 noctuids, 30 saturniids, and 300 pyralids. As for agricultural intensification and pesticide use, seed treatments, according to a 2014 study, provide negligible overall benefits to soybean production, and yet systemic seed treatments are known to cause problems for pollinators of all, particularly bees, and all visitors to agroecosystems. Lawns, U.S. has more acreage of lawn and turf grass than corn, soybean, orchard, vineyard, and nut trees put together. And per acre, lawns receive more pesticide applications than agricultural land. And yet, not planting a lawn, not paying money for maintenance, not expending energy to mow, can be replaced with flowering bee lawns that require minimal maintenance and maintain pollinator biodiversity. Just not mowing actually increases biodiversity. And some of the plants that are killed by weed killers are actually amazing resources for pollinators. So dandelions and white clover in central Kentucky, 50 different species of insect pollinators, including 37 bee species, and 2 of the 6 bumblebees are uncommon and possibly in decline. Weed laws are hard to deal with. They began proliferating in the 1970s. There is a case to be made for allowing private property to maintain natural communities in place of manicured lawns. Planting species that are more appropriate for the soil and climate considerations of the area. People want to conserve. And even politicians should realize not reversing executive orders can be as helpful as passing laws to protect pollinators. The executive order saving bear's ears, which is a national monument, bear's ears got restricted to a tiny fraction of its, by a recent reversal of an Obama era executive order. And bear's ear monument
that one location has more bee species than the entire East Coast. We still need to protect habitat, but maybe more people can be recruited to not do things in order to help insects. So the concept of not doing anything harmful is venerable. It's the guiding ethical principle in medicine. Maybe it could work as well in insect population health. Searching Google, without insects, humans would die, is 75 million results. Maybe we're woke enough now to prioritize studies of the animals that constitute the bulk of a planet's biodiversity and leave the apocalyptic fiction to fiction where it belongs. And I thank you for listening, and it's 2 o'clock, so thanks.
Hi, everybody. Welcome back. My name is Tane Danger. This is the live stream of the 59th Annual Nobel Conference here at Gustavus Adolphus College in beautiful St. Peter, Minnesota. If you're watching right now, you were just watching a pre-recorded presentation by one of our presenters who wasn't able to be here, but we're very grateful we got a little bit extra sort of in this Nobel conference because they were able to send that in, the magic of technology. But I'm joined by my favorite people like in this whole conference. So thank you both so much for being back here. Do we want to, I don't know. At this point, people probably know who we are, but do you want to just say quickly?
I'm Maggie Kurth. I'm a science journalist.
And I'm Amy Cusick. I'm a biology professor.
And this is one of our last times together because we're, more than halfway through day 2 of Nobel. We've got a couple of more amazing presentations that are going to happen this afternoon. But this is our chance to maybe just do a little bit of debrief. I feel like this is like the 7th inning stretch of the Nobel conference, right? Like we've gotten a lot. So maybe we should just, Maggie and I have had a little bit of time. We had a little bit of time earlier, but what is sort of jumping out at you today from the presentations we heard this morning, and then we had this really robust panel discussion this afternoon?
Yeah. So, I loved this morning. I loved the historical context that Dr. Lesnick put sort of insect consumption in and why we might be hesitant sort of in the Northern Hemisphere to consume insects. And then I think we had this great talk earlier in the day talking about all the ways that insects can be useful to society moving forward and help to solve some of these just really critical problems that we're facing. And then what I loved about the discussion was when we started to think, when the speaker started to think about, how could we revise food systems, which is just this giant question. I think we had sort of a contingent of dreamers, sort of like what's possible, let's dream about what's possible. And then we had a contingent of pragmatists that kind of like brought us back to reality and said, okay, yes, insects are important, but if it comes down to feeding your children or treating insects well, what are you going to choose? How are we going to choose, right? And I think we all know how we're going to choose.
The insects, right?
Right, exactly.
Yeah.
The insects. But I think it's just I think both of those perspectives are so important, and they lend a richness to that conversation. And it made me think back to the Soils Nobel Conference that we had in 2018.
Good callback.
Yep, good callback. When Ratan Lal was here, and he's a world-famous soil scientist, and he said, I'm going to paraphrase because there's no way that I could actually quote such a fantastic speaker. He said something to the effect of, our sustainability goals cannot be met on the backs of the poor.
Oh, yeah.
And I saw that message come up again, right? Like, who do we value in our society? Who's going to pay the cost? And when we think about how much we value farmers, they're not.
It's not high
Right.
necessarily.
It's not necessarily high. And these are critical members of our society that really needs to be elevated.
Well, we've had this conversation several times over the last couple of days about insects being presented as sort of a more ethical alternative to systems that exist right now. And then that also opens up this door of how are we treating the insects themselves ethically? Are we just substituting a new system for an unethical old one, or are we actually thinking about how you're going to make that Nuth system more ethical as well. And that goes down to like everything from like what you're talking about to even just like, if we're going to farm mealworms, what does ethical farming of mealworms looks like?
Right.
Right? Like
Right.
we don't want to end up with like mealworm factory farming, or do we? Does it matter as much? Because I mean, one of the things that the last speaker, the pre-recorded one, was talking about is that difference between how we treat the charismatic megafauna of the world versus how we treat.
That's what they call me sometimes. Yeah.
Oh no!
Nobody's ever called me
You're a
that.
charismatic mini fauna. at
Thank you.
best.
Thank you. Exactly.
Yeah. But just like
the value.
that
Yes.
value that you place
Yes.
on them, right? And like, do we care as much about ethical farming of mealworms, or does ethical farming suddenly not matter as much? Can you just pack as many mealworms into a box and call it a day? You know?
Well, right, exactly. And I think we struggle, right? We struggle with that when it comes to pigs. We struggle with that when it comes to chicken. Like, we're struggling with this, and I feel like we would have a tendency to be very much less sensitive about mealworms than we would about these other
Because they are
organisms.
not cute.
Because they're not cute. You can have a lot in a really little space. You know,
They're-
there's like
they're
certain
small,
advantages
and
to it.
we discount their intelligence already.
We do.
Right.
Yeah, I mean, we have also brought in though the climate impacts, obviously, both for the insects, but then in talking about some of these systems things, there is a piece of like the ethical way that you treat those insects, but nobody's disagreeing that if you were able to move a lot of agriculture towards being based more on insects rather than large cows and pigs and whatnot, that if you're just talking about carbon output, that would have very positive impact.
Right. Yeah. Exactly.
100%.
Exactly. 100%. Like that's more positive. It just it's not that it like I'm not meaning to argue that like, oh, I guess we shouldn't change anything. Nobody do anything. Everybody just hold still, really still, and hopefully we won't kill anything. But more just that like we still have to. It's not permission to turn off your brain and stop thinking about it.
Right.
Right. And I think some of the point that was made in the back and forth too was this is going to happen on some level.
Yes.
Yeah. And it's probably going to happen on different levels depending upon where you live, but this is happening. It's just a matter of like how much.
Right.
I think.
And I think there's also elements of that kind of came in that you were talking about of like, is it still ethical if it becomes a thing where the poor are forced to eat a certain way and everybody else doesn't? You know, like what are the ethics around that?
Right.
Because like there's nothing. There's I mean like one of the speakers this morning discussed is like there's nothing actually wrong with eating insects, and we treat it like there is, but that disgust is cultural. It's not real. But also that doesn't mean that we just get to like push soylent mealworm onto the people that don't get a choice.
Right, absolutely. And I think that goes with, you know, how farmers farm, right? We can't push all of these sort of innovative strategies onto the poor farmers and expect them to do something and sort of ignore industrial farms sort of en masse, right?
Right.
So, I mean, I think all of that is just a very complex question.
And it is one of the things, and Lisa Helke, our director of the Nobel Conference, talked about this morning, that one of the really neat things about Nobel is that it does bring these folks in from different disciplines who they're all thinking about these things, but they have a different grounding and context for where they're coming at it from, because they're like, I'm doing this work here in this way, and somebody else is like, well, I'm working on the same kinds of things, but just in a very different like space and field, and these are the things that I've had to do. And so then seeing that they are having these same kinds of discussions, and I'm imagining a lot of people watching are thinking about these things too, which is what's great, is that like it's not easy. Like this would be a very different conference if we were like at the end of 2 days we all know the answer.
Absolutely. Nothing is really resolved. It's sort of just like the struggle of the processing, right?
Yeah.
And allowing these people space to sort of think in slightly different ways. Like leave your silo for just a little bit. Let's really think in ways that are maybe a little bit more abstract than what you're usually
Right.
allowed to think of, and let's see what happens.
So we're about to go down to the presentation. We are going to get started here with a prelude by the Gustavus Wind Orchestra. And then this afternoon, we've got two more talks. Jessica Ware and Michael Young are each going to speak. And so with that, throwing it back to Lisa Helke, director of the Nobel Conference, who's going to introduce, I believe, the Gustavus Wind Orchestra.
Under the baton of James Patrick Miller. Thank you.
Bravo!
Thanks to the Gustavus Wind Orchestra, James Patrick Miller, and thanks to all 4 of our ensembles that have feverishly worked to be ready for this event that comes way too early in the year for them. So thank you so much. What a wonderful, one of the parts of the conference that to me is really important as an old band member at Gustavus Adolphus College. A final welcome to you this afternoon. I want to thank a lot of additional people. First off, the beautiful visual design of this conference, we call it the poster, but as you can see, it appears everywhere, was done this year by Jenny Kurth of our marketing department, and I just love what she created. It turns out it's not necessarily easy to convey what we wanted to convey. For instance, those legs on that insect holding the world and not crushing it, that was really masterful on Jenny's part. So Jenny Kurth of marketing, thank you so much. I want to thank the many folks who have made this conference run smoothly in there. We call it behind the scenes, but of course it's very much out in front. And Anna has given us many more ways to think about the role of what we call ecosystem services, perhaps, but in a college campus. our other human beings who are making sure that we all are able to be where we need to be, well-fed and tended. So thanks to physical plant, thanks to custodial services, thanks to campus safety, thanks to dining service, thanks to all of you who have, and thanks to the many people who sort of suspend their day jobs to come here and be ushers of the conference. Thanks to all the student sports teams who staffed things like the Learning Lab and parked cars and all the things. It really does take a whole college campus to put this together, and I'm going to start to cry. So let's move on. But first, let's give them all a round of applause. And back to Basho. The dragonfly can't quite land on that blade of grass. So with that, I invite Janie Franzen, my colleague in the Department of Biology, to come to the stage to introduce our next speaker.
Good afternoon. It is my pleasure to introduce Dr. Jessica Ware. She is an associate curator and the division chair for the Division of Invertebrate Zoology at the American Museum of Natural History in New York City. We are excited to have Dr. Ware here to talk about the biodiversity of insects. As an entomologist and evolutionary biologist, she documents diversity in insect populations and assesses how these populations vary over time and geographic location. Dr. Ware's research focuses on several different types of insects, from her self-proclaimed favorite, the majestic dragonfly, to those the general public is more likely to have negative connotations with, like termites and cockroaches. For her research, Dr. Ware was awarded the Presidential Early Career Award for Scientists and Engineers, the highest honor bestowed by the U.S. government on outstanding scientists and engineers beginning their independent research careers. Dr. Ware is also a proponent of encouraging people from underrepresented groups to become entomologists, and has co-founded the organization Entomologists of Color and co-organized the hashtag Black in Ento Week. Please help me in welcoming Dr. Jessica Ware to the stage.
All right. I'll bring my water here in case I have a fit of the coughs, but hopefully I won't. I'd like to thank Lisa and Margaret for the invitation to speak here today. I'd like to thank Janie as my faculty host and the student hosts and super hosts who have made the last couple of days so wonderful. So as we've heard over the last 2 days, insects are a remarkably diverse group of animals. They're charismatic, beautiful, and perhaps facing peril. We talked yesterday about the proportion of insects that would make up the US Senate. This is a pie chart showing just animals. And when we look at just animals, almost all animals are insects. They're the kind of pink-colored part of this pie chart. Indeed, when we look at sheer species number, there's around the same number of species of mammals as there are dragonflies and damselflies. 1.5 million described insect species, but when people try and make estimates of how many species are yet to be discovered, we think maybe there's 5 million or 10 million, or in the past, former colleagues used to say perhaps even 30 million species of insects yet to be described. So perhaps because we're used to this message, there's so many insects, perhaps because when we go fishing or canoeing or swimming, we see dragonflies landing all around us, and perhaps because we're sometimes surrounded by an abundance of insects that we don't really like cockroaches, that gives us this false impression that insects are doing okay. Maybe there's even too many of them. That's not true. Insects are in decline. We know that insects are disappearing. Certainly, this isn't news. I mean, there are publications from the early 1920s that were asking the question, should we be looking at the impact of the Industrial Revolution on insects? In the 30s, in the 40s, in the 50s, people were bringing up this question: what is the impact of human agriculture? What is the impact of the Industrial Revolution on insects? And it wasn't really kind of capturing human attention until perhaps around 2017, when there was a landmark study that had looked at the weight of insects, the total biomass of insects that had been collected over a 30-year period in Europe, and they showed that there was a remarkable decline, up to 70% decline in total biomass of insects that were collected over a 30-year period. And that led to these big dramatic headlines: insect apocalypse, insectageddon, insects are disappearing. Even my Nana, who's 97, who lives in Northern Ontario, she heard about it, and that's saying something. So that leads us to this question of what life would be like without insects. And my colleague Doug Tallamy would say it wouldn't exist. Life as we know it would cease to exist. He estimates that perhaps humans might last about 3 months in the absence of insects. So when we talk about insect decline, we're not just talking about an academic question, what would happen without insects? We're actually talking about our own fate, right? The fate of humanity. So before we talk about insect decline, it can be really helpful to kind of get to know the insects that we're about to talk about. So we've done a lot of work trying to understand the evolutionary history of insects. And in particular, I've been interested in finding out the when question. When did insects first evolve? So with colleagues, we sequenced a bunch of transcriptomes. Transcriptomes are the parts of your genome that are being transcribed at any moment in time. And I like to think of it like cooking supper, right? When you go to cook supper, you don't cook every single recipe that's in your cookbook, right? You just cook the one recipe you need for your one meal, right? And that's kind of what your genes are doing. Some of your genes are being expressed, and some of your genes are not being expressed. So when you sequence the genes that are being expressed at any moment in time, you're sequencing transcriptomes. And with transcriptomic data in 2014, we were able to show that the earliest winged insects were around 400 million years ago. This was during the early Devonian, when the world looked something like what these kind of globe representations are here. Before birds, before bats, before pterosaurs, there was nothing in the sky. The first thing to fly was insects, and probably it was something that looked a little bit like a dragonfly, maybe like a mayfly. To put this in the context of where we fit into this story, that means that for around 396 million years, insects existed in the absence of humans. We've never known life without insects. For as long as there have been humans, there have been insects. But for the majority of insect time on Earth, it was in the absence of humans. So we can look at these types of data, and we can reconstruct what's called a tree or a phylogeny. These are kind of maps that help us understand relationships of how close relatives certain groups are than others. This is kind of a cartoon, and in this cartoon, the camel and the bird are connected and joined and share a recent common ancestor, and they're more distantly related to the fish, which is connected with a more distant common ancestor. So we can kind of look at phylogenies and understand that things that are closer together in a phylogeny share a more recent common ancestor, and things that are further apart in a phylogeny or a tree have a more distant common ancestor. So we can make trees of life of anything. You can make a tree of chocolate. You can make a tree of sandwiches. You can make a tree of rock and roll music and figure out how language evolved. But you can also make trees for insects. And this is what we did using transcriptomes. So just to walk us through the tree of life of insects to figure out how they're related to each other, we have things like silverfish and firebrats, which are basal hexapods. Anyone who's ever lived in a dormitory, probably you've seen silverfish. And they're sister to the winged insects, or the pterygoda, which we divide into two groups. The non-holometabolous insects, these are things that have incomplete metamorphosis. A baby grasshopper looks kind of like an adult grasshopper. It molts to a larger and larger size until it's the adult stage. And this is in contrast to the holometabolous insects. These are insects that have complete metamorphosis. An egg becomes a caterpillar. The caterpillar goes into a pupa where there's a complete internal and external rearrangement of its morphological anatomy, and then it emerges as a butterfly. And what holometabola do very well is they've kind of compartmentalized their life strategies, with the juvenile stage focused on growth and the adult stage focused on dispersal and reproduction. There's more holometabola than there is anything else. So since we published this study in 2014, we've been steadily sequencing, and we have about 2,000 insect transcriptomes now. The tree that we get, the relative relationships of the different insect groups, largely hasn't changed. But I wanted to show it to you just to zoom in to this basal part of the tree, which is the part that I mostly focus on, and it gives me a good chance to do a segue into dragonflies. So dragonflies are striking. They're highly visual predators. I've heard my colleague once called them vicious. I think they meant to say voracious, but I like the word vicious because they are vicious predators, even though they are so beautiful. I study these in the tropics. I study these in deserts, in the Arctic. They're really found on every continent except for Antarctica, although we have fossils from Antarctica. We know that they're diverse. Sure, there's not as many dragonflies as there are beetles, but there's about the same number as there are all of mammals, but we spend so much time focusing on mammals. Odonata, which is the order that comprises dragonflies and damselflies, include dragonflies, damselflies, and this third group that doesn't have a common name called an Isozygoptera that sort of looks like a dragonfly in its body and has wings like a damselfly. We know that dragonflies and their kin are quite old. There are these things called griffin flies, which we call proto-odonates or pre-dragonflies, and these flew in the Carboniferous period around 350 million years ago. If you've ever heard people say, oh, didn't insects used to be very large? Often what they're talking about are these large dragonflies. They had about a 2-foot wingspan, and they had a beak, and they probably were pretty poor flyers, so you wouldn't need to be afraid of them. You could swat them away fairly easily, I'm sure. Modern dragonflies, we think, are much younger, and actually much better flyers. So we know that dragonflies, or something that was dragonfly-like, probably flew first. And in fact, we know an awful lot about dragonfly flight because we focus so heavily on perhaps one of their most remarkable characteristics, which is their wing venation. We focused a lot on wing venation, thinking it might be a clue to understanding the revolutionary history, when actually it's correlated with flight. It actually tells us more about the style of flight that dragonflies have. So we developed AI. We developed an automatic feature extraction that allows us to, when we scan dragonflies' wings, extract the information about particular patterns of veins and how they might be correlated with long-distance flight, fast flight, maneuvering in and amongst vegetation, more dense wing venation and parts of stiffer wing, sparser wing venation, a more bendy or flexible wing, and we can look across species of dragonflies and look for patterns. And of course, these patterns are beautiful in and of themselves, and we've used them in beautiful things like Tiffany. You know, Tiffany really made very biologically realistic dragonflies in some of their Tiffany lamps. I mean, you could almost get this to species, not quite, but pretty close, so kudos to Tiffany. When we look across these wing vein patterns, we can actually kind of come up with a gestalt, right? A certain style that a certain wing shape might have that's associated with a style of flight that allows us to make predictions and retrodictions of the way ancient dragonflies may have flown, and look for some of the stressors that we might predict to find in dragonflies flying in certain areas. We know that dragonflies can be great models for flight. I think yesterday, and even earlier today, people talked about insects being used for bio-inspiration. And dragonflies are no different. There have been dragonfly-like robots that have been made that fly like dragonflies, dragonfly-like robots that have wings like dragonflies. They're very agile, and some of them can fly up to 30 miles per hour. Some of them never leave the pond from which they emerge, maybe traveling 11 meters their entire life. And others, like Pentella flavescens, which is this one that's shown on the screen, they travel 11,000 kilometers. They travel across oceans, across the Indian Ocean, and our data seems to suggest actually across the entire globe. So I noticed that when I talk to the student hosts, there's a lot of biochemists, students that I've met. We can actually use biochemistry, the biochemistry of insect wings. The molecules that make up the insect wings actually come into the wings during the juvenile stage when they're in fresh water. The hydrogen, H2O, comes from the water, and the weight of hydrogen actually varies as you move around the globe. So you can look at the biochemistry of wings, and it tells you whether the dragonfly that you collected in Guyana came from water in Guyana, or whether it maybe emerged in Japan or Australia or in Equatorial Guinea. And when we look at cross-migratory dragonflies, we find that most of them that we collected actually were born on different continents from where we found them. So they're traveling really vast, huge distances. One thing that might make us want to love dragonflies even more is that they eat the things that we don't like. They eat flies. They love horseflies, black flies, mosquitoes, no-see-ums, midges. All the things that we tend to think of as nuisances, they consume both in juvenile stages, because many flies have aquatic juvenile stages, and in their adult stage. There's a story from the Kojiki, an ancient Japanese text, that talks about an emperor that was bit by a horsefly, and a dragonfly came and ate that horsefly. And the emperor was so pleased that one of the names that he gave for the islands of Japan was Islands of Dragonflies. And many ancient Japanese armor actually was kind of etched with metalworking of dragonflies because they were a symbol of power and of agility and of precision because they have a very good, very high catch rate when they're hunting their prey. We know from the literature, my colleague Ola Fink and others have shown that they do an excellent job at controlling Anopheles and Aedes aegypti. Why can't we just use those to control malaria, dengue, yellow fever? Well, dragonflies are very picky. It's been rare that people have been able to rear dragonflies in large enough numbers that you could actually deploy them for integrated pest management. They don't like to breed on command, and they don't like to breed in large numbers. They tend to just stay as juvenile stages forever, never emerging as adults. So this pickiness is something that we can actually use when we study dragonflies. This is what a female dragonfly, I don't know if I can go back. That was a quick snapshot of what a female dragonfly looks like when they're laying their eggs. Females lay their eggs either in still water or in fast-moving water. And females are somehow able to distinguish between water that has high turbidity, low turbidity, high oxygen levels, low oxygen levels. Some dragonflies are very picky. They require very specific requirements. And others, I've seen this, are able to oviposit and develop in a puddle in Newark, New Jersey, in a parking lot at Rutgers. So there's a huge variety in the tolerance to water quality. So that makes them great biomonitors. Fresh water is a resource that is limited on our planet. And this is a free tool that allows us to kind of assess the water quality in our area. Dragonflies are a great biomonitor. This is what the juvenile stage looks like. Not only can they take aquatic insects, but I just wanted to mention they can also take vertebrates. Like, they're no slouch. They can take small minnows, they can take tadpoles in their juvenile stage. Of course, they're food for us. We've talked about this earlier today. Dragonflies do make great food. I've eaten dragonfly food. All that flight muscle is basically pure muscle. Just break off the wings and the abdomen. That flight muscle would be a really good sustainer. I think if I was ever lost in the woods, that's what I would eat. And of course, they're great food for birds, reptiles. I've seen fish jumping out of the water, frogs jumping out of the water. In fact, these predators we think are what maybe led dragonflies on this path to being excellent and very agile flyers. So what that means is that if dragonflies, like other insects, are declining, that means this really vital food source for migratory birds, for reptiles, for mammals, is also declining. Which leads us to this question that if dragonflies, like other insects, are disappearing, what does that mean for life on Earth? We know a little bit about what the dragonfly tree of life looks like. Again, we can sequence different parts of the genome. We've sequenced thousands of genes for dragonflies and damselflies. Our goal is to sequence all of the 6,400 or 500 species of dragonflies so that we can understand their relationships. And we think, using molecular data and fossils together, that perhaps damselflies first arose around 225 million years ago, and dragonflies similarly arose around 225 million years ago. Again, around 220 million years before humans really came on the scene, before we started changing water flow, damming rivers, and polluting water sources. How do we know all of these data? How do we know about the biochemistry of wings? How do we know about the genomes of dragonflies? How do we know about flight? We know because we have specimens. This is a photo of where I work, the American Museum of Natural History, and it's just one of many natural history museums that exists around the world. We have large collections of different animals. Often, the majority of them are insects because there are more insects than there are other animals on this planet. And they really are invaluable resources. But they come with a dark history. Collections, like the collections at the museum where I work, were largely acquired during a time of colonialism. We know that many of the collections that we have were actually collected because there was wealth in the global north that allowed folks to go to different parts of South America and Africa, harvest as many species as possible, and bring them to be housed in the global north. Brandon Kilbourne, who's a friend and colleague, has written several poems about this kind of unnatural institution of natural history, talking about how the very transatlantic slave ships that moved enslaved people back and forth across the Atlantic were often used to transport natural history specimens from South America, from Africa, back to Europe, back to North America. He talks about this idea that you could have something so beautiful as a jeweled beetle existing on a ship that was carrying enslaved people to horror, right? Which was a slave labor camp, which was a plantation. That's how my father's family first came to the United States. So we have this complicated history when we talk about colonialism in entomology, when we talk about natural history collections, when we talk about access and who has access to the specimens that we've collected. So take, for example, this beautiful Lariathemis. This is a dragonfly that was collected in 1939, and most of our insect specimens have information about where they were collected and when they were collected. This was collected as part of the Archibald expedition. And Archibald was a philanthropist who in the 1920s and the 1930s used his funds, which were largely gained by his grandfather, who was one of the first oil refiners in the United States, to fund expeditions to Madagascar and to New Guinea. It's wonderful that we have these specimens. They're largely housed in the American Museum of Natural History and in the Leiden Museum and Naturalis in the Netherlands. But many of my colleagues who live in Madagascar, who are Malagasy, or who live in New Guinea, have never had access to these specimens. It might be cost prohibitive for them to come to the United States, and certainly with huge collections like what we certainly haven't even begun to scratch the surface of digitizing the collections that we have. We don't always lend out specimens, and we certainly don't lend out what we call types, or the specimen that we use when we're describing a new species. But nonetheless, we have these specimens, and we want to try and use them to use this invaluable resource and this knowledge. So one thing we can do with natural history collections is we can use the information from these cards or from the labels that insects have, and we can use them to create ecological niche models to predict where species should be found and predict where species might be found in the future. And here's a couple of maps that we've made using natural history collections and other digital collections for dragonflies and in the Arctic. And these ecological niche maps are kind of color-coded by richness, where yellow means that there's high species number, and dark purple means low species number. Lower species number as you go towards the Arctic, with a concentration of diversity very high in the Northeast. I got to say, New Jersey, people never think of New Jersey. We have 188 species of dragonflies in New Jersey alone. Very impressive. We can use these models under different climate scenarios to actually predict what might happen in a future that is X warm, this much warmer, that much warmer, or with different values of precipitation. We can do this for dragonflies because people often like to collect beautiful things. We have far fewer collections of maggots. We have far fewer collections of earwigs. We know a lot about some insects and very little about others. So take, for example, this beautiful bug. It's called Nasoconia ellenfutteri. We just described it this year. It's a tree-sucking bug. It's a bug that has a mouth part. It's actually very beautiful because its mouth part is golden. I'm not sure if you can see it kind of pointing downwards. And it actually feeds on xylem. This was collected in 1985 by Terry Irwin, who's a colleague, late colleague, who had this method. He wanted to try and sample canopy insects. It's very hard to reach the insects in the canopy. Our nets aren't large enough to go all the way up into the canopy. And so he designed this method where he would fog with insecticides, the canopy. Insects would die and then fall to the ground, and he would gather them all up, and then they would sit on the shelf for someone to eventually look at them. And we did eventually look at some of these bugs, and we found that this is indeed a new species. As far as we know, the only individual of Nasoconia ellenfutteri that's ever been collected. Where do we describe this as a new species, and we chose to have this species, the holotype, go to Ecuador, go to the home country from which the specimen was collected, so that way Ecuadorian researchers would be able to work and study on it. And we got some pushback from our colleagues in the States. Why would you send this only representative of this species, there's no paratypes, why would you send it to somewhere other than the American Museum or the Smithsonian? And people clutch their pearls. But it's because we can make decisions as collection people, we can make decisions as taxonomists to really include our colleagues from the places where we're collecting these insects as part of the conversation as decision makers in systematics and in taxonomy. Termites are another group that we know a lot about, a few termites, and we know almost nothing about populations of the non-pest termites. There's over 3,000 species of termites, and termites are actually just social cockroaches, or just kind of fancy cockroaches that happen to be social. And cockroaches in general, very small percentage of them are pests. The rest of them live outside of the human condition. Detritivores, ecosystem engineers, shaping our planet. Our planet would look very different in the absence of these social insects. We don't know very much about what a population is. Is a colony of termites a population? Are multiple colonies of termites a population? How are termites doing in terms of climate change? We really don't know. I just came back from Guyana, where we have been sampling termites in the Amazon for some time. And some of the termite mounds that we have sampled over a series of years, because it's been a particularly hot time in Guyana, because there's been unusual amount of kind of dehydration. There's a dry season that normally starts in December. It's already begun. There was things that were aflame, termite mounds that were burnt to a crisp. We really don't know the impact of climate change on social insects. And then, of course, there's things that fly at night. So I am an early riser, which means I go to bed very early, which means I often don't study things like what's shown in this photo. You can probably barely see them, but there are dragonflies that are flying here over this lake in Ontario. There are these dragonflies, shadow dragons, which fly when it's too dark to read a newspaper. Much like many of the insects that are nocturnal, we know very little about their populations. We don't know how they're faring, because entomologists, we go to bed at 9 p.m. We don't like to be out late, so we have far fewer samples in our collections and far fewer information about their behavior, just because we are diurnal. So these are tough questions that we as systematists, as collections people, and I hope we'll discuss on the panel today, these are tough questions that we have to face. Who has access to museum specimens? Where should specimens from the Global South be housed? If there's DNA in there, whose DNA? Who has rights to that DNA? Should we be digitizing everything? Who's going to do that? We have 23 million insect specimens in the American Museum of Natural History. Even if I was to devote my entire life to doing this, I would never finish. I would die. Why isn't everything databased? How can we increase access to collections? These are questions that are tough questions that we don't have answers to yet. This is morally complicated than the topic of insect decline, of course, because we need answers for this really, really fast. We want to start using these specimens right now so that we can come up with a solution for insect decline. And indeed, the questions that people have about insect decline are global questions. These aren't questions just for someone from Toronto. These are questions no matter where you are on Earth, because this actually affects the survival of Homo sapiens sapiens, the survival of our species. These are universal global questions that will require all of us to participate to answer them. So my colleagues made this paper called Death by a Thousand Cuts. It's a really good read, and I would cut out this and put it on your fridge and use it as a checklist. It talks about the main drivers of insect decline, and you can go through one by one and think about how the things that you do in your day-to-day might be impacting some of these drivers. And I found this to be a really good conversation starter at the supper table with my teens sometimes. They don't always want to talk at the supper table. With Dave, with Christy, with Chris, with Eliza, we actually have a grant where we're studying kind of synthesizing the data that's out there for insect decline, trying to understand and pull together resources in different languages from across the globe. The majority of studies that are often cited and quoted are actually from the global north, when, as was spoken about earlier, the majority of insect species are around the equator, right? So we actually need to prioritize, because of these historical inequities, the voices of indigenous people and the voices of people in the global south. So what can we do as individuals just here sitting in Gustavus? Well, first thing I would always say to people is we gotta kick our addiction to lawns and cars. We can do simple things like be compassionate and plant gardens that have insects in mind. We can have habitat that is has heterogeneity. We can have food for pollinators. It's great that people care about pollinators because it actually infects even the lowly cockroach, because some cockroaches are pollinators as well. I do a lot of work in the Arctic, and we know that there's impacts due to global warming in the Arctic. Many of the dragonflies that we study actually live in lakes that are called permafrost bottom lakes. Permafrost is kind of a fancy word for the mixture of ice and dirt. Part of it stays permanently frozen, and part of it kind of melts and freezes in this active layer every year. As permafrost melts, then often these lakes completely disappear. It has this added negative bonus, or deficit, I guess, that permafrost actually stores a lot of carbon. So as the permafrost melts, it releases this greenhouse gas into the air, which further exacerbates the problem of climate change. So for Arctic dragonflies, when we go to the Arctic, and we've been sampling in Scandinavia, Alaska, Yukon for many years, we see that the Arctic is changing rapidly, and dragonflies like Somatochlora selbergi, this dragonfly that's shown here, is doing fairly poorly. We see dragonflies that used to only be found further south, moving into the Arctic and oftentimes outcompeting native species. So we know what some of the causes are of greenhouse gas. We've known this actually for a very long time. But it's very hard for us to make decisions because some things are out of our hands. As was mentioned earlier today, we need to put pressure on those that have decision-making powers. We need to put pressure on industry. And there's a small little part at the bottom, residential buildings. That's where my teenagers and I fight every year when I have the heat on and they open the windows because it's a little hot, or when they want to have the lights on in every single room. Those are types of decisions that I myself can make, but that's a very small part of the energy use, right? We actually need to hold those accountable who are kind of causing some of these, or exacerbating some of these problems. We can also do a better job at getting to know the insects that are in our neighborhood, so that when we have invasive species, we know that invasive species are one of the drivers of insect decline, but we often don't find out that they're here until it's too late, until they've already started to establish. Take, for example, the spotted lanternfly, the scourge of New York this summer. They were kind of everywhere. And since they've arrived in the United States, people have tried to eradicate them, but perhaps by the time we found out they were there, it was too late. And I think one major part is that we're disconnected from what we expect to find. When we go for a walk to work, when we go for a walk to the park, we don't know what insects are supposed to be there. So if there is an invasive species, which are inevitable, because we have transoceanic commerce and we're in a port, New York, New Jersey, Pennsylvania, gets a lot of shipments. When we see something new that's not supposed to be there, we don't even recognize when it's not supposed to be there because we don't know what our neighborhood insects are. So when my kids were little, these are some photos of them when they were younger, I used to take them out and kind of have fun with them to collect insects, and really tried to encourage them to get to know their backyard fauna. And when I took them with me to Guyana, to South America, we didn't have a fancy insect collection. We had an old bingo box and some upholstery foam and some sewing pins that they used to make an insect collection, and that allowed them to really get to know the insects that were around the field station where we were collecting. Now, not everybody's going to want to make an insect collection, and perhaps after our discussion yesterday, maybe we shouldn't be pinning insects, but we can still make digital collections. There are apps like iNaturalist that will allow you with your phone to kind of, as you're walking to your regular coffee shop, snap photos of insects along the way, so you get to know what you should be expecting to see on your coffee walk. So if you see something that's not supposed to be there, or if the app says nobody else has found this, you can alert your local entomologist and say, I found something that I don't think is my neighborhood entomology fauna, and that can help us prevent things from establishing. And perhaps most importantly, of course, we can vote, right? Our vote actually matters. We should hold all of our elected officials, whether it's my school board representative, whether it's my town mayor, or whether it's my president. I should be expecting them to vote with insects in mind, because insects matter to me, and I think insects matter to the fate of all of humanity. This is really an all-hands-on-deck situation. It means that I need to be involved. We all need to be involved. My Nana in Northern Ontario needs to be involved, even though she's 97. There should be no barriers to participation. And yet, there are. So my kids would say, oh, that was terrible, man. You used to make us go and do all those collections, and what was the point? Those are data, right? I remind my kids, those are data. I now have these data of what was existing in Cranberry, New Jersey in 2008, thanks to you kids, right? We can actually, everything that we do can have consequence. We really need everybody. We need my kids' insect collection. We need the grade 2 monarchs that we were talking about at lunch. We need all of these data because they actually are going to help us kind of understand this bigger picture of what's happening to insects worldwide. And this is not the time for there to be barriers to participation. And yet there are. So we've worked with colleagues to create this foundation called Entomologists of Color, and our goal is to try and diversify the field of entomology in a time when we're facing the crisis of humanity, perhaps our biggest crisis. Why would we have barriers to participation for certain people to participate in entomology? So as part of this group, we do recruitment, retention, and advocacy to try and change the system, to make things less toxic, and to kind of address some of the historical inequities that go along with a long history of colonialism in entomology. And importantly, we need to listen. We need to listen to our colleagues from the Global South, my colleagues from Jamaica, from Barbados, from Guyana, from Nigeria. They've written articles, they've had webinars and series talking about colonialism in entomology, neocolonialism in entomology, helicopter science that's happening right now. And we need to listen. We need to listen. We really just need to listen. We can also look even to my own discipline of systematics and look at who's in systematics and natural history museums. Adania Fleming, who's a graduate student at the University of Florida, she kind of got a bunch of us together and we formed the Black in Natural History Museums group, which now includes the president of the American Museum of Natural History, Sean Decatur. And as a group, we kind of have been talking about who has been doing natural history. And it's always been everyone, right? There have been natural historians who have existed for as long as there have been humans, but who has been TM natural historian at a natural history museum hasn't always looked like people that look like the people on this slide here. We have a new exhibit. It's opening at the University of Florida Natural History Museum, where we're kind of highlighting people who you may not have heard of, who are kind of greats in natural history, including John Edmonston, who was an enslaved person from Guyana, who, when he was brought to England by the person who controlled his fate, he actually taught Charles Darwin how to do taxidermy. You know, there are many stories of people who have been doing natural history since human time began, and we want to try and highlight those folks so that people realize this is a job for everybody. We need systematists and taxonomists now more than ever. Why would we ever put barriers to participation? Insects are essential. And I think what we've learned over the last couple of days is that it's an all-hands-on-deck situation. Museums are essential. And I think what we learn when we look at, kind of peel back the veneer of museums, is that they're also very complicated. They have complicated histories that we haven't begun to really address or discuss. As we've talked about, what we need are global solutions. This is a problem that faces every human on Earth. The decisions that we all are making are actually global decisions, so we really need global solutions. I'm so grateful to Nobel 59 for highlighting insects for 2 full days, because hopefully that inspires all of us to kind of join in this fight to save insects. So with that, I'll say thank you.
Thank you, Jessica. We have a couple of minutes to stand and stretch in place before we prepare to welcome and honor our next speaker.
Hey, we are back. I am Maggie Kurth. I am a science writer, and I'm here.
Yep, I'm Amy Cusick. I'm a biology professor at Gustavus.
And that was just that was a really interesting discussion that she just had. I really love the idea that, like, even the dead bugs come with ethical complications.
Absolutely. Absolutely. Very unresolved and sort of, newly kind of thought about. What are the ethical implications of having these collections? How were they historically collected? Mm-hmm. Should we be giving collections back to their home countries? These are complex questions.
Well, and I also didn't really know the history of how the early zoologists who are going around collecting these samples that now reside in our national museums were traveling along on slave ships.
Right.
Like, this is information that's just completely left out. And it's something that I think really matters when we sort of start thinking about the institutions that allowed science to happen, which are not always clean and neat.
No, absolutely. And I think, you know, it wasn't only animal collections, but we had this problem in botany also, really similar dynamics with collecting. And then thinking about who were the original knowledge holders. You know, how was that knowledge transferred, and how is that knowledge then acknowledged later on, right? Because now we have sort of like the colonial collector's name, but we no longer have any record of sort of where they learned these things.
Yeah, there was actually a TikTok I was listening to this morning before I came over that was talking about that we use the term scientist only to refer to the Europeans that showed up, but that's not really true. And you have like all of these examples. I think he was talking about a type of shrew in Australia, but like of native peoples, native scientists who were introducing the European scientists to this idea of like, look at this cool thing, look at how it does. We've done these experiments. We understand like, how this thing works.
Right.
And that's where this knowledge came from and sort of how it was built up. And we either don't think about that at all often or we leave out the scientific agency of the people that first made these discoveries.
Right, or we somehow belittle any data that wasn't experimentally in our constructs of what an experiment is. Gathered in such a way. So why are not oral traditions counted as data, right? These things that have developed over thousands of years, those are actually data sets, right?
And often experimentally acquired. Just,
Absolutely.
It's just like a different set of rules, which is, there's some of the history of like, this is slightly off topic, but like some of the history of how like Western science developed the scientific method is super fascinating. And there was a speaker I watched a few years ago at a different conference who was talking about how some of the very earliest science research was based on like artisan craftsmanship. So you would do an experiment like 15 times, and instead of doing an average, you would pick the one that looked the nicest, just like you would pick the nicest chair or the nicest table that you made to display. And so it's not like it's not like our culture like birthed this into existence without any like things that we would now look back on and be like,
exactly. Exactly. And I think that's also I think we're heading back.
Yeah, I think we are actually heading back into
but
the
it'll
thing.
be
No, we can
preceded
pick
by
this conversation
a
up
special
after the next
ceremony
speaker.
with which we choose to honor when we are so fortunate as to have in our midst a Nobel laureate. We grant them an honorary degree, and we're very fortunate to have Dr. Michael Young with us during this conference. And so I wonder if I could ask the panel to join us. Dr. Young, Dr. Ferragamo, President Bergman, all to join me on the dais here. And on the panel here, or I'm sorry, I keep calling it the panel. On the dais, I have Margaret Block-Causey, the chair of this year's conference, Dr. Mike Ferragamo in the Departments of Biology and Psychology, who will read the citation, President Bergman, and me. Okay, so Mike.
Hi. Today, Gustavus Adolphus College honors Dr. Michael Young, the Richard and Jean Fisher Professor in the Laboratory of Genetics at Rockefeller University, where he also serves as Vice President of Academic Affairs and where his work has been supported by the NIH Merit Program and the Howard Hughes Medical Institute, each granted exclusively to experienced investigators who have a record of superior research. Dr. Young is a member of the National Academy of Sciences and a fellow of the American Academy of Microbiology. The preeminence of his contributions to the advancement of scientific inquiry have been honored by numerous prestigious awards, that include, among many others, the Louisa Gross Horwitz Prize, the Massry Prize, the Shore Prize in Life Sciences and Medicine, and the 2017 Nobel Prize in Physiology or Medicine with co-recipients Drs. Michael Rosbash and Jeffrey Hall. Professor Michael Young's career has been devoted to unraveling the molecular and biophysical mechanisms that organize the daily rhythms of life in the fruit fly, an organism widely used as a model system in the life sciences. He made the remarkable discovery that the activity of genes and the levels of the key proteins that they produce wax and wane to the beat of a 24-hour period. Every one of our cells has a clock controlling its actions. Some cells work best at night, and some during the day. This knowledge has had significant implications for medical practice. For example, by allowing precise timing of medications, or in mitigating the health consequences of shift work. However, as individuals, we are more than a collection of cells. We also are social creatures. And interaction with others can alter the timing of our daily activities. Very recent work from Dr. Young's lab show that the humble fruit fly can provide insight into this broader human experience too, as he and his colleagues itemize the costs of social isolation on physical and mental well-being. Dr. Young has illustrated truly that in biology, timing is everything. For his contributions to medicine and physiology in revealing the vital mechanisms of keeping cellular time, Gustavus Adolphus College is both honored and privileged to bestow an honorary degree upon Dr. Michael Young. Upon recommendation of the faculty, Dr. Young is presented for the degree of Doctor of Science, honoris causa.
Dr. Young, for your unwavering efforts to advance scientific discovery from circadian rhythms to loneliness, Gustavus Adolphus College is privileged to bestow an honorary degree upon you. And now, by the authority vested in me by the Board of Trustees of Gustavus Adolphus College, and upon the recommendation of the faculty and approval of the Board of Trustees, I hereby confer upon you, Dr. Michael Young, the degree of Doctor of Humane Letters, honoris causa, with all of the rights, privileges, and honors pertaining thereto. Dr. Young, we now welcome you to share your remarks.
Thank you. So this has been quite a conference. I don't think I've been at a conference that has shown such a diversity of expertise in quite a long time. So it's been truly enlightening to be a part of this. The title of my talk, as you heard, is, if I can get this to advance. Oh, now I have to go back. There's a delay. Okay. So as you heard, the title of my talk is What Happens to a Lonely Fly? And as you might suppose, the experiments I'm going to tell you about were initiated at a time when many of us were lonely. We were undergoing the social isolation associated with COVID. And the many effects of social isolation that were produced during that time raised questions about whether, once again, we might use Drosophila, which has helped us understand behavior so many times and other features of biology so many times in the past, might provide some information once again. So this is a data that was collected by University College London, going from March of 2020 to March of 2021. And the data shows responses from 70,000 United Kingdom respondents with regard to their assessments, their personal assessments of their sleep quality. And what you see at the beginning, there's both male and female data presented here. And what you see in the beginning of the study, sleep quality is pretty good, but monotonically declines over the course of a year. And the vertical lines that you see here are just indications of when lockdowns occurred in England and or throughout the UK. And of course, those lockdowns were very much pointed at trying to achieve a high degree of social isolation. Now, it's very difficult to know whether these sleep effects, these effects on perceived quality of sleep, were derived in particular or in part from social isolation being implemented at the time. And it would be very difficult to ask that kind of a question in a human population. But we thought maybe we could get some ideas about this from looking once again at the simple insect that's taught us so much in the past. So on the right, you see a cartoon, essentially, that shows a device that we use to follow sleep-wake activity in the fly. It's a computer monitor that's hooked up to a plate that carries several vials, each with a single fly in it. And those flies can move freely back and forth in that vial. There's food at one end, and the other end is stoppered, so there's a gas exchange, air exchange. So the fly can survive in there for a couple of weeks at a time. And across this set of fly-filled tubes is cast an infrared light beam. And much like a burglar alarm system in a home, when the fly passes that beam, a signal is sent to the computer that's recorded an event, a moving event, so you know the fly is in motion. So the way we think of sleep, ever since work done in 2000 by colleagues Hendricks and Shaw, is to observe the flies and notice that when a fly's been quiet for 5 minutes or longer, the fly often tends to display characteristics of sleep or rest, a different posture, and often other behaviors that can be associated in insects with sleep. So what we're doing on the left side of this chart is looking at 30 individual records of flies in this kind of monitoring. The blue bars on each line going from one side to the other is a single fly, with the blue bars representing times that they're asleep. And one of the things you'll notice is, this is during a light-dark cycle, flies sleep a lot. In fact, in these assays, they get up in the morning for a few hours. They take a noon siesta that's quite long, and get up a little bit in the evening, and then sleep through most of the night. So we've taken the data from all 30 of those flies and plotted it in a different fashion beneath, and you see once again that kind of a curve that indicates a midday siesta followed by fairly consolidated sleep at night. So does social isolation reduce sleep in Drosophila? Does it affect sleep at all, for that matter? So the method that we used to ask this question was to allow newly emerged flies, after they've developed in their pupae, after they've emerged for 3 or 4 days, we put them together, males and females, in a bottle so that they can have some social experience for 3 to 5 days. And then we take the individual flies and we move them into vials that contain different numbers of flies. And what you see represented there is a vial of 1 fly, a vial of 2 flies, 5 flies, 25 flies, or 100 flies. And then we hold them in that situation for 7 days, and then we move them one at a time into these individual smaller tubes where we can monitor their sleep-wake behavior. And what you see at the bottom of the slide are a series of curves that represent the sleep-wake patterns of all of those experiments. And you'll notice that there's one deviant from the group. Most of the flies show a very similar pattern of sleep and wakefulness, with the exception of the isolated fly. The single fly is showing less sleep during the day and about the same amount of sleep at night, but less sleep during the day. And an impressive feature of this that surprised us at first is even another fly, 2 flies in a vial, will keep this from happening, which is maybe one of the reasons so many pets were bought in New York during the pandemic. Social isolation, is it duration dependent? Does a fly know how long it's been isolated? We knew very little about how a fly perceives time over multiple days. So again, we allowed the flies to gain social experience together, and then compared what happens to a group of 25 flies versus a fly that's isolated all by itself for 1, 3, 5, or 7 days, and again monitored in the same way that I described before. And what you see here moving left to right is that the longer the isolation, the more profound the level of impact on daytime sleep. So 1 day of deprivation, 3 days of deprivation, 5 days of deprivation, you begin to see an effect, but at 7 days, you see a maximized effect, which is held after that time. So somehow the fly is able to determine perhaps that things are hopeless after a week, that they're never going to get out of this situation. Now, this is something that we've seen in many different types of Drosophila. We've seen it in inbred lines that naturally vary with the amount of sleep that they have under normal conditions. We've got short sleepers and long sleepers, maybe like some of you in the audience that brag about sleeping only 4 hours a night, others that want your 8 or 10 hours a night. There are fly lines that vary in that way. But this kind of an effect occurs in all of those lines. It's a uniform response. It occurs in young and aged flies. It occurs in many other isogenic strains and in many Drosophila species, every species we've tested. Now, this shows that the maximum effect that we see after 7 days is focused on a time early in the morning, so that most of the sleep that's being lost is lost from the early part of that midday siesta. So at the bottom of the slide, you see that about 40% of the total sleep expected at that time of day is lost after 7 days in this kind of an experiment. And there's a progression so that total sleep is lost, but much less severely. Daytime sleep is lost to a higher degree, but that first 4 hours of daytime sleep is maximally affected. We've tried to ask whether there are exposures that we can produce to these isolated flies that will relieve whatever this stress of social isolation might be. So for example, in the middle of this graph, we show 50, plus 50%, minus 50%, and 0%, where 0% would be the level of sleep loss that is associated with being in a group, which of course, as I've shown you, that's your control value, that's your baseline. And what we're measuring here is we're focusing on that first 4 hours of daytime sleep, the loss of that, the early part of the midday siesta, to get the most significant insights that we can. So what we've seen is a strong depression. Again, about 40% of that sleep is lost when a fly is by itself for 7 days. On the other hand, and we've tried things like leaving a group of 25 flies in a vial number 3, for 2 or 3 days, and then removing those and replacing them with a single fly. Does that make a difference? It doesn't seem to make a difference. So there aren't olfactory cues that are left behind on doing that kind of an experiment. We've tried placing a glass slide down the middle of the vial and having 20 flies on one side and 1 fly on the other side of the glass slide to see if visual stimulation will produce a different effect. But that doesn't work. The flies still do sleep. And we've tried adding netting to separate the single fly and the group of flies, netting of different sizes, some that will only permit chemical cues to get across, and others that would allow the fly to reach through and perhaps touch flies on the other side of the screen without actually being able to move to the other side of the screen. And what we see in these cases is a further depression in the amount of sleep, almost as if being excluded from that group when you know they're there and you can interact closely with them, but you can't join them, produces an additional stress with regard to the measurement of sleep. We found that if we add a single fly of the species Drosophila simulans, which is a related species to melanogaster, that will be treated as a melanogaster cluster of flies in the same vial, and so that will relieve the stress of being alone. And in 2 cases, we've tried ladybugs to see if just some other living object or living being in the tube makes a difference. And in one case, these were from 2 different vendors. In one case, we got a response that was very much like adding back flies. So it may be, again, I refer to pets as something that many of us ran for during the pandemic if we were alone. Another kind of insect, milkweed bugs, did nothing for this, and we really produced a strong negative response by adding a series of foam balls in the. So unlike the bees that we saw the other day playing with rolling balls around, this didn't produce that kind of interest at all. So how do we go from there? We really did want to understand what was going on in the fly's head and the brain that might be responsible for this response. Is the fly hardwired in some way to have a response to social isolation that involved a depression of sleep? So what we decided to do was to take a sample of RNA, total head RNA, from flies in that critical window when they were losing sleep and just before it, including the time just before it, to see if there were gene expression changes that were associated with a fly that had been chronically isolated versus a fly that had been isolated for only a day, where we don't see a sleep defect, and compared to flies that have never been isolated, that have been reared in a group. And so you see from top to bottom the plots of behavior of those 3 different groups. And so what we were interested in, are there changes that separate the chronically isolated flies, that is flies separated for 7 days, from both those that have been isolated for only 1 day and those that have not been isolated, since neither of those latter 2 groups are showing a sleep deficit. And what you see from the intersection of these Venn diagrams is that we recognize 274 genes that were uniquely being altered in those comparisons where the comparisons were flies that showed the sleep deprivation versus those that don't. On this slide shows that there are only 2 categories of gene expression changes that would be of interest in this kind of experiment. We need to either see genes that are going down in response to social isolation, chronic social isolation, 7-day isolation, or going up. We're, of course, not interested in things where all 3 categories differ. So on this slide, category 2 and category 4 are the genes that we're most interested in. And you'll see rather few genes shift down in their expression, but many genes are shifted up in performing and producing chronic social isolation. On the right is shown a graph of the kinds of genes that are affected. And the majority of these genes are genes that affect metabolic pathways. And you can read the classes of metabolic pathways that are affected at the top. At the top, for example, oxidation reduction processes. So a great number of genes that affect metabolic pathways. The open bars represent the probabilities that those would occur by chance, with the longer bar being the lower probability. And the black bars are just indicating the numbers of gene expression changes that fall in that category. Now, if we focus on the right side of this graph, of this chart, we see the top 20 changes in gene expression. And once again, these are almost all metabolic changes in the fly's head. If you'll note the arrows that are on the far right side of this chart, those are all arrows that show the positions of genes whose expression was most altered in flies that had been starved for 24 hours, Drosophila in another study, an earlier study. So this profile of gene expression changes strongly resembles flies that have been starved. And of course, these flies have had full access to food. The only thing they haven't had access to have been other flies. So this was an interesting puzzle to see. So what we thought we should ask is whether or not we've been looking at sleep, but maybe we should look at something else. Maybe we should look at feeding and appetite. And to do this, we had to modify the behavior assay that we were using somewhat. We used a video assay and a feeding paradigm in which we put liquid food in a capillary tube and provided that through a hole in a stopper at the top of a tube carrying a single fly. And we can look at the response of a single fly that's been group reared or reared for 7 days in isolation using a video camera to look simultaneously at the change in the amount of food in the capillary versus over 24 hours versus the movements of the fly. So we can plot both of these simultaneously. We can mark the meniscus of the food with a dye so that the video recording will very accurately tell us how much food is being consumed by the flies. And what you see, if you look at the bottom, you see that there's a substantial difference in purple of the amount of food being consumed by the fly that's been socially isolated. So 7 days of social isolation produces flies that consume about twice the food that a fly that has been reared in a group consumes. And again, you've got a fly that's been in a group for 7 days, a fly that's been alone for 7 days. You put them in this monitor for a single day, and you see the after effects of that social isolation. Now, if we just look at the bottom of this slide in purple and green, we see these changes in food consumption with different measures, different lengths of daytime. So total food consumption is on the bottom left. Food consumption during the daytime is shown in the second panel. The third panel is nighttime consumption, and the last panel is that food consumption during that first 4 hours of the morning. And what you see is in every case, at least twice the amount of food is being consumed by the lonely fly, by the fly that's been in isolation. The top just reinforces our earlier conclusion that sleep is also being lost. But I think the change in feeding is so striking that I wanted to concentrate on that. So this reminded us of another study, again in humans, which took place during the same period of the pandemic. This involved a little over 3,000 adults in the U.S., well, mostly adults in the U.S. And as you see, many different groups are being surveyed here. 2/3 of the adults in this study either had a significant weight gain or a significant weight loss, with most of the changes on the weight gain side. And some of the gains can be quite substantial. For example, in millennials, maybe that's surprising. We get an average of 41 pounds of weight gain over that year of time. And that's for ages 25 to mid-40s. And you can read the rest out, but it's a fairly consistent pattern where there was a very strong tendency for weight gain in this human population of about 3,000 individuals. So this fly is doing two things that we've seen in previous studies. This study was from the American Psychological Association. The initial study I showed you on sleep was from University College of London, but this is, in both cases, we see this striking parallel of our own behavior and the behavior of these much simpler, as we view them, flies, Drosophila. Now, when we look through that list of gene products or gene activities that are most changed, there are a few things that stand out, especially since we've now seen that food consumption is so strongly affected. Limistatin, which you see in the center of this slide, is an appetite-stimulating hormone. And what it's doing in these socially isolated flies is it's lifting in its abundance very significantly. On the other hand, drosulfikinin is a satiety signal. It's a signal that's produced when the fly has had all it wants to eat and doesn't want to eat anymore. And what you see is that that's depressed in our socially isolated flies. So we're seeing something very consonant with the observation that chronic isolation is producing a change in the amount of food consumed. So we thought this might be a pathway into understanding where in the brain these responses, what part of the brain might be responsible for these responses to social isolation. So this slide shows on the left the pattern that we get in the central brain. On the right side of this slide is shown a cartoon of a cross-section of a Drosophila brain. And in the center of that image, you'll see something called the FSB, for the fan-shaped body. This is a densely populated, it's full of neural projections, and that region of the brain is known to be involved in a number of very complex behaviors, ranging from sleep to orientation to olfaction. And one of the great benefits of this region of the brain is that Genelia, part of the Hughes Institute, has published a complete connectome of this region of the brain, so that every neuron has been mapped and every synaptic connection for those neurons have been mapped in a vast connectome that was produced by looking at serial electron micrographs from hundreds of investigators who pooled their energy to make this extraordinary resource. So on the left side is shown a pattern of immunostating that comes from using an antibody to lymphostatin. Remember, lymphostatin is turned way up in these socially isolated flies, and if we look at which cells in the brain are producing lymphostatin, we find it limited to this group of neurons. On the top are shown the positions of the projections of those neurons. On the bottom, in purple, are shown where the cell bodies, they're at some distance away from the fan-shaped body. And so they project into the fan-shaped body where they make these many synaptic connections that I was talking about. Now, the trick that we wanted to take advantage of is we've got many, many tools in Drosophila that have been created to allow us to specifically produce activity of a gene of our choosing in a specific cell type, in a specific neuronal type in this case. And so we identified one of these tools, which is labeled P2Gal4 in this slide, which specifically produces a gene activator, which is in these cells. And in this case, what you see is that tool P2Gal4. Gal4 is the activator. P2 is the cellular response element that gives it specificity in just this group of cells. And this is driving the expression of a fluorescent protein, green fluorescent protein. So now a specific group of cells will respond to that tool by creating a green image. And what you'll notice is that that green image overlaps with our lymphostatin immunostaining. And if you superimpose the two images, the immunostaining image and the fluorescent image, you see that you get white overlap essentially everywhere that you saw the lymphostatin cell bodies and their projections. So this is a very useful tool then in probing the function of this specific group of cells that is responding by producing lymphostatin at high levels in response to social isolation. So what can we do with a tool like that? Well, what we can do is we can, instead of expressing a fluorescent protein, we can express a channel, a protein channel that allows ions to flow into these neurons. So we've chosen to use the P2 tool to drive, instead of the fluorescent protein, a protein called KIR2, which is a potassium channel, a specialized potassium channel. Overexpression of that channel in these cells will produce hyperpolarization of those cells so that they can no longer fire. So essentially what we're asking is, what happens if we now have identified the cells that we're interested in? What if we silence those cells with this special channel? And what you see on the top, what you see on the top right is that this normalizes the sleep pattern. The sleep pattern now, we don't have that deficiency in daytime sleep that we saw before. On the other hand, if we have just the tool or just the channel, but not in the same fly, we get the responses that are seen on the lower two panels, where once again we see a return of the loss of sleep. So putting these two things together abolishes the sleep problem that we have by maintaining a fly for 7 days to give them chronic isolation. And again, at the top is just shown the overlap of the lymphostatin expressing cells and the cells that are being driven with this tool. This is the result that we see in that experiment when we look at appetite rather than sleep. And what you see on the left are the two elements of this construct, which by themselves should not relieve anything about chronic isolation. But on the right, we've combined them, and we see now an activation through hyperpolarization of those cells, and you now see the purple trace of appetite collapse on the appetite of group-reared flies. So we can eliminate both components of this response to social isolation by turning off this specific dedicated group of cells in the central brain. Now, this is a little more complicated experiment, but it's an important one, and I'm just going to focus on the right side and the rightmost set of experiments, which are indicated in red. What we have here is the same kind of experiment where we're driving in the same neurons with the same tool. We're now driving, instead of something that will silence those neurons, we're expressing a channel which will activate those neurons when we raise the animal at slightly higher temperatures. We're using a channel called TRPA, which was discovered because it's the channel in your mouth that recognizes chili pepper as hot, but it's the same channel that recognizes hot temperature. And the reason that chili pepper tastes hot is because it's the same channel. So we're using that channel to say what happens if we activate these particular neurons in the fly brain with temperature. So we have, on the right side, you'll notice in the right bottom, you will see flies that are marked as 22, 22, 28, and 28. And the 22 refers to the temperature being 22 degrees, which does not activate this channel, and 28 represents the temperature-sensitive response of that channel, so that we get activation of that channel. We get activation through 28-degree temperature. And what you notice is that the purple response goes up fairly dramatically over on the right side relative to both the 28 without that channel, and the 22 with or without the channel. And an important point to make here is that we don't see a response if we just activate those cells in group-reared flies. The fly has to have some social isolation, but only a single day. So now the fly is interpreting a single day of social isolation as if it's been chronically isolated for 7 days. So somehow, not only do these neurons control a response to social isolation, but there's some measurement function involved so that the fly is able to use these particular cells, fire these particular cells in a fashion that reflects how long that social isolation has been. And now what we've done is we've fooled these neurons, and therefore fooled the fly into thinking it's been isolated for a full week. So why would there be a convergence of sleep and appetite in response to social isolation? Again, we've seen it in humans as well, and it doesn't have to be the same mechanism, but in the fly we can explore this pretty directly. And we know from other studies that there are convergent projections to the same area of the brain coming from well-studied sleep-promoting neurons in the dorsal brain. And those are shown here. There is a tool that will mark those particular neurons called R23E10. So when we express that tool along with that green fluorescent protein product that I was speaking of previously, and we look using anti-lemmastatin at the antibody to lemmastatin versus this new sleep-promoting tag, which is going to be tagged with fluorescent green, we see a strong convergence once again, but this time the convergence isn't an overlap. It's a direct juxtaposition of the, on the right, of the projections of the sleep-promoting neurons and the projections of the lemmastatin-expressing neurons. So it's possible that the sleep response or the appetite response is due to the fact that you can't both sleep and eat at the same time, and therefore you have, you're measuring a consequence of of having two behaviors that are mutually exclusive. But it's also possible that we have a wired system that deliberately generates a response in both sleep and appetite in response to social isolation. And some evidence in that favor is that there are abundant synaptic connections between these two groups of cells, of neurons, the projections from the sleep-promoting neurons and the projections from the lemmastatin neurons. So let me just finish up by giving you some conclusions. First, we never really thought of Drosophila as being highly social, but we're impressed that a fly does care whether other flies are around. And in fact, we have a good deal of evidence now that they can distinguish long periods of isolation from short periods of isolation, acute from chronic social isolation. As I've shown you, chronic social isolation activates central brain neurons that regulate appetite. The increased feeding, the starvation brain state induced by chronic social isolation appears to broadly reset expression in metabolic proteins throughout the brain. So we believe that now is beginning to make sense. Since we've stimulated appetite, we've stimulated consumption, we might expect to see metabolic a ramp up in genes handling higher metabolic rates. As I said a minute ago, the drive to feed may indirectly inhibit sleep, but again, these connections, these tight synaptic connections, abundant synaptic connections between these two groups of neurons make us entertain another possibility, which is this is really a deliberate wiring of these two sets of behaviors together in response to social isolation. And then finally, the hyperactivity of the P2 neurons, which alters the duration of social isolation that's needed to get to reduce sleep and to elevate appetite. Our ability to manipulate that by stimulating these neurons, again, suggests that those neurons also have some capacity for determining how long the fly has been isolated, to distinguish chronic social isolation from a brief period of social isolation, since we can convert one to the other by prematurely activating these neurons. So this is a bit of a ways from our original interest in circadian rhythms, but I think it was a fascinating journey that was stimulated by a huge health problem in human populations, which we thought might be much more difficult to study in human populations, and maybe we could get some insights from Drosophila. And so I think once again, Drosophila has proved itself to be a useful model for asking questions. We're gaining insights that would be difficult to have in any other model system. So thanks very much, and that's it.
Hello there. That was an amazing final lecture. I gotta say, I, for one, was really interested to come from having this whole conference being about the differences between us and the insect world and the ways we conflict, and come around to this thing that was like, here's how we're similar.
Absolutely. Absolutely. I think it really demonstrates that we are organic beings, that we are related to the insect world, and that there are things that we can learn about ourselves from the ways that insects operate.
Right? Yeah, and I mean, I think one of the other things that stood out to me is like we've kind of also, like you and I have talked about the soft science, hard science conflict that kind of comes up around conferences like this where you're trying to talk to a big swath of the public. And I thought that this was really interesting also as kind of a topic of research that bridged those two things, because you have this idea of like the hard science of genetics and like the rational, data-driven metabolism changes, but then what it's all coming back to is loneliness.
Right.
Which is such an emotional thing that's often not really given, I guess, like a full authority as real.
Yes.
Right?
Yes, yes. And a real emotion that I think we all can think back to the pandemic. I mean, as he was talking, I was sort of like reliving my pandemic, experience, right?
Oh, no,
Yeah, where he's even like, okay, we would put a glass between a fly and a group of flies, and they seem to get worse. And I was just thinking about Zoom. Right?
It makes
We're
us worse.
sitting in our homes isolated. We're watching other people on a screen, and somehow, except in this case.
In this case, it makes everything better.
Fantastic. And it just kind of made it worse in some ways. There needed to be some like physical interaction, some community to alleviate that isolation in flies.
Well, I think we all innately understood the eating because you felt sad.
Right.
The comfort eating. We all understood that. We all understood that like, oh, I'm sleeping really badly. And to see that there is something to that beyond just like your own moral failings.
Absolutely.
Right? Is so interesting.
Yes. And I think he really demonstrated so many levels of data for us from like, here's the genes. What happens with the genes? Okay, which cell types are those genes expressed in? Okay, and then how are those cell types interacting with one another? And what's so cool about using a model organism like Drosophila that we know essentially almost everything about, the genome is, we know everything about its genome. Now we can take those genes and say, okay, out of these genes that we know Drosophila have and that are being activated, what are the ones that are similar in humans? It gives us sort of a target for humans. Where suddenly we have 20,000 genes to deal with.
Right, and also, I think it was also really nice to find that like, oh no, just having like that one access to that one other fly. Or having
a pet
your pet ladybug. whatever your equivalent of a pet ladybug may be.
Right. We all got pets, and it seemed to help.
It did. Weird it did, but it did.
Yes. But what an impactful final talk.
That was a really lovely way to wrap up everything today. We have still a panel discussion that is coming up. So don't shut off things quite yet. We have a little bit more to come. I'm sure there's going to be a really cool conversation around this, around the previous discussion. There's a lot of stuff ways that these things can kind of interact, because we're talking about like how we treat bugs.
Right.
How does that affect the ethics of how we treat bugs when we are isolating them and torturing them for fun?
Exactly.
Yeah.
Or to understand that our outcomes are entwined.
Yes. Absolutely. how our outlet, how those things are intertwined, how the ways that we study them are affected by what happens to us in the real world, in our world.
Yes.
Like all of those things are connected, and I think that the Q&A session that's coming up very shortly will probably end up getting a lot into that.
Yeah, absolutely. I think it's going to be exciting to see the direction that they take the last Q&A session, because I think there's a lot of possibility in where it might go.
Yes. And I think there was also a little bit of a hint at what's coming next year.
Yes.
In this session as well.
Absolutely. So they were talking about sleep deprivation, and next year is about sleep.
Next year's going to be about sleep, but we will get back into more of that later on after this Q&A is done.
The spirit is willing, the flesh is a little bit weaker at this time of the day, right? We know what we want to be talking about, so let's dive right in. Margaret.
Yes. So I'd like to ask if any of our speakers in particular have questions either for Jessica or Michael.
Yes, I have one for each of you. Let's see if this works. Yes. If I can start asking you, Michael, this, I mean, seen from an ecological point of view, you would think that if you were isolated, you would go looking for someone to hang out with. So did you see or did you measure whether these isolated flies moved around more? I mean, they slept less, but did that actually mean that you could sort of interpret it ecologically as if they were trying to find others?
Right. So we have looked at whether they showed more motion, more movement during the waking time, and they don't. But then they may know it's futile to look for others because they're in a tube by themselves. But it's, yeah, I think it must be a pretty strong signal that something's very wrong if none of your compatriots are around. I mean, it strikes me as something that would have pretty deep evolutionary roots.
Yes.
Why would this happen? Why is this a benefit? And you know, if everybody's gone, maybe that's a pretty good signal that something catastrophic is at hand. So what do you do to protect yourself if you're a fly? You know, you stay alert and you consume as much food as you can handle. But this is a good, it might be interesting to put them in an arena and see what kinds of interactions emerge on being reunited. You know, we know that if we reunite them, that that sleep comes back to normal and appetite comes back to normal, but we haven't looked at the behavioral aspects of, you know, did they show some level of extra effort to get back to their colonies?
Yeah, it would be interesting. If the cage was bigger, maybe it would be possible to study something like that. I don't know. May I also ask a question for Jessica? I was super interesting with this focus on taxonomy and collections. I think that is a very undercommunicated issue, the fact that we need these collections for so much of our science. And I just wanted to ask you, do you have any ideas of how we can work to get more students to study taxonomy and become taxonomists?
Yeah, that is a good question. We actually talked about it a little bit during the lunchtime discussion about what was considered to be a womanly pursuit was often to go out and collect feathers and shells and create cabinets of curiosity. And there was a history in entomology of this being something that could be an excellent pastime, you know, to do taxonomy and we like to sort things, we like to categorize things. I think somebody showed yesterday their child kind of identifying differences amongst animals. We love doing that, and somehow we get away from the idea that taxonomy is cool by the time we reach high school. So I think it has to start from a very early age, perhaps, underscoring that systematics and taxonomy is important. It's not dusty. It's not something from the past. You know, we're still actively discovering species. I mean, I feel kind of like Indiana Jones, you know, you're kind of like going out and exploring and doing, and really discovering new species can be exciting. We just don't communicate that. We imagine taxonomists kind of as the two characters in Silence of the Lambs who are just kind of, you know, deprived from sun, just sitting in a dark room, you know, looking at fly maggots. That's not what systematics is. You know, systematics is a vibrant science. It includes, you know, biochemistry and genomics and genetics and morphology and histology. It's more than just staring at an insect in a vial or on a pin. So we need to kind of glamorize the, you know, taxonomy, I think, from a very early age.
Thank you. Yes, Jonathan.
Yeah, I think this is a question for Michael primarily. A big theme in your work clearly is finding surprising commonalities between fruit flies and humans, including these extraordinary data you were just presenting about social isolation leading to loss of sleep in both cases. I'm curious about how this has changed the way you see the flies as individual creatures and whether it's changed the way you treat them. Whether, for example, it might have, it might lead you in the future to think, let's be careful about keeping these flies in socially isolated conditions.
Yeah. Well, this is the first thing my wife brought up when I showed her the data. These little flies have some kind of an emotional response, and of course, that's one way to look at it. And it's when I was in college, I can remember that you were always there was a lot of pushback if you ever tried anthropomorphic kinds of thoughts were always pushed back. But the more homologies you see, the more you think, why would you rebuild the system from scratch? And so if this is the way you can get a certain behavior, then why wouldn't it apply to another organism? So are they depressed, in anything like the way we would think about it? And I think it's a real possibility that there's something like that going on. One of the things that I'd really like to understand, and we're still struggling with ways to pursue it, is when we turn those neurons off and make the responses go back to baseline, have we relieved the stress of social isolation? Or are we just, is the animal still in distress, but we've cut off its ability to express that? And I think I lean to the side of the former, where the easiest explanation for losing two different responses, I think, is to say you've relieved the stress that was associated with the problem. It also brings up questions about humane treatment of prisoners. Isolation may be a form of treatment that has many more connotations than we originally. You throw somebody into isolation, and in the penal system, for example, is this an added form of punishment that was not appreciated before? And
I'm sure we'd all agree that in the case of humans, social isolation raises serious ethical concerns.
Yeah.
Do you see this as having practical implications for how you treat the flies?
Yeah. Yes. Yeah. It rang strong. We didn't, this story wasn't told yet when you were talking about these things, but as I was thinking about our own work, I could understand where the question was coming from. And it goes back to this question, is it one chicken or one fly? What's the difference? Is it the weight of the animal, or is it the response that is elicited by whatever insult you produce? I guess that's maybe why there was concern about, raising millions of insects instead of one chicken, or was that a stronger ethical position? And I think that's a difficult question. Hopefully we'll know more about what's going on in a fly's head in the future.
Can I ask a question related to that? It's a technical question. It's a quick one, I think. Yep. So these were, I assume, like a lab reared strain? Yeah?
Some of them were.
Okay.
Yeah, some of them were isolated from nature.
Michael. Okay.
And, okay. I hope that's better. Some of the strains that we tested were isolated from nature, and particularly those that had naturally longer or shorter intervals of sleep. So as I was saying, like many individuals, human individuals, that will tell you that they have lesser or greater requirements for sleep. And it really didn't matter. And as I was saying, this wasn't just limited to Drosophila melanogaster. We saw it in several Drosophila species. So we haven't gone beyond that, but we probably should.
And I was just thinking, because I'm thinking about their ecological aspects of Drosophila, right? That they often do feed together. I know that they're often on a fruit together, and I'm just wondering if you suddenly end up that you're the only one on the fruit. I'm just wondering if it's loneliness or like maybe, like you said, maybe something's wrong and I should be going somewhere else. So I'm just to push back on your loneliness just a little bit, because this is what we're supposed to do here.
No, I agree.
I mean, maybe it is that the reason that the metabolism and everything is upregulated because the fly is going, all right, there's something wrong with this food because I'm the only one here. Maybe I need to fly somewhere else, but of course they can't because they're in your tube. I'm just curious.
No, I agree.
How
I
you'd
agree.
respond.
It's almost as if you need a taster.
Yeah, like if you're the only one in the restaurant, you look in the restaurant. You always look in a restaurant. And if there's nobody in there, you think maybe I shouldn't be eating here. Yeah, I'm just wondering if it's a similar. So it's not loneliness, but kind of like maybe this is really there's something wrong with this food.
Yeah.
So.
Only these guys are eating.
But then I mean.
But I take your point, because who knows what the response to that kind of stress would be. But you're absolutely right.
Second, you had a question? So my question is to Jessica. So I was really, thanks for the excellent presentation, but I was really struck by the colors, a variety of colors of dragonflies. And I was thinking, why would they have evolutionarily that type of color, knowing that maybe they will be the next meal to a variety of these other organisms. So why do they want to attract attention with the colors, or does the colors have any impact on that? Why do they have that color? Yeah, those colors. Yeah.
That is a good question, and color is a really big part of the story of dragonflies. And there's two main types of color: structural color, where there's kind of bumps and ridges on the cuticle. When light bounces off of it, eyes can perceive it as being kind of a metallic or shiny color. And then there's the coloration that is granules of pigment inside their epithelial cells that can migrate up and down, and that actually dragonflies are able to kind of change their color. So they can be what's called bright phase, where they're a very bright color, and they can be what's called a dark phase, or a very dark color. And we don't exactly know the purpose of all of their color, but we know that their eyes are capable of seeing a wide range of colors. So we think communication is part of it. Some of the color patterns on their wings, we see behaviors that dragonflies do where they kind of flash their wing patterns, and we think that there's sexual, communication that's taking place. But we also think thermoregulation is part of it. So dragonflies that are quite cold tend to be in dark phase. And early in the morning, when you first find dragonflies, they tend to be in dark phase. And we think that perhaps they're absorbing as much sunlight as they can and heat, because they need to have a certain amount of energy for their flight muscles to kind of whir and for them to be able to take off in flight. And indeed, frogs jump out of the water to catch them, fish jump out of the water to catch them. And there are some dragonflies and damselflies, they're all doing this kind of mating dance at the water. Very vulnerable time for them. And there are some like RJ apicalis, which is a damselfly, that as soon as they complete mating, they actually go from bright phase to dark phase. And some, like Amanda Wispel did her PhD on this, she argued that this was so that you could avoid being seen by predators. So it's a very complex story, the story of color in dragonflies, but we think a lot of it is this combination of thermoregulation and communication.
Perfect. Thank you.
I have another question from the audience for you, Jessica. The research you and your colleagues have done on dragonflies is fascinating, and it's complex. And the question is, are there similar types of exploration of the systematics of other insects, perhaps some that aren't quite as charismatic as we might consider odonates to be?
For sure. I mean, there are systematists to study all different types of insects, but there may not be as many of them. So there are groups of entomologists who spent their whole life studying lice. They collect lice, they dissect lice, they do genomics of lice, but they don't have big international lice meetings the way that we have big international dragonfly meetings.
Well,
take
they
place.
do.
I mean, every meeting could be a lice meeting if you have lice. So there are entomologists who study different groups, but I would say that the one common lament is that if you look at across all of entomologists, and you look at what they study, a majority of them study social insects, so bees, ants, and wasps. Very few, relatively speaking, entomologists study the evolution of termites, even though they are social. A lot of people study butterflies. Butterflies are large, charismatic animals. And if we look at those two groups, social, that relates to humans, and beautiful, butterflies, and we like beautiful things. So things like earwigs, things like various, like for arachnology, various ear mites, head lice, pubic lice. These are things that people spend far less time pondering, but they have an evolutionary trajectory that's really interesting and fascinating too. So I would say there's an entomologist for every kind of insect, but the number of entomologists for some insect groups is really high, and the relative number of entomologists for other insect groups is really low.
So coming back to your comment about reaching out to students, encouraging them to go into systematics, you need to be pursuing lice and fleas and earwigs. Is that what I'm hearing? Okay.
So when I started,
That's going to get them in.
everybody loves a lice biologist. I will say, I had a graduate student years ago who's now a professor at the University of Illinois at Urbana-Champaign. His name's Dominic Evangelista, and he said he really wanted to study, he was interested in cockroaches, and he really wanted to study cockroaches. I said, there is almost no one studying the social behavior of the subsocial roaches. There's very few people that are studying the evolutionary behavior, the evolutionary trajectory of cockroaches. If you go and study cockroaches, you could be like the cockroach person. And he really liked that idea, of becoming the cockroach person. And now he kind of is the cockroach person, in the States at least. And so if you like the idea of being an expert on a group, there's a lot of groups for which we don't really have kind of living experts, because unfortunately we lose taxonomists and we lose expertise. So there's probably a beetle group that hasn't been studied since the 1920s that you could be the expert on. You just need to dip your toe into entomology.
I think earwigs could do a service to themselves if they would rename themselves, because that's not that doesn't invite participation.
Great. Another question that has been, this has been kind of a theme, I think, through all the talks, is, so this is for you two in particular, but really anybody on the panel. Why do the diverse experiences and perspectives of individuals who study insects influence the nature of what we learn? And why does this matter?
That is a good question. That's a big question. I guess I could, do you want to dive in?
Well, I was going to say, I think, we've seen how different the perspectives are, the different focus that's represented here, and I find it pretty refreshing. And something that we're, many of us molecular biologists, go to molecular biology meetings and, talk molecular biology. This is much more exciting. So I think it's important to hear these different ways of looking at biology, and the sense I get is maybe some of these tracks are something that can be added on and mixed with what we're doing. So it's very valuable.
I think in the area of systematics, one thing that we see for insects, certainly for dragonflies, is that traditionally when people talked about selection, they talked about sexual selection, almost everything, and pardon me for talking about genitals again, but almost everything focused on male genitals and male control of reproduction. So if we look at the kind of literature for insects, for dragonflies in particular, dragonflies are unique because males have two penises. They have a penis at the tip of their abdomen, a secondary penis at the base of at the beginning of their abdomen, through which sperm is transferred. And people thought that was fascinating, and we kind of have focused a lot on male genitalia. But it turns out that female genitalia is actually fascinating too. Females have there's a lot of interesting things. Females are able to store sperm. In some cases, for dragonflies, they can store sperm for quite a long time. But in cases of things like termites, they can actually store them for decades. So understanding how female reproductive system works has been largely overlooked, and there's many people only just now that are trying to kind of understand this other half of the piece of the story. And I think it's because we're having a diversity of voices. We're having kind of more gender parity in the scientists that are actually studying this, thinking maybe it's not all about the penis. Maybe there's something interesting that's happening in female reproduction as well.
Every diverse background that comes into the field brings a new perspective, and with new perspectives we ask new questions, and with new questions we learn more. And so to to go back to braiding sweetgrass, Robin Wall-Kimmerer mentions how she wanted to study and how she she went up to an advisor and he said, what do you want to study in graduate school? And what she had to learn was sort of how to speak academic, but what she said she wanted to study was why goldenrod and whatever the purple flowers that go with it, so why do yellow flowers and purple flowers look so good together? And she was laughed at, and she was told that, you know, that's not a scientific question. But we saw it was beautifully in your image, and insects can see those colors better. And so it was her perspective of understanding the patterns in nature from her perspective, and she knew that purple and yellow happened together a lot. And so she needed help in the academic, like how do you form this into a testable question? But it was a genuine observation that deserved further, you know, looking into.
Anyone who has not read Robin Wall-Kimmerer is welcome to leave right now and rush out and start reading it, because it is a spectacular book. Braiding Sweetgrass, or really any of her works, but that one in particular. Anyone else have thoughts about Margaret's question?
I think they answered it well.
Okay.
Go ahead.
You know, we have to go back to philosophy a little bit here. And you can correct my pronunciation here, Lisa. The philosopher Wittgenstein once said, if a lion could speak, we wouldn't be able to understand it. He would certainly have said the same thing is true for flies. By that, he meant that our worldview is too distant from other animals for us to understand them. Do you think he was wrong? Can we really learn to think like flies? Can we really recognize the joy of bees?
Yes, I'm sorry. my advisor told me that you can. And I mean, yeah, we're completely different. I agree. We can't see UV and polar rise light. I'm sorry, I had to just say this, but we live on the same planet. It's not like they're living on some other universe that we have. I agree, you know, maybe we'll find an alien species and we have no relationship to it because they have such a completely different world, but we live in the same world. We see the same blue and yellow flowers and the same, I mean, there are commonalities. Is everything the same? You know, as I think it was you that said that, you know, they don't have symphony orchestras and things like that, for sure. Insects don't. But we can learn how they do. As I said, there's basic things that they have to do. They have to eat. Michael showed this so nicely in his talk. You know, they do sleep, most of them. There's a lot of their basic needs that are very similar to ours. And the way that they find food by foraging is not so unlike the way we find food. They just do it while flying, like Jessica's beautiful dragonflies do. But I think it's I think it also it's a little bit this otherness that we create is also false, and it's also a little dangerous, because the more we other, the more we also, you know, you had talked about this with humans, but I think it's also doing it with other animals as well, as if they're so different than us that we can't possibly understand them, and then that starts this process of not caring about them. And they are really important. So yes, we can think like a fly.
Thank you.
Mike, Jonathan.
Yes, I think understanding comes in degrees. There may be aspects of the ways of life of insects that we will always wonder about and that we'll always struggle to understand. You think about something as basic as seeing the plane of polarization of the light and seeing UV. We know they do that, but we don't know what it's like from the insect's point of view to experience like that. So in some ways, there'll be aspects of the experience that remain inscrutable, but the same is true of other people. You know, there are aspects of other people's experience that will remain inscrutable to us, but nonetheless, a huge amount of understanding and bridging is possible, and I think the same is true of insects.
Thank you.
I think that it's not that part of me, I think, is that not what we wouldn't understand them. I think it would make me feel inferior that I can't sense the world the way they can. I mean, there's all these things. I can't see UV. I can't sense magnetic fields. And so I would know it, like, I wouldn't, it's not that I wouldn't it's like a, it's the difference of like not understanding another language is like, I don't have that, like you have the skill I don't have. And I think there's a different way of looking at it instead of ignoring them, being like, oh, I don't understand you. I think if we really think about it, we see how limited we are in our sensory perception. And so it's a lot of like, humans are not the exceptional ones on this earth.
A new form of imposter syndrome.
What?
A new form of imposter syndrome.
Yes, right.
You know, years ago when I started work on circadian rhythms, I had more senior colleagues that were behavioral biologists that said, what are you studying a fly for if you're interested in circadian rhythms? And it always struck me as quitting a little too early. And you know, now we know, you know, as you look at the components that are involved in regulating something like that behavior, they're the same components that we have. So how many other things are there that we can build from the bottom up that show, you know, we really do have a fly inside of us. You know, it's part of our heritage, and so why wouldn't there be vestiges of some of this? And you know, I agree completely with the issue of, you know, particular sensory abilities and so forth, but there's so much that we keep running into that is tangibly the same. So the last thing you want to do is assume that to my mind, you know, again, when I was an undergraduate, I was told that you couldn't assume that if a deer was running from a lion that it was afraid. It just runs from lions. And I think that's the wrong way to think about the problem.
Anyone else? So I'll follow up also with another question to you, Dr. Young. Research related to human well-being has generally focused on fruit flies. How could the study of other types of insects enrich our understanding of human well-being? And this question is going to you, but I also suspect that other folks on the panel can provide interesting perspectives on that.
Yeah, that's a tough one, having spent so many years, you know, being grateful to this little fly. You know, part of the problem is that part of the problem of changing focus to another system is that there's so much that gets developed that you can use by others. So, you know, every year, every 10 years, you have completely new tools that allow you to go deeper and deeper, and those are not just immediately transferable to other systems. It'd be wonderful if some of those were, but I think there's still a long way we can go asking these kinds of questions in Drosophila, and you've got a large body of people that have developed expertise in working with them and again, developing tools. And so I tend to run all that out before throwing up your hands and going somewhere else for certain kinds of questions. There are other kinds of questions where it's the wrong organism.
Were you going to say something? And then Jonathan? Yeah.
Well, I mean, if there was another insect group, I would just advocate to the group, right? This, maybe we can all agree on this, that there should be a non-holometabolous insect. So fruit flies are in the holometabula. They have an egg, larva, a pupa with a complete rearrangement in the adult stage. So I think if you wanted to have another model organism, I think we should all agree, we'll write it down now, that it should be a non-holometabolous insect, perhaps something near the base of the insect tree. Perhaps something dragonfly-like would be even the best, I think. Let's go for it.
So like maybe we have a little bit of cockroach in us too, along with that fruit fly.
Exactly.
Thank you.
Yeah, crickets as well, I think, very interesting. It's, I mean, it raises this big picture issue about the role of model organisms in science, and in particular in the life sciences, where if you think of Drosophila, there's just so much stuff you can do with Drosophila that you could not do with any other animal, because it has this whole technology built around it. So you can use extraordinary technologies like optogenetics to study in just extraordinary detail and have incredible control over which neurons are active. And that can't be reproduced in bees or crickets or anything else. So when we're doing studies in Lars Chicker's lab at Queen Mary, it's much more low-tech, and we're not imaging individual neurons. We're looking at whole animal behavior because we're not using Drosophila. I think we need both kinds of research, really. We do need to be making the most of these technologies that exist and that have been developed for specific model organisms. But we need diversity of different species as well. That's what's motivating us to try and study crickets and try and study black soldier fly larvae. Let's have insect cognition labs that are full of different species of insects. And let's try and learn some of that stuff that we still don't know about their observable behavior.
I was just thinking, I mean, with this loneliness, being alone versus being together with others of your species, like locusts, maybe it could be something interesting studying locusts that shift between these gregarious phases and the solitary phases. I mean, see if anything is parallel. And I mean, I guess we do know a bit about what triggers those shifts. And it's super interesting because while they're solitary, they hate each other. And then something happens and they are suddenly very good buddies and they love to be together. So I think that would be one group of organisms that could sort of maybe fit or fill in the picture from what we have learned about, being lonely as a fruit fly. And I was also thinking, I mean, insects can not only help human health through model organisms, but also like these studies of leafcutter ants that have specific antibiotics to make their fungi farming stay healthy. And I mean, by studying these ants, we can hopefully we can look for and hopefully find new forms of antibiotics that would also be super helpful. And that's another way of looking to insects to gain new insight that is good for human health.
Can we move on? A number of people are asking a question about the implications of your research about loneliness. How can we use this information to help understand and prevent loneliness while also preventing a prescription drug culture to develop something that would cure, quote unquote, loneliness? Do you have hopes?
Well, I guess, so I guess early steps might be to look at a mammal, a research, a different research model. Does this happen in mice, for example? Are there specific areas of the brain, or even before that, do you have changes in behavior that parallel what we see in a fly? And that would also make the comparisons that we've made, they're distant comparisons to look at behavior in humans that seem to be in parallel, but I think you could gain a lot more confidence if you were looking at mice and you had molecular tools to say, yes, this behaves in a certain way and certain areas of the brain light up when the animal is isolated. And I think that's the kind of path I would tend to advise.
Do you have, I guess I would ask, do you have worries about possible choices others might make in using your research? All we need to do is invent a technique or a drug that would enable us to incarcerate people more effectively, for instance.
Well, I think before you get to that, you get to an appreciation of what you're doing when you incarcerate people. I would hope that would be the thing that hits you first and that you respond to. But that would be like dreaming up new forms of torture. I don't think that, I don't see anybody advocating for that that I work with.
Yeah, that's a dangerous part of science. I mean, that's the history of my field. I mean, anthropology, we started out as typology of one group versus the other and started making value assessments that, based on your skull size and this skull size, these people usually the white people, were considered to be superior by their measures. And then that was used in the Holocaust as justification. And so like we have to be very careful in sort of how we do our science and present our science. You know, so I don't know what you could do different to prevent this road, but I think we need to, I think that is a legit question and a concern.
I think it's also the choices of the experiments you do. I mean, I don't know if anyone thought about this, but the VR work we were doing was essentially target search behavior and extracting that. And I don't think it's a very long road from, you know, coming up with more efficient algorithms for target searching where you can get to some pretty scary conclusions. So we made very clear decisions in the experiments that we were doing to really tie them to ecology and really be careful that they couldn't at least very easily be misconstrued into something that could be used for, let's say, weaponry or something like this. And I think you really have to think very far ahead. I know nobody, like an apple fly in an orchard, like it doesn't, but honestly, that's how a lot of things happen, that you're actually only concerned about your little world, and you don't even think about how it can be taken to a completely different place. So, but I think we have an obligation as scientists to do that at all times and to really think like, what is the craziest thing that somebody could do with our research? And do your best, as best you can, to be true to the science, but also be very careful that you don't open up doors that maybe you don't want to open from your ethical and moral standpoint. So,
Jonathan, did you want to?
Yes, I mean, I think it could be a really good outcome of this research on loneliness if it feeds into preparation for future pandemics, because we know that we will face situations like COVID-19 in the future. And governments were flying blind at the start of the COVID-19 pandemic, really not in a position to make decisions on the basis of evidence. And so the benefits of lockdowns in terms of keeping people out of intensive care could be quantified and could be seen to be very large. But the costs on the other side, the costs of imposing a lockdown on people's mental health, these were unquantifiable, and so people simply had to guess. And so one really good thing that could come out of this is if we work towards a situation where we have a much deeper understanding of the costs of these kinds of interventions and can weigh up the costs and benefits more accurately.
Yeah, no, I agree. I mean, you know, some of those that suffered the most are the youngest people in the population, those that didn't get to make those early social interactions that I think most of us think are pretty crucial. I guess you can build those up after the fact, but would you want to do that again? I guess, you know, the next time around, and there will be a next time around.
Even something as simple as the effect on weight you were describing will have health consequences decades into the future. Really hard to quantify.
Margaret, I wonder if you'd like to ask a question that will enable us to bring this wonderful discussion to a close.
This one's a short one this time. We're fortunate to have such a diverse range of expertise. I was wondering if any or all of you would speak to something that you have learned from one of the other speakers over the past 2 days.
Good question. You're modeling now for our students.
That was when you asked if we had questions for the speakers. I'm like, no, but I want to like talk about the things I learned in the afternoon talks. And so for Jessica, the collections being moved on the slave ships, I had not heard that. That was new to me. We reckon a lot with, you know, the colonial history and anthropology, so it's nice to see it being discussed in entomology, and so that was new to me, and thank you for enlightening me to that. And then for your talk, Michael, I'm just so fascinated by the idea of ladybug pets for fruit flies. And so I just want to know more about, you know, about that. Like that was just the coolest thing to learn. So.
What constitutes a good pet?
Yeah, exactly. I want to know what species, you know, view each other in that companion kind of way.
That's great.
Yes, for me, I think learning is really nourishing. every day I ask myself, what have I learned new? And if I'm not learning new, I always Google something to read. So this week was just marvelous because I learned from each person amazing, amazing things. So I'm really hoping that I would find the video online so that I can listen to everybody's talk again in the privacy of my home. It is just fabulous and wonderful to meet all of you. So it's really wonderful, and wonderful for the audience also, the excitement and the question, the interaction is just wonderful. Thank you. Thank you to you both.
Yeah, I thought your description of these symbiotic interactions in mosquitoes was something that was completely new and extraordinarily high potential. And you know, there have been so many attempts to do something about malaria that have hit dead ends, and this sounds like an extraordinary new opportunity. And your plantings with plants, with propellant plants versus the crop that you want, and the weed killers, those were all fascinating to hear about. Completely
Thank
new.
you. I will go back and tell the team that they received a compliment from a Nobel laureate. They will be very happy.
Jonathan, did you have?
Yes, I was thinking, I mean, I've learned a huge amount, but one thing we've learned is that there is an audience. You know, we've learned that there is an audience for hard discussions about the science and ethics of insects. And I think that wasn't really obvious at all, you know, at the point of planning this event. You know, it was a brave thing, in a way, to make this the theme. And I think it's been an ingenious theme, because we've seen over and over again how important and urgent and huge these issues are, and how entangled insects are with questions of sustainability, how to live together, how to preserve the natural environment. And that's a great discovery. And I think the more conversations like this we can have, the better. We know people are ready for them, and we need to keep that going and just try and talk to more and more people, get these conversations happening in more and more different places.
I think we're at a very precarious point in humanity. I think we're at a point where there's a lot of potential, and I think there's a lot of possibility for destruction as well that we're already seeing. And I think we have a choice to make in how we're going to go forward. And I think what I learned from each of these speakers is that whether you're taking it from, you know, an ecosystem or ethics or human well-being or food security or biodiversity or living in the global south, that there are real questions that we need to answer and real avenues that need to be pursued. Insects are part of these questions, but there's also a lot of work that needs to be done on a lot of fronts. I think we covered so many things, right? This was not a conversation about insects. It was a conversation about the role of governments and corporations and you know, ethical science ethics and so many things. And I think that it really shows that we need to have more conversations like this. I think this conference is a very good example where bringing together people from very diverse backgrounds is needed at so many levels today. And I hope that the people in the audience continue to have these conversations in their communities and at your school, and seek out people different from you to talk to them about suffering or loneliness or whatever you talk about, right? Because I think this is what is needed today, and this is what is going to drive change.
Yeah, I guess I'm the last one out. No, I think, like you said, Sejanette, I think also I've learned something from each and every speaker, and I've really enjoyed being in a setting like this, really all of it, the entire conference, with this much contact, also with the student groups. And I think at some early stage, I guess yesterday, I talked to someone about that we have too few sort of playgrounds in science or for scientists or in academia where we can sort of have advanced discussions, but still on sort of a level where we are allowed to ask this sort of dumb questions or say that we don't know the words in that sort of a discipline. So I really think it is so rewarding to be part of a setting like this, and to exchange, to listen, and to have conversations, and to exchange opinions about such a broad concept. And like you were saying, in a way, this is how we should talk about insects, because they influence every part of our society. They do influence all this food security, the ethics, everything. So yeah, I think this conference really sets a good example of how we should talk about insects, include them in all sort of walks of life and all parts of society. So thank you for being part of this.
I like everyone has said, I really feel like I've learned a lot. And I think the thing that I learned, as I do whenever I've had the good fortune of speaking in an interdisciplinary setting like this, is how different our styles of presentation are. I actually find this fascinating because different schools, the way that we present data, the amount of data that we show, the way that we use the stage and the podium, the way that we talk about insects, the vocabulary that we use is slightly different, even though we might be talking about very similar things. So I've actually learned a lot just from my fellow presenters about science communication, and so thanks for sharing your different styles of presentation.
That's fun. That's great.
Absolutely. Absolutely.
I've commented a number of times about the ways in which TED Talks have transformed our process of selecting speakers, because many of you have done TED or TED-like talks. I did say to Jonathan, you've wrecked it for all future philosophers, because some of you may know that philosophers haven't always been among the most engaging presenters at the Nobel Conference. There, you heard me say that. And he has now wrecked it for future philosophers. The bar has been set differently. But that's absolutely true, so thank you. Thank you for that. Interesting. We often try to make there be at least some kind of a thread connecting a particular conference to the next conference. And interestingly, that was one of the reasons that we chose to end our conference with someone who does research on the fruit fly, who has done research on circadian rhythms, because next year's conference, and I have to say, I came up with this title, I'm very proud of it, Sleep Unraveled. All you Shakespeare scholars in the room will get the joke. And just a nod to what people were saying about the diversity, the breadth of disciplines that will be represented here. We have psychologists, we have neuroscientists, we have cultural historians, and we have someone with degrees in performance art and theology who describes herself as the Knapp Bishop and has founded the Knapp Ministry. So it is going to be a conference that is going to transform, and she has a very serious social justice agenda connected with that. It's going to blow your minds, and you're going to realize that you all deserve 9 hours a night. So that happens next year, October 1 and 2. Please mark your calendars now. It's going to be dynamite. It's going to be really spectacular. So drive safely home and get a good night's sleep and dream of insects.
Well, oh my gosh, you just caught us while we're sitting here eating crickets. Thank you again for joining us for the 59th Nobel Lectures on Insects, Little Body, Big Impact. This was such a great conference, and strangely delicious bugs. This last discussion, I think, really wrapped up a whole lot of big ideas here. And I think that, like, one of the questions I sort of came away with that I want to ask you, Amy, because you have, like, you're a scientist, right? Mm-hmm. I'm sort of curious about how having a conference like this that ties together science and philosophy and some of these big ideas, how that sort of affects scientific research at Gustavus Adolphus the rest of the year. You know, does that does having this conference every year add something to the way that you do science?
I think that it does. When I think about science, I think about it just being sort of one way that we understand the world. And I think this conference showed us that there's so much more richness when you incorporate, ethics, philosophy, history, all of these other disciplines that can add to what we would think about as sort of just an experimental approach, right? That's only going to give you a suite of certain answers. But this makes the experience so much more rich.
Yeah, it seems like it would. And I really loved what Siganet Kelamu was saying about finding that thing that I'm learning new this week. And I feel like this conference did that for me. I hope it did that for you.
Yes.
I'm assuming it did that for the rest of you as well.
Even listening to the discussion about the different presentation styles, I know that we get that question a lot in my field of communication studies about how do I present scientific information. So I think one of the things that's the beauty of this conference is it engages the entire campus beyond just the kind of particular disciplinary focus of the conference. So all of our students are kind of raising those types of questions.
Well, and you're here to talk a little bit about the particular disciplinary focus for next year.
That's right, Nobel 60. I'm going to violate
Nobel 60.
a fundamental rule of
There it
public
is.
speaking by bringing up a visual aid.
There it is.
It's bigger than I can deal with.
Sleep unraveled.
Sleep unraveled. And we're just really excited about the conference next year. I realized this summer that I'm the same age as the conference, so really.
So maybe you need a nap, is what you're saying.
That's right. That's right. But I think it's going to be a terrific conference.
Well, where did the sleep topic come from? What got this put up as the thing that's going to matter?
I think part of it is that just sleep is at the center of so many important conversations, so many aspects of our physical health, our mental health, our ability to process emotions and recall learning. And so when we started talking about, how do we construct a conference from a bunch of different angles, that was something that was always with us. I thought it's the one truly unifying experience. And for me, at least, why I got interested in it, Lisa asked me to think about serving as the host for the conference, was there's a great story of John Bell Hood's army fleeing Atlanta in haste. And of course, you can only be awake for a certain amount of time. There's a story of these soldiers literally running for their lives and falling asleep while they're marching and waking when they hit the ground. And I thought about that as a capstone experience for this truly unifying piece that unites us all. And that got me thinking about some of the subtopics.
What are some of these subtopics? Can you give us a little bit of a hint?
Sure. So we are thinking about kind of how to grapple with sleep architecture and how to define sleep.
What is sleep architecture?
So if you think about different animal species will sleep differently, right? So it may seem as if a shark is always awake. Well, that's because the way that their brains work, half of their brain will experience the type of sleep that we would relate to as humans in terms of brainwave patterns and so forth, and half of it won't. So there's a whole series of questions that arise when you think about how do we define sleep? Is it a physiological process? Is it a neurological process? Is it a prolonged state of simply lack of response to stimuli? And so even that kind of basic fundamental question is something that's really interesting to look at.
Yeah.
There were definitely connections to some of the things that arose at this conference in terms of circadian rhythms, sleep as a regulatory question. What is the I was thinking about the perennial questions that arise about should your teenagers be waking up at 6 a.m. and getting on the bus at 7, or should, it is true that teenagers sleep in the aging process. We do sleep differently, and we have different requirements for what constitutes good sleep.
What time should a professor start a class versus what time should a student start a class? Might be different things.
And you know, there's so much introspection because I wake up every morning at 4, and I'm dry. And you know, so when I've been thinking about this, I'm like, oh, a lot of my sleep practices are deeply unhealthy in ways I never thought about. So I think this conference will be something that everyone will be able to relate to some aspect of what it is that we're talking about, whether it be obstacles to sleep, whether it be they disease-related or age-related or noise-related, light-related, or you know, the whole question of what constitutes quality sleep.
Yeah.
And you know, a lot of people think, well, I missed a lot of sleep, maybe I can make it up.
Right.
And so I think there will be something in it for everyone. I noticed that Lisa had referred to some of the folks that we're inviting are really unusual. So maybe I should say a little bit.
I would love to hear about that. I noticed that somebody works for something called the Nap Ministry.
That's exactly what I was
which
thinking. Yes.
I am very into. Like that sounds like the kind of musical retirement home I want to be a part of this ministry, but naps.
That is Tricia Hersey, and she's an American poet and performance artist. And she examines these questions related to social justice work. And if you think about the 24-hour economy, right, we really are asking ourselves questions about who has the right to quality sleep, who has to work against those circadian rhythms to keep that functioning, who has to sleep in a noisy environment or an environment that's too hot. We have Mariska Brown coming from the National Institutes of Health. She's the director of the National Center for Sleep Disorders. A lot of her research looks at the role, I'm going to make sure I get this right, the role that circadian biology plays in heart, lung, and blood disorders. So again, a connection with this year's conference theme, thinking about, we do have, I think it's Dr. Young that said, we have this little fly inside us. I love that description because we really do have the same types of responses to those natural rhythm patterns. Mary Cascade is a professor of psychiatry at Brown University and the director of the E.P. Bradley Hospital Center for Sleep and Circadian Rhythms and Child and Adolescent Health. And so again, getting at some of those questions about if I'm focused on cognition and learning, memory consolidation, what is the best way to do that and when is the best time? Maiken Niedergaard at the University of Rochester School of Medicine is the co-director of the Center for Translational Neuromedicine. She is an expert on the neurological and physiological process of sleep and the relation between sleep disorders and a variety of other medical conditions. So again, I think a lot of people can relate to that aspect of it.
For sure.
Benjamin Rees is maybe the most interesting one for those who are non-scientists. He's a professor of English at Emory University and the author of Wild Nights, How Taming Sleep Created Our Restless World. And he uses literature and history to talk about the sociology of sleep.
Oh, so probably getting into some of that stuff about how like people used to have a middle of the night time.
The night watch. And
I
how,
can't wait to hear
you know,
about
and how we have evolved from a notion of good sleep being a solitary experience, whereas previously it would have been thought, no, we sleep collectively, we sleep as a group for, you know, protection or for cultural reasons. And so his work is really interesting and again also gets at some of that question of social justice.
Yes, that sounds amazing.
Amita Sahel is a neuroscientist and molecular biologist at the University of Penn's Perelman School of Medicine. Her focus is to understand the cellular basis of sleep behavior, what's happening in our cells as they're refreshing and replenishing, and that's kind of the focus of her research. And then last but certainly not least, I did these roughly in alphabetical, or at least that's how I had them written down, but Robert Stickgold, he's a professor of psychiatry at Harvard Medical School and director of the Center for Sleep and Cognition at Beth Israel Deaconess Medical Center, and his work examines the nature and functions of sleep and dreams from a cognitive neuroscience perspective.
This all sounds so fascinating. And I want to ask you, because you've been putting this together, what is the big thing that you really hope to come away learning this next year? Because I think it's really easy for something like sleep to just get into like, oh my God, all the ways that it's bad.
Right.
Is there like an answer that you're searching for? Is there something that you're going to be looking forward to learning that might make you feel uplifted?
I think As I've been doing the kind of research reading for each of these folks as we were kind of inviting them and introducing ourselves to their lines of work, the thing that's been most exciting to me is the type of interdisciplinary dialogue that could be at the end of, just like at the end of this conference, because I don't think that there are that many opportunities for people from such completely different vantage points to come together and have a high-level conversation. So that's what I'm really looking for.
That's where you really learn stuff. I mean, I think in my history as a science journalist, one of the most interesting things has been the times that I have been working on a story, interviewed somebody in one specialty, interviewed someone else in another specialty, and mentioned what this other person had told me, and they were like, oh my God, really? What's their name? How do I get in touch with them? Because people just don't know. They don't know what's outside of their own little bubble. And this is such a great opportunity to learn about all of the things outside our bubbles.
I will say the single most surprising thing I learned was last summer I read a piece about a substantial portion of the weight gain of first-year students is because of the change in their sleep patterns.
Oh my gosh.
If anyone had told me I could sleep my way to fitness, I would have definitely done that rather than waking up at 5 in the morning to spend 40 minutes or so
on the treadmill or whatever. Yeah,
I'm
yeah. Go take a nap.
only known. So you heard it from me. You can sleep your way to wellness. That's my main takeaway.
Feel less crappy.
Amazing.
Get better sleep. Lose weight. Sounds great. I love it. Wonderful. Well, thank you. Thank you so very much for coming and telling us about what's coming next year. And thank you also to everyone who puts on this conference. I know it is just a huge endeavor. There are so many people involved from top to bottom. We have volunteers, we have students, we have professors, we have speakers. Just everyone that comes in and is a part of this deserves such a huge round of applause for all of the work that they put into making the Nobel conferences happen. And also a reminder to everybody that the videos that you saw on this feed this week, those are all going to be available online on the website. You can watch the whole thing again. You can learn more stuff. You can see the stuff that you missed. That is all available. Do either of you have any closing thoughts before we end?
No, I just want to thank people for joining us online, if they had joined us online. And just remember, go out, enjoy nature, look at some bugs.
Go find the coolest bugs you can today and tell us about them.
Exactly.
Send us a tweet, send us a text message, send the university an email. Show us your bug pictures.
Thank you both so much.
Thank you, and thank you. And have a lovely rest of your autumn.
So many of the different fields in science, most people aren't exposed to in their day-to-day life. The more you can get out there and communicate to the young people coming up that are the future of science, the better. You never know what's going to grab one individual, and so that's vital to inspire the next generation.
The Nobel Conference brings together students, faculty, and members of the general public to explore revolutionary, transformative, and pressing scientific questions and the ethical issues that arise alongside of them. We bring leading researchers to Gustavus, including nearly 100 Nobel laureates. We're trying to explore the ideas of science using all of the liberal arts, so the arts, the humanities, the social sciences, all of them working together.
There are a lot of people who are not scientists who like to share in the adventure of discovery, and to be able to share that joy with people who also get it, that there's something really exciting and cool going on. It just makes it just an enormous amount of fun.
There's sort of a festive atmosphere, which makes it part of a broader experience.
there aren't that many forums for us to have the discourse at that level with that sort of range of expertise, yet still focused on one sort of overarching topic.
One of the wonderful things about the Nobel Conference is it does give people the opportunity to ask people questions in an informal way. And you never know what statement somebody makes or what interaction that happens will make a change in somebody's life.
It's always very, very encouraging to see other colleagues of yours who are at the top of their game, and it makes you want to lift your game.
As you can feel the audience, and you can really feel when you've helped people to know something that they never knew before. It's really a feeling of discovery that you share.